Yes. Good morning, everybody. So I hope that you are hearing me.
Yeah.
Hello. So, sorry for missing the meeting, for not being there physically. I wish I was there to attend the meeting with you guys, but unfortunately, I'm presenting from Ibadan. Let me try to. Oh, I cannot share my screen. Oh no. Apparently, I cannot share my screen. Maybe the presentation I sent can be displayed. Hello?
Yeah.
Apparently, okay, it's working. Yeah. Are you seeing my slide?
Yeah.
Hello?
Yes.
Can you see my slide now?
Yes, it's coming. Yes.
Okay, great. Yes. So good morning, everybody. I hope that everybody had a good night and a good breakfast. So I would like to tell you what we are doing here in Genetic Resources Center of IITA to support the yam breeding and AfricaYam also. So when we are talking about supporting yam breeding from genetic resources, we start with the international collection of yam that we are keeping at the Genetic Resources Center. And there is no need for me to highlight again to remind you how important the genetic diversity of yam is critical for crop improvement, especially in this era of climate change, and also with the recurrent target of increasing yield. So definitely genetic resources of yam is a vital resources, is a vital asset for the crop improvement. So here in the gene bank in IITA, so just to remind you that we are having now more than 6,000 accession of yam that we are conserving here, that is available and accessible for, especially for crop improvement. And from that collection, we have the defined core collection that is including all the 9 species that we are having here. And you can see in bold the wild species that we are conserving here in the Genetic Resources Center. And this collection is conserved in 3 conservation systems. So in a kind of integrated conservation strategy, I can say. So the collection is basically conserved on the field, where you can see where we have the entire collection, and the collection is also conserved in vitro using slow growth conditions. You can see the number of accessions that are already in the in vitro conservation system. And one of the advantages of the in vitro conservation system is the opportunity we can have to clean the germplasm, and you can see the proportion of in vitro collection that have been declared clean. And yam cryobanking also is implemented in Genetic Resources Center. So I can just give an indicative number of 10 accessions that are already into the cryobank. So it's not yet fully routine, but we are yet to have a big boost on this on this cryobanking of the yam collection. So all this conserved collection is available and accessible for pre-breeding and in particular, in general crop improvement. So yesterday I missed in the discussion the point about how the breeding programs from all the partners of AfricaYam, how are they tapping into this diversity that is kept in genetic resources center. So I guess we can have some exchange about that, and it will be really important that this diversity is used for the benefit of the improvement of yam. So of course, all this one, as I told you, is conserved in active collections and long-term collection with the cryobank, which is just starting, I can say, this year in 2022. So big part of our work on this collection is the cleaning. So we are trying our best to remove to make them safe from pathogens, especially the viruses. And of course, we have the opportunity to have a high rate multiplication from the in vitro system that can feed the SAH or even beyond. And the possibility we have to distribute all this collection for use, including, and this is very important, including the compliance to all the regulations, especially the phytosanitary ones, which are very important when we talk about germplasm exchange. So that is the core collection. The costed collection. But now, in terms of breeders collection, so we are also keeping for the breeding team the collection into the in vitro gene bank. So you can see from, since 2018, how the number of accession have been increasing. So as at last year, 2021, so we had almost, a little bit less than 1,000 accession conserved for the breeders in the in vitro conservation system. So I just give a brief breakdown of also, the general conservation. The other collections that was sent from the MAF project, from GIRCAS. We have some land races also. We have what we call the old breeders line. These are the accessions that have been kept since more than 10, 15 years, I can say. We have some accession also that came from the project funded by the Minister of Foreign Affairs, I think, of Japan, 43 accession, and some accession kept from the ploidy work that was done in bioscience. So all this collection, both the costed collection and the breeders collection, are going through the same handling, I can say, especially in terms of virus cleaning. So I just want to give you a quick summary of what we are doing in terms of virus cleaning. So we are trying to clean all this germplasm for in vitro conservation and also for the cryobank. Definitely, you can understand that it will also be useful for the breeders in terms of distribution, multiplication and distribution. So the first method we are using here to clean the germplasm is meristem culture, but as you know, yam is not very responsive to that method because it's very difficult. The rate of cleaning is very low when you use only the meristem culture system. It's quite suboptimal. That's why we have associated with this meristem culture other cleaning methods, which are using heat, chemicals, or cryo treatment. So doing all these tests, we were able to have quite good results, and we are about to use them routinely. Actually, we are already using them for some urgent work, but routinely we are going to include them in the protocol soonest, and it will be used to have a higher proportion of the germplasm conserved that are clean from viruses. But as you may know, facing all these difficulties to clean the viruses from yam, we still have a big part of the collections, both for the costed collection and the breeders collection, that are not yet treated, not yet clean, not yet, so consequently, not yet available. So in terms of heat treatment, for example, we have capitalized on the work that have been published by the ISWA colleague in 2017 using heat treatment. So we have tested many accessions, not using, using more temperature than what was used by our colleague from ISWA. So we use 30, 34, 36, and 38 degrees Celsius. And this temperature was applied on the in vitro plantlet for 3 weeks before we test them. So all treated accession have been tested from in vitro material, and also after hardening, because it's very important. You will see it later that it's not because they are clean from in vitro samples that they are confirmed clean once they are put on the field, or on the screenhouse or on the field. So from this work, from this kind of reproduction of the work published by the ISWA colleagues, so we observed that the better results in terms of virus cleaning was obtained when the material was treated with 34 degrees centigrade. So in terms of cryotherapy, so we have also done some work using the true cryogenic method, which are mostly used in GRC, droplet vitrification, and V-cryoplate. So we have tested 3 Diascorea rotundata accessions and 3 alata accessions. And in the same way, we did, and we indexed treated material from the in vitro samples, and also once they have been hardened in the screenhouse. So for now, the results are quite, let's say, scanty. So it was difficult to follow, especially after the COVID disturbance. So it was difficult to get all the results. So the results was quite scanty. So that's why we don't have a very consistent conclusion we can give from that. But I can say that it's promising, and it may be interesting to try. To have a better experimentation on that, to have a complete conclusion on that. But because of the missing data, we cannot conclude really on cryotherapy of yam. This is a quick view of what have been done in the frame of the BMGF funded project to set up a better cleaning method for yam, among other crops for yam, and also a more accurate indexing system using SRSA. So the work we have done for the chemo treatment, so I make a mistake because this should not be there. Anyway, so we have used 3 antiviral agents, so ribavirin, lactoferrin, and salicylic acid at different doses. And when we apply that to the yam in vitro plantlet, so we have observed the effect of this antiviral agent in the virus status of those material, indexing them, testing the virus status from in vitro material and hardened material, as usual. And we were able to observe very good results from this antiviral agent, especially from ribavirin and lactoferrin. So it was quite, clear. The only problem is that, as usual for yam, it was observed quite a bit variation between the genotypes, and also sometimes it's not the same dose of the antiviral agent that is effective in terms of virus cleaning. So we have validated this work, and we are trying to see how we can include it in terms of type of antiviral agent and dose, how we can include it in the routine work. But we are already using it to clean some material, especially those that are going for the cryobank, and if necessary for those that the breeders want us to distribute, to send out of the borders. So in conclusion, we can say that the yam virus cleaning system in GRC is starting to work. I can say we have some good results, but it's not yet straightforward. So we still play with the, for example, for the chemo treatment with the antiviral agent and the doses. Really heat treatment and chemo treatment was able to give quite good results. So I mean, the future is quite bright in that way. And there was something that we have observed also was that it's very beneficial to see also when you are sampling, when you want to get clean material, especially, I mean, for example, when you want to establish the material from the field into the in vitro collection. So usually it's beneficial to try to sample wine that are not having sign of disease. So even they may have the virus inside, but so far they don't have symptoms. Usually those material are easier to clean once they get into the in vitro collection system. Anyway, combining all this efforts to clean the material, so we were able to have some clean material from the breeders collection, I can say. If you can see in, I mean, for the breeders collection in general, so we have 34 accessions that are confirmed clean from the second indexing. As I said, we usually, I mean, we routinely need to index twice the yam material to make sure that they are confirmed clean from in vitro and after indexing. So from the 160 clean material in vitro, once we acclimatize them and index them again, only 34 was confirmed clean. So you see the gap between. So just to tell you, I mean, how necessary it is to go through this step. And for AfricaYam, we have 26. So we have added over the last year 8 accession clean. And in total, we have 6 accession that are clean from all the breeders' collections. So this is showing also how important will be the routine use of the new indexing method that has been developed in the frame of that BMGF funded project that is using SRSA. So maybe Dr. Laba will have the opportunity to explain a little bit more in detail how, I mean, what it is about, but at least that indexing method that is using sequencing tool is helping us to not have to not have to do this second indexing. So only from the in vitro material, we are able to have a very accurate indexing results. So you can see the gain of time. It will be gain of time, resources, and effort. That will be once we will start the routine implementation of this indexing using SRSA. In terms of distribution, so in 2020, I mean, since 2020, so I don't think that we have any distribution from this breeders collection. So, but anyway, that is depending on the breeders. So we are distributing based on their requests, on their needs. So I guess COVID-19 was one of the major causes, but also maybe there was not urgent need for them to take from the collection we are keeping for them in GRC. So after saying that, so these are the points we are working on. I mean, work is already on them, but just to tell you that we will keep on putting a lot of effort on that. For example, the establishment of the lines that the breeders want us to keep for them. So I want to highlight, I mean, this aspect because also from the BMGF funded project, we have improved the yam material establishment into the in vitro collection. So we have now showed the importance of adding active charcoal in the meristem regeneration culture medium. So the optimized establishment, in vitro establishment is used now routinely. So that is very good for the establishment. In terms of cleaning, of course, we will keep on putting a lot of effort to make sure that we have a better cleaning protocol, routine protocol for yam material. So of course, including AfricaYam germplasm. And once all this optimized protocol in terms of cleaning, in terms of indexing will be routinely implemented. So I feel that we will have a great step forward. So they are actually being used. So I can say that they are already routinely used. It's just that we have not yet get the improvement in. I mean, at least for the. I mean, since this report is based on the 2021 work. I just want to remind. Maybe you are aware, but we are doing a lot of work also to make sure that the collection we are keeping can be more useful to yam breeding. So we are going through some genotyping. So this is just a quick summary of what has been done. So 966 yam accession have been genotyped, including wild, inbreed lines, and land races using that sequencing, and also quality control. So the accession genotype was able to be grouped into 9 different yam. Was from the 9 yam species. So you see the filtering that was done. In summary, so the conclusion was that the diversity of yam collection have been confirmed from this genotyping, and we have seen that wild relatives was quite close to, was closer to Rotundata than Alata. And you see the grouping, and the heterozygosity proportion was higher in the wild species Bryancelis, followed by Rotundata and Alata populations. So in the future is even already ongoing. So we want to use this sequencing data for routine identification of unique and true to type material, mainly for cryobank and quality control, but also breeders can benefit from that. And also this sequencing, this genotyping data can be used for gen discovery and GWAS and so on. That's why, there is a lot of work that is being done by my colleague Rajesh Paliwal, who also show that, we can say that all this genotyping data can be useful for allele mining and threat, for useful threat in breeding. So if you want more information on this point, you can liaise with my colleague Rajesh Paliwal, who is in charge of this aspect of GRC work. So these are some other research that are happening in GRC for yam. So of course, I told about the yam cleaning. We are about to start some work on the epigenetic variation on yam once, when they are conserved in the field, in vitro and cryobank. So in collaboration with colleagues from bioscience, Livia, Dr. Livia Stavolon and Andreas Diesel. So this is an advanced way of assessing the methylation of the germplasm. So we have optimized the yam cryo protocol, but is still there is still room to have it better optimized. So we are working, but at least as it is now, the yam cryo protocol is quite responsive for yam cryo banking, and we are planning to adapt the method that was developed from potato using mini-tuber from in vitro to clean and to cryobank yam material. So the work is ongoing. We are at the stage of producing the yam microtuber. So that maybe can help in terms of virus cleaning and also in terms of cryobanking. And this is just some work we are doing in collaboration with the Faculty of Pharmacy of University of Ibadan, trying to characterize the anti-diabetic properties of Dioscorea dumatorum using genetic, agromorphological, biochemistry, and transcriptomics analysis. So to make sure that we have a better characterization of this pharmaceutical property of yam. And in the same logic with this work, there is a workshop that will be organized in November So I use this to advertise it and announce it. So you have some colleagues from yam breeding who will be giving some important keynote and speak on that. So we have Dr. Asrat, of course Rajnish also, and Ranjana, who will be giving some talk on that aspect. So of course the collaborators coming from Germany and from France will be also invited to give some talk, and there will be a hand-on training on the transcriptomics analysis for students that will be carried out in bioscience in Ibadan. So that's it. So I hope that I was not too long. So thank you very much, and I wouldn't mind responding to some questions, and I will welcome some suggestions and advices. Thank you very much.
Okay, thank you very much, Dr. Gay, for your good presentation on the good work you are doing at the Genetic Research Center. Because of time, we will take only 1 or 2 questions, and then we will hear Dr. Teraoshi from Japan. Question, yes?
Thank you, Badara. Very, very good presentation.
Thank you.
One question in terms of a gene pool collection, is there any thought to keeping true seed collection in addition to your clonal collection? Because it would be possible to generate seed, and I don't know the storage life of yam seed, but presumably it's fairly long.
The same
Yes.
with the same with pollen. I was going to ask the same.
Exactly. Actually, we are even looking at that possibility, especially for the wild varieties, because conserving them using the clonal material is not very optimal, I can say, to conserve all the diversity from the wild. But it has been published very recently, I mean, some pollen conservation protocols that can be adapted to yam, and also for seed conservation. I mean, we can even conserve them, I mean, using normal seed conservation, I can say, but also there are some seed cryo protocol that can be adapted to yam. And it's very possible. We are just waiting for the interest, because at least from the Genetic Resources Center, the point is to start working on it for the wild varieties. But if the breeders are interested in that, we can have a bigger emphasis on that and finish adapting the protocol that are now quite well established for other crops. So definitely that is something that can be emphasized on, and we are ready to work with the breeders to conserve them. And why not? Yes, it's possible.
Dr. Way, this is Carlos Iglesias from NC State University, and I'm very, very interested in pollen preservation. In fact, earlier on in this project, we discussed some alternative breeding schemes in which instead of exchanging clones, we exchange pollen. Okay. A question, a couple of questions actually. Are there any efforts to bring some of the breeders' lines from national programs? That's first. How close to saturation are you in terms of the demand and the work that you do and the budget that you have and the resources? And the third one, I'm very, very interested in introducing a few materials into the US so we can proceed to do studies in flowering, dormancy. That's why I was very interested in talking to you. I wish you were here, but I guess we're going to have to communicate through Zoom. How feasible do you think that is?
Frankly, we have not been working effectively on the pollen for now. I just know that there are some very new reports that was out there in terms of pollen conservation, but I know that it is very possible to adapt it on yam, and that can be useful. And as you say, I wouldn't mind starting on that, and that can be very interesting. And I know that is something that can be very beneficial in terms of germplasm exchange, in terms of also long-term conservation, because they can also be conserved in the cryobank. So yes, it's a pity that I'm not there for this session, but I'm reachable and we can have further discussion on that. Definitely.
Okay, Dr. Gay, I think because of time, we'll stop there and go to Japan to hear Dr. Teraoshi.
Oh, I have not picked my passport. Let me pick my passport for Japan trip.
Okay.
Thank you very much, Dr. Dancy. Can you hear me?
Yeah.
Hello?
Yes, we can hear you.
And how about this slide?
Yeah, it's okay. Yeah.
Okay, thank you very much, Dr. Dancy, and thank you very much for all the members of the AfricaYam community. And I'm sorry that I can't make it to come to Abuja to meet all of you. And it is because of some stupid restriction of our government to get back to Japan. And so anyway, I'm very happy to be able to hear all the nice talk of my colleagues. And taking this 30 minutes time, I would like to share our update of AfricaYam Phase 2 project. So I'm going to present this slide on behalf of all the members of IBRC, IITA, Japan, and also Kyoto University, and colleagues in IITA, and also GEOCAS people as well. So the mission we have been assigned is enumerated here. 5 articles given. And the first one is identify QTLs using mapping population and GWAS panel. And second one is to elucidate sex expression system of diaspora protein data. The third one is do the RNA sequencing to understand the regulatory network of important traits. And the item number 4 is the exploration of the genetic diversity of Guinea yam wild relatives. And the final one is the genomic prediction. And due to the COVID-19, we couldn't come to Africa. And because of that, some of the kind of missions could not be pursued in much depth. But today I want to share our recent development about the sex determination system understanding. Before going to that, just briefly, I mentioned about the phase 2 kind of achievement. And the left side is using mapping population to do the linkage analysis of some trait, including tuber color, using one segregating population, MP2, comprising 156 F1 progeny, and we could map the QTL, which determines the tuber color. On the right side, we have the GWAS data of stem numbers, and so with more than 300 accessions, this GWAS can be achieved. And I don't need to go into detail. Yesterday, we had a lot of development headed by IITA and AGRI, explained about the huge capacity of this genome-based QTL isolation. And I'm very happy to hear about that. And I just briefly touch on the wild relative of Guinea yam. On the left side, you see the picture of the leaves of wild relatives of Guinea yam. And so the leftmost one is Togoensis and Abyssinica. Abyssinica is this one. And the second picture is Minutiflora and Liplexiana. Third one is Prahensilis, this shining leaf, and Smilasifolia. So what I want to convince you is that these wild relative species are distributed in the same habitats. And due to the dioecy, which is male and female flowers born on separate individual, this implies that they can undergo frequent outcrossing and hybridization. So this is the message. And so in this 2020 paper, we published in PNS together with IIT people, we proposed 3 hypotheses. And you can see here, this blue one is rotundata, which is cultivated Guinea yam. And the species indicated by green color is Abyssinica, mainly distributed in savanna region of sub-Saharan Africa. And this Prihensilis, indicated by magenta color, this is mainly distributed in rainforest. And we could propose 3 different hypotheses. Rotundata is more closely related to Abyssinica. This is the first hypothesis. Second hypothesis is Guinea yam rotundata is derived from Prihensilis of rainy forest. And the third hypothesis is rotundata is indeed hybrid between Abyssinica and the Prihensilis. And our genome analysis concluded that this hypothesis 3 has the highest likelihood. And so this has a very strong implication, as you can see here on the right side. This is the so you don't need to go into detail about the scheme, but what I want to draw your attention is this number n years. This is the effective population size, and the square indicated by red color. This is Dioscorea rotundata, Guinea yam effective size. If you take this as 0.09, if you compare this value with effective size of Abyssinica, NA, this is 0.51. Effective size of Prihensilis is 0.38. So the Guinea yam cultivated, Guinea yam genetic diversity is far less than the genetic diversity of its progenitor, Abyssinica and Prihensilis. So we have an ample capability to exploit and tap the genetic diversity of these 2 wild progenitor of Guinea yam. So I think this is a very important, project to pursue further to exploit the genetic diversity of wild relatives to enhance Guinea yam breeding in the future. Okay, taking the rest of time, I want to focus and share with you about our recent finding about the sex expression of Guinea yam. And this work has been mainly done by Aoi Kudo, shown here, and Yu Sugihara. They are both Kyoto University students, and they are very much talented people for genome analysis. And we have published 2021 this review article, Population Genomics of Yams. And in this article, this is freely available, open access. So we made a kind of overview of reports about the sex determination of Dioscoriaceae. As you know, the most, I think I can't tell all, but the majority of Dioscorea species are dioecy, which means male and female flowers are on different individual. And as you see here, the majority of the reports so far available are XY type male heterogametic sex determination system. But sporadically, you see ZW or XO type sex determination system. And in particular, Dioscorea rotundata corresponding to Guinea yam, we showed that this sex determination system is ZW. So this is a kind of anomaly. And so we are interested in this, how this ZW kind of sex determination system is operated. So this is a summary of what we know. And the majority of Dioscorea is XY type sex determination system. And this section, Stenophora, including Dioscorea tokoro, this is a very old kind of lineage of Dioscorea. And we showed that this sex determination is XY type male heterogametic system. And if you go to the Enantiophyllum containing majority of edible yams, including Arata and Guinea yam, and this Enantiophyllum Arata is XY type, and only Rotundata, Guinea yam, is Z-dub type female heterogametic system. So this is very strange, and we have to elucidate, how this kind of sex determination system has been evolved and switched. So our current understanding is probably the ancestral state of Dioscorea had XY type system, but on the some time point of evolution, this system has been converted to Z2W in Guinea yam lineage. In the paper of 2017, we published the genome sequence of Dioscorea rotundata, and this is Tamil and others. And in this paper, we described the sex determination system of rotundata as Z2W. And as you see here, Z2W is human and ZZ is male. But this is not a very stable manifestation of the phenotype. Usually, the ZZ genotype are mostly male plants, and there is no variation too much. But even though the genotype is Z2W, the plant is majority of cases this is a female, but sometimes you see the male flower and also monoecious, which means that in the same inflorescence you see the male and the female flower. So this is a very unstable, manifestation of Z2W genotype. So we are now addressing this system. In order to understand this system, so we set up the experiment. Beforehand, I just wrap up our understanding. So male is Z2Z kind of homozygous state in chromosome 11. So chromosome 11 containing the sex determination locus. And if this locus is homozygous with Z2Z, it becomes male flower, and it is quite stable. But if this locus is Z2W, so in principle, the plant becomes female, but sometimes they are monoecious or male. And our hypothesis is like this. So this locus in female is G2W, like this. Maybe some differentiation between the chromosomal structure. And we hypothesized this W chromosome contains some gene or factor which determine the femaleness. So our hypothesis is like this. Maybe on this W chromosome segment, we have a gene or a microRNA which suppress the expression of maleness. And maleness is gone and the plant becomes female. But in the case of this factor has been abrogated, then the male flower can be manifested. So because of instability of this factor, the Z2W plant become female or male or monoecious. So this is our hypothesis. In order to verify this hypothesis, we did this experiment. First, we selected the genes or microRNA specifically expressed in female flowers, not in male flowers. Second, we tried to locate this gene locus onto the chromosome 11 sex determination gene segment. And for this purpose, we tried to make a much better reference genome of Guinea yam using TDR96F1, which has been published in 2017, but they are very much fragmented. So we tried to make a much better reference, version 3. So using these three approaches, we try to address this question. Okay, first, RNA-seq and microRNA-seq. So we collaborated with Dr. Iseki of Jirukas and the IIT people. We focused on the flower, in the male flower and the female flower. Stage 1 is a primordial stage with very small kind of, the tissues which may develop into the fluorescence. Stage 2 is a half, developed inflorescence, and stage 3 is a fully developed inflorescence. And for these 3 stages for male and female, we made the triplicates of the library, and we did RNA-seq and the small RNA-seq. And we compared the expression of the genes between male and female flower, and the left top is RNA-seq data, and we focused on the genes which are specifically or highly expressed in female flower only. And the number of such transcript was 558. And the bottom one is microRNA, and we found 130 microRNA which are highly expressed in female but not in male fluorescence. So we confirmed the position of the sex determination locus by using MP2 segregating population. And as we reported before, this chromosome 11, the end of chromosome 11 had a position which shows high association with the sex of the progeny. And so the distal part of this chromosome 11 indeed contains the genetic factor which determines sex of the progeny. As I said, we selected more than 500 transcripts that are female biased and 130 microRNA female biased. We tried to find the gene or microRNA located, positioned on this chromosome 11 distal part, and we found 3 genes are located in that region and 49 microRNA candidates. But after confirming by mapping the parental DNA short reads of female and male parent, and there was no transcript which was specifically mapped by female short reads. So we tentatively forget about RNA, protein coding RNA, and we switched to focusing on microRNA, 49 microRNAs, which are specifically located on this chromosome 11 tip and expressed highly in female. And to make the analysis much better, we developed the version 3 reference genome using nanopore long read. And the result is summarized here. This version 2 is the previous one, and the version 3 is the latest one. As you see here, the numbers of contigs have drastically decreased, and the size of longest contig also increased, and the Busco value was also increased. So I'm sure that this version 3 is now the best reference genome available, and we will share this data as soon as possible. If you focus on chromosome 11 part, which contains the sex determination factor, you see the version 3 and the version 2 dot plot is very much disformed. So it indicates that our latest version gave a very much resolution of this region. And in the previous work, we identified a DNA marker named SP16, which is located on this W region specific to female plant, and it is located here. So this red one is the coverage by female plant, and the blue one is the coverage by male plant. And as you see here, SP16 marker is located in this female specific region. But as I said, this reference was not of very high resolution. And using this version 3, we identified one contig named contig 216, which corresponds to this SP16 DNA marker. And the size of this contig is 0.6 megabase. And so this graph shows the depth of the short reads from female bulk on the top side and the male bulk on the bottom side. And within this contig 216, again, the left side distal part is covered, predominantly by female bulk, but not by male bulk. And SP16 location is here. So we suspected this region is very important for, for sex determination. So further, magnifying this region, so we focused on this 0.2 megabase region of contig 216, and this is the region. And you see this blue one, blue coded bar. This is a region covered by only female weeds, and this white part is the region covered by both female and male weeds. So, and SP6 is indeed located on female only region. So we tried to identify microRNA, which is located on this blue coded region. And indeed, after selecting 49, the number of microRNA residing on this female specific region of chromosome 11, contig 216, was only 13. So these are the list of this microRNA coding microRNAs, and among them, we focused on this, microRNA 708. So this was, found only in female flower, and we focused on this, column here indicated. So this, microRNA locus of contig 216 has a very perfect structure of precursor microRNA. So this is capable of generating microRNA, and so other microRNA candidates did not have this kind of complete precursor structure. So I think as a first priority, we focused on this 708 as a strong candidate. And indeed, this 708 sequence, this was 24 base pair, had one target gene. So what was the target gene? So this target, polygalacturonase 1 beta-like protein 3 gene, which is residing on chromosome, I think, 20. And as you see here, it is a very good kind of good target. And if we look up the literature of the function of this polygalacturonase 1 beta-like protein, so the one paper of Salix, this is a willow, and this paper indicates that this gene is male flower specific gene. And another paper of cotton is so they found a male sterile cotton, and in this cotton this gene was mutated. So from this literature we can infer that this gene is very important for making pollen in male, and it is reasonable that microRNA targeting this gene abolish the development of maleness. And indeed, we compared the expression of this target gene in male and female fluorescence. As you see here, this is the expression level. And so the red bars indicate female flower, and blue bars indicate male flowers. And these are stage 1, 2, 3 is indicated by the number. As you see here, in female, stage 2, the expression of this polygalacturonase gene is highly suppressed. But in contrast, this gene is highly expressed in male flower. So from this result, we hypothesize that this microRNA may play a role in determining or suppressing maleness in female ZW plant. So this is the hypothesis, summing up our current observation. Maybe in ZW plant of chromosome 11, this microRNA 706 is generated. And in this plant, this microRNA target to the targeted genes, the polyglucosidase 1 beta-like protein 3 gene located on chromosome 20. And if this suppression happens properly, then plant will become female. But if this suppression is not complete, then plant may develop some pollen and show the male kind of structure. So this is still kind of preliminary result, and we don't think this is the only kind of sex determination factor, because there is a huge difference of the structure between male and female. It is not only the pollen development, but you see the elongation of fluorescence found in female, but not in male. So I think there should be other factors that may determine the sex. But our hypothesis is that these genes are showing, making a supergene with strong linkage disequilibrium in this W region. And our current endeavor is to identify these key factors located on this region. Practical perspective is that if we identify the very key genes, including this microRNA, then we may be able to exploit that knowledge to develop hermaphrodite plant or monoecious plant, which has the male and the female plant, female flower in the same individual. And so in the background of ZW genotype, usually the plant make a female flower, but by manipulation of this microRNA or some other genes, we can convert the flower into the male. And by crossing, doing self-pollination within the same individual, we may be able to get to the self seeds. By doing so, we can fix the desirable allele in very short time. So I think this is a prospect we can pursue further. Okay, so this is the end of our updates of sex determination system of IITA. The prospects. So as I said, we want to be able to manipulate IITA sex determination system to accelerate crossbreeding. Number two, we would like to extend this pangenomics, including wild relative of guinea yam, Dioscorea abyssinica, Dioscorea prahensilis, and I think focus on the disease resistance would be very important, because this phenotype is usually determined by a very small number of genes, and deployment of allele from wide genetic resource would be very, very effective. And I think this could be possible, and we may be able to focus on the gene of the category of MLR, which is a nucleotide binding leucine-rich repeat plant resistance genes. And I think using bioinformatics approach, we may be able to exploit an important resistance gene against a variety of disease of guinea yam. And number three is just a repetition of what I said. We have to broaden our focus of deployment of a genetic resource using wild relatives, and not only the alleles, but we have to focus on haplotype. And in this regard, this graphical haplotype graphic analysis is very important. Okay, so with that, I want to finish my presentation. I would like to extend my special thanks to all the AfricaYam community. I enjoyed very much working together with you. And what I explained today is the culmination of the work shown here. And thank you very much for your attention.
Okay. Okay, thanks so much, Dr. Terauchi, for your beautiful presentation on the important work you are doing to assist the breeders in understanding the sex issue in yam. We will take 2 questions because of time. 2 questions before the next presentation. So questions to Dr. Terauchi. No question? The presentation was so clear? Okay.
Hi, Dr. Terauchi. This is Carlos. Wish you were here.
Hello.
It's so stimulating to bounce ideas with you. You may have mentioned this, but what is the genetic determination of sex in the ancestors that you have clarified, Abyssinica, and I don't recall what the other one is?
Prahensilis.
Yeah.
I think no one has ever tested the sex determination system in these wild relatives. Maybe they have the XY type, but we still have to explore.
Okay. Another questions? Yes, that's fine.
Okay, thank you, Professor Terauchi, for your nice talk, and really inspired me. Based on the hypothesis you I think formulated on to reconstructing routine data from the wild relatives, we try to make progenies from Prahensilis and Abyssinica, but we have not confirmed their hybridity. We have sent the materials to genotyping, and hopefully we'll get it. But is there any like mechanism or maybe from the genome which we can see that we can or maybe able to generate synthetic routine data from those progenitors?
I think, yeah, maybe, the cross between Prahensilis and Abyssinica, between these 2 wild species might be not so easy. But if you can use rotundata as an intermediate kind of parent, so rotundata and Abyssinica cross might be much easier, or rotundata and Prahensilis cross might be much easier. So I think my recommendation is try to use the Guineam cultivar as one of the parent to cross with wild relatives. And
Dr. Terauchi, Carlos again. Interested to have your idea. This polygalacturonase gene, can we envision producing some sprayable RNAi or something that we can use on male flowers to stimulate female adult?
You are right. And if this microRNA is a determinant, I don't think so, but maybe it is doing some role. And assume that this is a determinant, then we can synthesize some small nucleotide which binds to microRNA, and we can inject kind of RNP, protein RNA complex, to the primordia of the flowers, and which has the complementary sequence with this microRNA and block the function. Then we can convert the ZW type female into male. This could be possible.
It's okay. Okay, thank you, Dr. Terauchi.
Thank you very much.
Thank you so much for the beautiful work you are doing. We are now going to Sirat to hear Dr. Denny Kone. Voilà. Thank you.
Thank you. We need to share it. Okay, good morning, everybody. My name is Denis Cornet. I will try to present what we've done last year at CIRAD. I will present with Komivi Dosa, my colleague, who will talk about the genetic part of the presentation because it's over my skills. So we contributed to the flagship 1 and 2. The first one to develop and transfer medium and high throughput method for agronomic and quality traits. And the second one to identify QTL and genes controlling these traits. We also contribute to the organization of the joint meeting, but as Eglantine will present it in detail later on, I will not talk about it. So in Guadeloupe, we have two pedoclimatically contrasted sites, and we use it to study one diversity panel of the ALATA. And we also have two mapping populations, but Komivi will detail it later on. So we focus a lot on phenotyping, and we try to encompass as much as possible the whole growing cycle of yam, starting from the germination in storage up to the final quality of the product within the RTB food project. So last year we made a huge phenotyping work thanks to our PhD student, Ezekiel Umbo. We started by observing, of course, the phenology of the crop, date of germination, emergence, senescence onset, and complete senescence. And I added this diapo because of yesterday's discussion about dormancy. So I just extract the data we had last year about dormancy length, defined as the time from the final senescence up to the germination in storage. And we can see that we have quite a large diversity of dormancy lengths, starting from a bit less than 2 months up to nearly 6 months. So this illustrates the potential, I think, for improving and managing this dormancy length, thanks to breeding. In a second time, we looked at the emergence dynamic. It was already highlighted in previous presentation. Heterogeneous emergence is a problem. It is a problem to manage breeding, but it is also a problem for the farmer dealing with weedings. The longer the Okay, thank you. So let's continue. We were talking about emergence dynamic and its importance for breeders, but also for farmers. So we made weekly observation. And from this weekly observation, we applied the log logistic time to event modeling in order to characterize the whole emergence dynamic. And we extract some phenotyping traits of interest, like steepness of emergence, emergence duration, and emergence rate. So we looked at the correlation between all these traits, and we found that the longer the latency, so it means the longer the time between planting and emergence, the longer the emergence duration as well. So it means when the genotype is good, it is good in all these traits related to emergence dynamic. So if it has a short latency, it will have a high steepness and a high maximum emergence rate. So this is a good news. There's no trade-off between these traits. After emergence, we look at characterization of the ground cover dynamic. The idea is to look at the vegetative growth, but also at the senescence dynamic, because we think that senescence dynamic play a great role in filling the tuber by the end of the season. And there is quite different behavior between genotypes in terms of senescence dynamic. So we are taking image every 10 days, and we applied then image analysis with segmentation and so on. And from this, we applied a beta function modeling in order to characterize this vegetative growth, the plateau, and then the decreasing due to the senescence. This allows us to extract precise estimation of maximum ground cover speed, senescence duration, and so on. Starting from last year, we also tested some theory coming from ecological field and evolutionary biology, which is called ecological strategy of genotypes. This is just to giving us a general framework and get some guidelines in classifying our genotypes in competitive one or stress tolerant one. And because there's not so much work done on this stress tolerance, breeding for stress tolerance, this is already something that can guide us to choose some very different genotypes in order to study a bit more deeper what is going on. So we applied it on the diversity panel. We were happy to see that there was quite a large diversity for species in terms of ecological strategy. And we also observed a huge phenotypic plasticity, because the same genotype on both sides present quite different traits. And all these traits are related to traits like specific leaf area, chlorophyll content, leaf thickness, leaf area, leaf dry matter content. And this all deal with the strategy of the plants, where to allocate resources and which strategy to use. Rapid small leaves with short duration, short lifespan, or bigger leaf with a longer lifespan, for instance. So we have different behaviors there, and we hope to find different behavior also in terms of stress tolerance. And this is also a good framework to think of ideotyping. We looked at leaf stomata, and we characterized leaf stomata of the diversity panel with stomata index, stomata density, and stomata size. We made some microscopic picture of the leaves, and we analyze it afterward with image analysis. Within the RTB food project, we focus on different quality traits. The first one is, of course, the color. And we build up a method to measure color of heterogeneous sample, like tuber, but it can be also pounded yam or whatever. We apply it to raw yam for now, and it allows us to extract the color value. I think the most important thing with this method is the custom color chart. So it means we build a color chart with color specific to what we want to observe. And this color chart allows us to precisely measure repeatability and accuracy of our method. How far we are from theoretical color, and are two different photos giving the same color from the same chart. So this allows us to be really precise in what we are doing and to correct if there is a too high difference. From this color value, we can deduce some color index like whiteness index, yellowness index, and brownness index. These were easy to do because it was already well documented in the literature. But for purple yam, it was more tricky. You can see here all the colors of the diversity panel we have. There's a few purple yam, purple genotype. And for those ones, the only index we found was a color index for red grape, but which didn't work so much to characterize purple one. We can see here that there's a mix for the same index value, a mix between purple and yellow genotype. So we had to find something else. We finally found that U value is really discriminating well our genotypes. So this is this could be interesting because purpleness is linked to anthocyan, which is implied in many different processes, and which also could have an impact on the health. So we also made for the second year the characterization of pounded yam moldability. We gather also information about cooking time and peeling yield. And we linked this observation with NIRS spectra that we took from fresh samples, raw yam, and from flour samples. So regarding NIRS, we already presented a previous publication with CNRA where we built a model for starch, soluble sugar, and protein content. Thanks to 2 other years of observation, we were able to validate this model, external validation. So this, of course, reduces a bit the accuracy of the model, but it gives really high robustness to the model. So now this is really predictive model. We can use it routinely to predict without doing any more measure of this composition, only some checks time to time. We made the proof of concept for NIRS model also for other traits, important one, amylose content, which is linked with many textural traits, moldability, springiness, and cohesiveness. So all these traits still need an external validation in order to be used routinely. All this NIRS work was possible thanks to a generic pipeline we developed using deep learning. We've done it outside AfricaYam and RTB Foods. We made the proof of concept on very different products, Arabidopsis, rice, beer, cocaine. So we tried it on many data sets, and it systematically was more efficient than traditional methods like PLS. So we are very happy of it, and we are trying to build an API, so a web interface, in order to allow people to use it. And at the end, I hope to be able, we discuss it with Lucas, to link it with BreedBase, so it will offer one more tool for chemiometrician or breeder to build some NIRS model, predictive model. We try to transfer these methods to SNRI through SOP. We built some SOP within RTB Foods, and we want to do the same for agronomic method within AfricaYam. We also host Dr. DB during 2 months for an intensive training on phenotyping methods. It was very fruitful because he appropriated a lot of these methods, and back to CNRA, he applied it. So he was able to build a photo studio from the scratch. So starting by creating a custom color chart for sweet potato, yam, and cassava collection. And after that, he applied it to the yam collection of CNRA, being able to characterize color and oxidation. So all the picture are acquired, and now we are in the process of analyzing it and trying to transfer also this knowledge about image analysis. We made a backup mission in Côte d'Ivoire a few months ago also to discuss other protocols about phenotyping of agronomic traits. So this is all going on, and it's very positive. As I said, we have one PhD student working on identifying environmental and genetic driver of yam quality, sorry. So he finished to gather all the data and is now in Montpellier to analyze it. And I hope he will be able to be here next year to present his research and his results. Recently, we are in the process of a few publications. Two are accepted about Q2L quality-related traits. And we also contribute to the special issue of RTB food GSFA on yam quality, on RTB food quality. So where we wish to publish 4 papers. We contribute to the ontology traits and to the yam base. We added some new traits which were validated and pushed already. Some are still ongoing. It means they still need validation or they need to be pushed to the yam base to be used. And we want to add all the new traits we extract from the emergence dynamic and from the ground cover dynamic as well. We upload data of 3 years trials in Guadeloupe to the yam base, adding tuber composition traits as well. And we wanted to link it with spectra we collected. But by the time I tried, Lucas explained me that they were changing their management of sampling NIRS spectra. So there was some bugs, so I have to try again. Now it is solved. They came from a plant sample to tissue sample, and this made a bit of a mistake or bug to me. But it will be added. I let Komivi continue for the genetic part.
Thank you. First of all, let me introduce myself because I think it is the first time some of you have seen me. My name is Komivi Dosa, and I'm from Benin. I joined CIRAD last year as yam breeder, and I'm located in Guadeloupe, and I'm in charge of genetic and genomic research on IITA. So I'm very glad to be part of AfricaYam community, especially because I'm from Benin and yam. I know how yam is important for our local community. And yes, I think we will have a very good journey together. So as Denis explained, we developed a lot of protocol methods for phenotyping, and they gathered a lot of data before I came to the team. So from last year, I mainly focused on leaf traits because we know how these are important for productivity and biomass accumulation. So today I will present briefly a few results on 9 leaf traits I have analyzed for our GWAS panel. We have a very small GWAS panel composed of 55 accessions. I know it is not enough, but this is what we have before I joined the team. And what we can see is that we have a very high variability for these leaf traits. In terms of traits, we have leaf dry matter content, the leaf area, the transpiration rate, photosynthesis rate, the stomatal traits. These are very, very important. The number of nodes, which is also important for the plant architecture, and the leaf thickness. So we have highlighted the correlation between these traits. You can see in the plot at the right side. Some are very positive, some negative, some are high, some are very low. So these are very important for us in breeding perspective to see how we can target and combine these traits in our breeding pipeline. What is also important here to highlight is the heritability of this trait, because we, as Denis said, we do the phenotyping in different environments in Guadeloupe. So we can estimate very accurately the heritability. We can see that for leaf traits in IITA, we have from moderate to very high heritability, and this is also a very good point for inbreeding perspective. Besides that, we decided to use whole genome sequencing approach for the genotyping, not low-cost sequencing like GPS or DATSEQ. because we know that yam, especially Gialata, is mostly dioecious species, and the genome is highly heterozygote. So we need to have very high SNP density to be more closer to the positive variant in the genome. So in total, we have in our group 1.9 million SNP, and we use for association analysis. You can see that the very high density and cover very well the whole genome. We analyze the structure of the population, and we can see that we have very low structure. This is also very good, important point when we are doing association analysis. So we have all ingredients to do GWAS. We have a very high irritability of this trait. We have a low structure, and we also apply very high density, SNP density in our pipeline. So these are some ongoing work, so I will not present all of them. I just pick few to display today, and we are finalizing this analysis and try to publish some paper and share with the community. So in total for this trait, we have identified 18 QTL, very strong QTL actually, and we perform the analysis at 2 locations. So we try to identify what are the stable QTN that we can focus, rely on. So for 2 traits, we have identified very strong stable QTN for this trait. And also we perform some haplotype analysis to find which one are the favorable allele at each peak SNP. Around the peak SNP, we have identified 33 candidate genes, and we are now investigating with some students their function, how they modulate the leaf trait in Gialata. Here I display an example of the stomata density trait, which is very important for plant in terms of intake of CO2, in terms of control of water evaporation, and how plant can, how we can modulate like stomata density in Gialata leaf to improve biomass accumulation, even sugar synthesis. So we see that we have a good variation of stomata density in the leaf and at the 2 locations. And as I just showed, we have very high irritability for this trait. The GWAS analysis highlights one strong SNP on the chromosome 18, and this SNP has been identified at 2 locations. So this is a very strong and stable QTN. And we try to find the favorable allele at this SNP, and the contribution of a SNP of a phenotypic variation is nearly 36%. So this is a very major QTN for stomata density. And we can see that the GA allele at the SNP increased significantly the number of stomata per leaf unit area. So we went down to look at the gene nearby this peak SNP. Since we are using very high density SNP file, we found one gene. We found that the peak SNP is located in the promoter region of one gene. The name is there. And we check the function annotation of this gene. And it is the gene we call KCS. KCS is like an enzyme that is involved in the biosynthesis of very long fatty acid, and is involved in the accumulation of, in English, how you call it, a wasp, cuticular wasp in the leaf. So we try to find its homologue in Arabidopsis thaliana. Arabidopsis thaliana is a model plant in plant genomics. And we found that this gene is the homologue of HiC. And HiC has been reported since 2000 by Gray and collaborator. This gene, when they knock out it in Arabidopsis thaliana, they were able to increase the stomata density by 42%. So when we got this result, we were very happy because we know that we are very, we identify something very special in yam. Besides that, this also has been reported in different crops like mung bean, cowpea, for modulation of cuticular wax. So besides GWAS, we also have 2 biparental population. database built by Chema Ano, our colleague, who has done several QTL analysis on tuber appearance and some quality traits. And in one of the population, we have a very good segregation for leaf traits. So it is yes, it is an opportunity to confirm what we have obtained from the GWAS panel. So we also did the phenotyping activity for leaf trait in terms of sclerophyll content, leaf size, stomata trait again. It is very laborious work, but we have several master students who assist us. And here you can see in the picture, 17.2F is a female parent, has a large broad leaf and long peciole, have a lower chlorophyll content compared to the male species, male variety, which is Kabuza. And in the population, we have very good segregation. So in total, we have identified 16 QTL using a linkage mapping approach in the bee planter population. We can see that some QTL, like QTL for stomata density, is exactly what we have obtained from the GWAS analysis in the same region, just that the contribution here is lower, and the size of the QTL is also bigger, much bigger. This shows that yes, this GWAS approach with high SNP density is much more powerful than QTL for some traits, especially for very quantitative traits. And we also identified some new QTL for leaf size we could not find in the GWAS panel, probably because the variation is not enough in the panel. But from the bee parental population, we could identify some very good and strong QTL, major QTL for leaf area, especially for peciole length. We were able to find very strong QTL for peciole length, and also located very good gene underlying these traits. As I said, when I joined the team, they have assembled about 50 accession as GWAS panel. For me, I think it is low. It is very low to have enough power and detect a good QTL. So we spend a lot of time to try to increase, to extend the diversity panel. So I contacted IITA here, Dr. Gay, who presented today, really helped me a lot to provide nearly 100 accession from West Africa country. So today, with 30 variety from CRBPT, Vanuatu and Guadeloupe area, and 20 accession from China, we are near to 200 accession in our diversity panel. And this is very good, especially because we have nearly half of the diversity panel is from West Africa. And I'm confident that what we are doing in that environment in Guadeloupe, we can easily transfer this QTL back here for breeding, for breeder. Now we can reanalyze some traits that are not working in the initial panel because we did not have enough diversity. But now we can reanalyze this threat. And also we are moving forward for genomic selection in our team. So I think this panel will be the core for, and to build around this accession to build a very good diversity genomic selection prediction panel. What also we are doing now is we are trying to select some varieties from these new materials to validate what we have obtained from GWAS or from QTL mapping analysis. Here I display an example of the marker for sex discrimination in Dia Lata. Okay, in Dia Lata, we have confirmed that this marker is working very well. All our plants we have a phenotype as female or male are segregating very well in the Caspar machine. I have to say that we have our own Caspar machine in our lab, so we can do this kind of work easily. Just we have to design and order the markers. Yes, quick briefly, the work plan, what we are planning to do. We are still Denis will continue with validation of SOP for phenotyping method of different traits related to the competitivity against weeds, and also develop new calibration for NILS in terms of tuber quality and also for agronomic traits. He will also contribute for capacity building here at CNRA for data analysis, development of method and protocol. And I know that now he is very inclined to work on drought tolerance using drone and imaging phenotyping protocol. From my side, I will continue to do the GWAS analysis on the different traits, especially for ground cover dynamics and the phenology, and also to try to validate some markers that we can use daily in our breeding program. One point also for me is very important. I realized that in yam, we are still struggling with flower induction. I read a lot, but I think this should be one of the major issues we have to tackle, because if we cannot have flower, we cannot induce flower, we don't have enough gene pool to do crossing and to develop a population. So this is something we have already started with CNRA, and we have one PhD student who is working on this specific topic, and we will combine genomic approach, biochemical approach, and to try to highlight what is going on, what block flowering, and how we can induce flowering in D. alata. Okay, the last point is we also have a lot of data. Actually, we have a lot of data. We have a lot of, for instance, genome sequencing for D. alata male species in our team. We have pan genome data. I think this data should be released, and we are working to release a platform, online platform called YAM Hub, and which will be very useful for scientific community with different tools. This part will be handled by Hannah Shair and myself, and there will be also some tools for molecular marker mining in D. alata. Okay, so thank you very much. Hope I have not to be long. I don't know if Denis want to add something. Okay, thank you.
Okay. Thank you very much for your presentation. We will now take questions. 2 questions to CIRA team.
Yeah, thank you. Very, very nice, very elegant work. On this last point, the YAM Genome Hub, is that restricted to Dioscorea alata, or will that expand to include Rotundata as well? Or will it be a universal YAM hub?
For YAM Hub, 80% of data are on D. alata in terms of transcriptome data, molecular marker, but we also have a lot of data on different Dioscorea species. So we will also load them online. But I think those who are working on D. alata will be more interested by YAM Hub. Of course, we'll open it if there are scientists who want to join and to add more data on different species. They are free to do it. It will be public platform.
Other question? Oh, okay.
Thank you for the nice presentation. I want to find out about when you use senescence, from senescence to sprouting as the dormancy period. And we know that senescence can be influenced by climatic factors. What do you think about the idea of taking calculating senescence from tuber initiation to the break of dormancy, because that idea had come out some time ago. Yeah.
Thank you. You're completely right. Environment is playing a role, and storage environment also. If you have millibugs, it will delay germination. If you have higher temperature, it will hasten germination. So the idea behind is to have as many years as possible for preservation to have more robust data. And on the idea of taking the tuber initiation or emergence, I mean, something visible earlier, it could be possible. Tuber initiation is quite difficult because it's different for every genotype. So you need to dig and to observe it. So it's a huge work to dig and to observe it, tuber initiation, which also can be influenced by environment. And moreover, there is a risk of confusion effect between growth cycle length and dormancy itself, because growth cycle lengths can vary from one genotype to another one, independently of the dormancy. And if you take the tuber initiation as a starting point, I think there will be some confusion effect. And if you look at QTL after, you will find QTL linked to phenology and some QTL linked to dormancy. Anyway, if we have the data, we can try. But I agree that only 1 year is not enough due to the environmental influence on this trait.
Quick question. Have you guys or somebody determined any effect of high or low stomatal density on production traits or other things?
We're reaching the point to have enough data. So this is a job of the PhD I talked about. He will try to link all these data, agronomic traits, to the yield and also to the quality, and try to differentiate direct and indirect relationship between all this. Of course, for now, it is a theoretical observation. It is still not linked to the yield or to the drought tolerance or whatever. The only point is that we tried it with comparing diploid, triploid, and tetraploid genotype, which have very, very different stomatal density and stomatal size, trying to reach in the area exchange, gas exchange area. And there was an effect. Maybe it was not the only driver, but there was an effect, and triploids and tetraploids behave better, and even better as the environment was bad. So there may be a link.
Okay, congratulations to CIRAD team. We will now move to, the next presenter will be IITA.
Thank you very much. I'll briefly go through the YAM-based update. Thank you very much. So I'll briefly go through the YAM-based update. But first, I would like to present my acknowledgements, and really I'd like to thank the entire YAM community to work with us and to work in a very productive way, and indeed the entire RTP community. And of course, I also like to thank all my colleagues at BTI who do a lot of the programming work and creative work on the basis. So thank you, everybody. So YAM-base, as many of you already know, and many of you have already seen this slide many times. YAM-base is a digital ecosystem for yam breeding, meaning that all the data is collected in a digital way and processed as such. And it goes through, creation, evaluation of phenotyping trials, analysis, making selections, and so forth, and then managing crosses and managing pedigrees. And it interfaces very heavily with these tablet apps, such as Fieldbook or Coordinate app, to collect data in the field. So some of the features are, it's completely web-based, and most of you know this by now. I just want to mention it. It's a multi-species, multi-breeding program, multi-user database. It manages all the important breeding data, such as accessions, fields, locations, traits, and of course, phenotypic measurements. But it also manages genotypic information, and it can correlate the phenotypic and the genotypic information. It also supports very special phenotypes such that we call high dimensionality phenotypes. And one example of those is NIRS data that we have just heard from Denny. And also it can manage product profiles and stage gates and so forth. And it has a fairly good collection of analysis tools. It can do mixed models, clustering, stability analysis, heritability, and of course the SolGS app. So I'll go through some of those in more detail in the presentation. And this is just to mention that we published a paper about BreedBase recently. And this is the paper, and there are, some of the co-authors are in this room. So if you're interested, please go and read that paper. This slide has been shown by other speakers already. This is a statistics page of YAM-base, and you can see all the data that's in YAM-base, and you can see that we have about 800 trials in the database, and there are almost 300,000 stocks, of which about 75,000 are accessions. So that's a fairly good data set. And we have 1,300,000 phenotypic measurements. And I think last year we had about 1 million. So that's 300,000 more, or 1/3 more, which is quite amazing progress, actually. So we have worked a lot on the analysis tools in recent years, and we have created the analyze menu with all these different tools that you can use to analyze your data. And we've also created a sort of analysis framework so that all these analyses actually work pretty much in the same way. And if you know one analysis, you can easily run other analyses. And we've also started a project to kind of review the statistics capability of YAM-base and of BreedBase. And we are improving how it handles statistical data. So what is this analysis framework? So what we have tried to create is a consistent set of features that allows you to manage input data, the methods and the models and the training data and the results in the database. And all this metadata can be stored for a specific analysis. So, for example, if I run a SolGS prediction, I can then store the analysis results, the blobs and the adjusted phenotypic values in the database and use them in downstream breeding decisions. But also I can take analysis results and feed them into other analyses and create chains of analysis. One of the really central tools in BreedBase is the SolGS tool that is a poster child of how genotypic and phenotypic data can be integrated to make useful predictions for breeders. And what it can do, it can do the training and the prediction of phenotypic values. And it's being constantly developed, and the author is here in the room. So if you have any questions, please talk to Isaac. Now, recently we have expanded this prediction capability to also predict mate selection prediction, meaning that we can predict which parents give the best offspring. And this really started with the AfricaYam project, this initiative, which is really great. And it's like SolGS, it's also based on genomic predictions. And there's actually a new graduate student in the lab, Christine Yaga, who is working on this, of course, along with Isaac. And some of the code has been contributed by Marley Labru of EBI. And you know, the main drivers, of course, are Agre Paterne and Azrat Amele from the breeding programs. So while we implemented this tool, we realized that, for example, our mixed model tool should be improved, and it uses a backend, or used a backend that was not as powerful. So we moved it to a more powerful backend. So it used to be LME4, and now we have an implementation that also supports the summer package, which has somewhat more features that will be very useful down the road. So I want to show you a little bit how this GCPC tool works, the mate prediction tool. And there's this workflow that you have to follow. And we have been wondering, you know, we have this analysis framework, and how will this tool fit into that framework, right? And so we have tried to reuse all these analysis tool framework features to implement the GCPC tool. So the first step is to create a data set that contains all your phenotypic and genotypic data using the wizard. And for everybody who attended the workshop on Thursday or Wednesday, Thursday, you have probably seen this, how this works, how you create a data set using the wizard. Then you have to use the selection index tool to create a selection index. And then you open the GCPC tool, you choose the data set you have created, and you choose the selection index that you have created. And also you choose, and something is missing here, you choose some model parameters for the mixed model, and then you hit submit. So I'll just walk you through that real quick. So this is what the search wizard looks like. As you may remember, the search wizard looks at your breeding data as a big n-dimensional cube, and you can, along the sides of the cube, you can specify different entities, and you create an intersect in the database, which is then your data set. And you can store that in the database with a name. And here I just made a random example. I choose a breeding program. I took IITA. I choose genotyping protocols. I use DART sequencing. Oops. I chose accessions. And these are all the accessions that have been sequenced by DART and are associated with IITA. And then I selected traits. And so all these accessions that have this genotyping also have these traits associated with them. And so that I stored in the database. Then the next tool that you use is the selection index tool. And you go to that tool and you can create a selection index, how I've done here. Here I chose just some random traits as an example. Of course, these traits have to correspond to the traits that are in the data set. And then you can give coefficient to weight that trait in the index. And you can store it again in the database and give it a name. And then you can go to the genomic prediction of cross performance tool, or that we call GCPC tool for some reason. I'm not really sure how GCPC, what it stands for, but we just talk always about GCPC. And so it lists what it does, this tool, it lists all the data sets that are in the database, and you can select one of them. So maybe you select the one that I created. You just click on this corresponding checkbox. And then you click on proceed to factor selection, and you can enter, the tool will analyze the data set and see what factors are in there, and it lists all the factors, and you can then choose if they're fixed or random factors for the model. And it always sets the germplasm name as being a random factor. And then you can choose the selection index that you want to use for the model to rank your germplasm. And then you click Run GCPC. And after a while, you will get this table, and it contains all the pairs ranked by the cross predicted merit. And you can also see that the parent sex is also displayed, and it actually filters out combinations that are not crossable. So you get only male-female pairs in the table. So another thing I want to briefly talk about is multi-dimensional phenotypes. So these are data types that don't consist of one value per measurement. Like plant height, you go to the plot and you get, 1.57 meters. So these are things like expression data, proteomics data, metabolomics data, and ground penetrating radar data, as well as actually NIRS data. And we work a lot with Jean-Luc Yanning's group to implement that. And we also work with Peter Selby of the BRAPI project to modify the standards of BRAPI so that this data type is supported. So this brings me to NIRS, and I guess we have heard a little bit before about the NIRS analysis that's being done at RTP Foods. And so these NIRS analysis can now be uploaded to Yambase. And also the calibration can be performed by Yambase. And this was initially implemented by Jenna Hirschberger and Nick Morales and Chris Simoes. And a similar data type is ground penetrating radar data. And again, this is a non-destructive method to visualize roots. And it's currently ongoing at Ibadan, where Alfredo et al. Are taking measurements. And here we also store raw data currently, but in the future, hopefully also, the database will be able to analyze the data and store the actual traits derived from this raw data, which is quite voluminous. Then I guess we have heard already from Denis about the RTP Foods project, and we have done a lot of work with adding ontologies and traits and terms. We created a lot of post-composed ontologies, meaning ontologies that describe how maybe food samples are prepared and so forth. And so another thing that you can now work with in BreedBase is product profiles and stage gates. So you can manage product profiles in Yambase, and they are defined by traits and trait values, or also reference accessions. Then we have a new genotype search, and it allows you to search genotypes by allele states. So for example, you could create so-called marker sets that contain certain allele states for markers that you know contain all the resistances that you're interested in, and you can run it against the database, and it retrieves you all the accessions with exactly those allele states. Another interesting project that we have is creating an ordering system, and this has been asked for by the Banana Project. And what it does is different breeders want to exchange germplasm, but there's no real good formal way of doing that. And so, of course, building this into a database such as BreedBase makes a lot of sense, because all the accessions are already in the database, and all the users are in the database. So we can attach a new role to a specific user that's called a vendor role. And then this user can elevate accessions into the catalog. And the catalog is a special search that shows you only cataloged accessions. And you have a little shopping cart icon. You can put them in the shopping cart, and that will send a message to the vendor, and the vendor can then update the state of the order and finally ship the accessions to the customer. What's very important to us is BRAPI. BRAPI is an important technology underpinning many of the breeding functions in BreedBase. It's a breeding application programming interface, meaning it's a systematic way of exchanging data between databases or other devices, such as Fieldbook. And it's an evolving standard. We need to keep it up to date. And there will be a hackathon. Every year there are a couple of hackathons that we participate in. The next one is in October. And Mirella Flores is the person who implements the BRAPI at the BreedBase. So just to finish, some people may have wondered what happened to the YAM-based FTP side. But unfortunately, the FTP protocol has been discontinued and is not legal anymore. And that's very unfortunate, because it was heavily used by the biology community and the genomics community. And now it's become impossible to use it. So we still make all the directories available through HTTPS, but instead of typing ftp-yambase.org, you type https://yambase.org/ftp, and you will see all the files there. But the FTP protocol cannot be used anymore, unfortunately. And that's really something that's above my pay grade. I mean, that's something, that just happened because FTP is an unsecure protocol. So another thing we have been doing for now a couple of years is we have created so-called Docker implementations of the code. And so you can easily install the site using Docker. You can download it very easily and install it. We have generated, I think currently we have generated about 50 releases in about 2 years. So it's a release every other week. And these Docker images are being downloaded. Anybody can download them. There's no limitation. So there are a lot of people who, for example, university labs that are breeding or small breeding companies download them, and then I get emails how to make it work. And so it's been downloaded about 4,000 times, which is quite a large number. So thank you. Any questions?
Okay, thank you for your presentation. Question now. 2 questions, yeah.
Thank you so much, Lucas. 2 things. One of them, we discussed it before, but it was on the sidelines. Of all the things, one important one that we may be urgent is to bring the tricot data into this system. think it'd be more useful, it'd be very, very useful for breeders actually. And also the large plot trials. I don't know what they call them in YAM. Is it demand creation trials? Are they the same as trichot or they'll be different? But I know in maize we have large plot like strip trials where a farmer is doing a half an acre, something like that kind of data. Then the other one you've talked about bringing the search of product profiles. That one will also be a game changer. My experience in the private sector, I remember we had our marketing giving us a new profile of a competitor product that was challenging them. That's in maize hybrids. And then I asked my breeders to say, go back into all the trials and look for something that matches or is closer to this. We picked about 80 hybrids that were dropped in 2005. We were in 2016, 2017. And then when those 80 were picked, seed was produced the following season, they were tested against the competitor product. We had 16 above the competitor product. And one of them is now a market-leading hybrid coming out of that. So you see that with all this work we are doing about product profiles, some crosses might be there with the breeders, but because they were not looking for that particular profile, they were dropped then. We can move faster if that module is incorporated. Thank you so much.
Yeah, thank you for the question. Yeah, the trichot data that's been on the, on the agenda for a long time to integrate those data. We just need to find a good way to anonymize all the data, and then we can fairly easily store that, I think. But so far, maybe because of the pandemic or other reasons, that hasn't happened yet. But yeah, this is something I think that's extremely important to integrate also. And yeah, the product profiles I think it's super important because it really gives you, your breeding targets, and so really everybody know if it's in the database, then everybody knows what those are, right? Because if it's not there, then it's maybe kind of more mysterious what the actual targets are. Other questions?
Yeah,
Oh.
okay.
Thank you, Lucas. Would it be possible to link the breed base or the YAM base to the gene banks?
I mean, yeah, that's possible. We heard this first talk this morning, and I thought, how is that data linked to breed base, to YAM base? And I don't know, but it should definitely be linked. Yes. So yeah, definitely a good idea. Yeah. Also, I thought, hearing about the YAM hub, I thought that might be something good to link to YAM base so that people can get all this up-to-date genome information from there easily, just with a click.
Okay. Thank you, Lucas, for your presentation. We will now move to Beatrice from IITA to present on seed production. And so we have that.
Okay. I'll be scrolling down here. Okay. I should have given you. I wanted to see. That's too late to copy. That's what I did. Yeah. Good morning, everybody. Good morning. Good morning. Okay. Nice to know you're live. Yeah. Okay, I'll be going a bit of what we've been talking about since yesterday. We have been looking at how to improve our yam varieties. And now we'll look at how we are going to take those varieties to our farmers. I sent it in PDF, so I have to be scrolling them. So it's stuck. Okay. I won't talk too much on the importance of yam because we know much about it, but the pie there really shows us the value of production of yam compared to other staples in Nigeria. You can see the large segments of yam. In fact, if we put the other 5 staples together, it's almost same as that of yam. This figure has actually changed. Now it's about 47%, not 44%. And the background of the research on yam system came from the study of these traditional systems that showed that it was prone to limited supply, poor quality. You will use about 1/3 of your production as seed. And we have almost 40%. Sorry, I think this presentation has actually been updated. It's the old version that we have here. But we have between 40–60% of the production cost being that of seed, with an extremely low multiplication ratio of 1:3. And then we have an absence of dedicated seed producers because every farmer will produce their own seed from their ware crop. They recycle seeds, and we don't have improved varieties in the system. This picture shows us this slide shows us how we get seed traditionally. We have small hole tubers, or we get the big tuber and it is cut into large chunks of up to 500 grams, or we have the double harvest that is further cut into sets of planting, or we will get the materials from the local market. Sometimes also the large whole tubers are cut at the first harvest and will be buried. Then during the final harvest, then we take them out and use. Or we could also use those large, the well-sized tubers, up to 1 kg for ceremonial yam production. The yam improvement for income and food security in West Africa, the IITA project, actually came into being started in 2011 for the first phase and ended in 2016, with objectives to increase the productivity for smallholder farmers and ware yam producers in Nigeria and Ghana, and also to deliver global goods research in seed production. In the second phase, the project tried to transfer the advances that were made in seed systems to the private sector and the NARIS. But in summary, IITA's objective was to improve seed quality, availability, accessibility, and affordability in Nigeria and Ghana. So some of the innovations of IITA include cleaning of the seed stocks and multiplication. We already heard from Dr. Badara how they are going forward in cleaning materials that they have in the gene bank. So these materials were cleaned and then multiplied and distributed to national partners. Then we worked on developing high ratio propagation techniques. The bioreactor system was a major development in that respect. Then we had also the aeroponics system and then the hydroponics that use several substrates. We have cocoa pits, rice husk, and sand, river sand, and use that with liquid nutrients. When we started with the aeroponics, we actually were trying to look at the potato system that was being practiced in East Africa. They used the aeroponic system and they were able to multiply the potato crop very fast. But in the case of yam, we still ended up with our 1 or 2 tubers. And we saw that the foliage was so enormous in the system. Then we started focusing on using the vines for multiplication. In the past, the vines didn't give a good response when we tried to multiply, but after cleaning and the luxurious foliage that was being formed, we tried it again and we saw that it performed very well. So the multiplication now focused on the yam. That way we had more tubers saved as food, and then there was a reduction in the spread of nematodes and other soil-borne diseases. We've had multiplication rates that have increased from 1:3 to almost 1:300. Then the production season has become more flexible, and that has added further value to the yam crop. We use the leaf bud cuttings. So the picture there, that is what we actually focus on planting. We plant those in seed trays in the screen house, and we get those mini tubers. The field there to the right is what you see in the field when we use those mini tubers of between 1 to 10 grams. And we plant at very high density, 100,000 plants per hectare. And this gives us tubers that range from 50 grams to 500 grams after 5 months in the fields. Now, to get this single bud, leaf bud cuttings, we grow them we plant those mini tubers in the screen house, and then get them to produce large foliage. Then we start cutting from them from about 3 months after planting. We have also tried to transplant. That's we get those leaf bud cuttings rooted in the screen house, and then we transplant it to the field. We get tubers as heavy as 7 kg. But because we are looking at seed yam production, we limit the time that the plant stays in the field to about 5 months, and we get more of seed size. But if you allow the plant in the field much longer, you can even get well-sized tubers. Now, during the last 2 years of the YISWA project, we also came up with the leaf bud cuttings that are planted directly in the fields. We just get those cuttings, treat them with fungicides, and we plant them on beds. These are provided with shade, and then we mulch the beds with rice husk. And we have very good field establishments, up to 98% establishment. And you can see the very lush foliage that forms at about 12 weeks after planting. And we get also very good yam seed tubers from it. That's after 20 weeks after planting. Now, this direct planting of the leaf bud cuttings has given us the highest rate of multiplication. And we have trained seed companies in Nigeria and Ghana on how to go about this. There are 6 companies in Nigeria and 3 in Ghana. They have various options that they can use for seed production, but this particular one that we discovered late, they've also been trained on how to use that. And I'm glad to say it is one of the methods that they have actually taken up for production. That we have other outcomes that came from the YIFSWA project. We have been promoting 3 yam varieties. Those are the varieties that we use to test the systems that we have in place. We've told the seed companies that there are many more varieties. If they need more, they can get them from IITA or the national partners. Then we've developed the diagnostic tools for virus detection and elimination. We have the quality management protocols for standard certification, which is being used by the National Agricultural Seed Council in Nigeria and the PPRSD in Ghana. The private sector have been trained on the business plans. We made their business plans for breeder and foundation seed production. And then we also have had a lot of product promotion events with demonstrations, advocacy, awareness, and all of that. And these NARIs have been, their capacity has been developed on quality seed yam production. The quality regulators, the private sector, they were provided with tips, the aeroponic system, hydroponics, solar power, and the ICT tool that they need. That is the seed tracker. In Nigeria and Ghana, they've been trained on how to use that so that they can locate the farmers, the producers, what varieties are available where, and it's also used for the quality control. Then we also started a multi-stakeholder forum for policy and advocacy. I think that has graduated to the National Association for Yam Producers, Processors, and Exporters in Nigeria. So now we are looking at redefining seed yam, and we asked our seed companies, with all that they know on the seed yam production, what do they think will be the best seed yam for them to use, at least for foundation seed production? We can see the seed yam that we were recommending before YIFSWA, a whole seed tuber of about 250 grams to 300 grams. But now we are looking at this leaf bud cutting, and most of all of the seed farmers actually said they would prefer small whole tubers of between 1 and 10 grams. That is what is needed for foundation seed production. Then for the ware yam production, we have carried out experiments which show that if we have improved varieties, good quality, between 30 and 50 grams is what you need for seed production. So you can, to plant 1 hectare, you just need a few bags of seed, not the truckload of seed as we used to have. So these are the seed companies that we have. There's Dow Green in Kaduna, in Nigeria, Biocrops Biotechnology in Abuja, the Nwabudu Agro Seeds Inputs in Abia State, Strategic Seeds in Abakaliki, the PS Nutraceuticals, those ones, they are not doing too well after all the training that they have received. Then we have Go Seed in IITA Ibadan, Umudike Seed, they came on board during the last year of YIFSWA, so we didn't have much time with them. We are hoping that more work, we still need to train them more before they can go into large-scale production. In Ghana, we have Fusua Food Chain and Iribov in the Volta region and Hikma Seed in Tamale. But then what is the status of the delivery of quality seed? We can see that the breeder has produced so much seed and the farmer is still doubting, how do I get my field to be as productive as I would love to? So there is still a gap. In fact, there is just a thin line that is linking the improved materials to the farmers. So the projects try to establish a formal seed system to enhance the availability of and use of the released materials. We've started with the breeder seed production, the foundation seeds. They have been trained on how to go about that, but the certified seed stage is still not well developed, because in YISWA 1, a lot of focus on the research. During this second phase of YISWA, the first 2 years were to be used as a proof of concept, whereby we had to actually prove that with so much seed, we can plant so much hectare. So that one was done. And then the last 3 years is all we had to train the seed companies and then train them on all the technology. So that time was not enough for us to go down to certified seed production. We also still have this informal seed system there, and we are hoping that it will be developing too, it will be improved as we are trying to develop the formal seed system. What is happening there is that some of them already have some of the improved varieties, at least the varieties that were promoted by YISWA, and we are hoping that we are going to use more of them as the certified seed producers, as seed entrepreneurs. What is the way forward? We are going to continue to promote the new technologies, and then to develop agronomic packages for high ratio propagation technologies. In the past, we have been looking at the agronomy when you are using mini sets or small hole tubers. But now we have come in with the leaf bud cuttings. What is the agronomy that you need for these new technologies? Then we also have to look at how to reduce reinfection. Once you take materials to the field, then they get reinfected with the viruses and nematodes. How do we reduce that reinfection of the clean materials that we take out? And then we also have to develop technologies for handling, packaging, transportation, and storage of these mini tubers. That is what we are going to be promoting. We need catalogs of the new varieties. The seed companies we've trained, they know only of these 3 varieties, that is the Asiedu, Pamio, and Swaswa. Which other varieties are there that they will need? So we need to get catalogs so that they can select from those ones for production. Then we also need to establish a pool of seed entrepreneurs to multiply the seeds further for farmers to have access. So then the awareness creation for the potentials of this quality seed, and we need to have tools that will give us good estimates of the demand for seed. Institutionally, we need to promote the central seed coordination. That is in terms of quality controls and all of that. And then to promote policies that will advance the formal seed system and at the same time improve the informal system. So in conclusion, we already have a lot of these rapid multiplication techniques for the seed producers to select from. The former seed system has been established in Nigeria and Ghana, and we are scaling these technologies. We've also sensitized stakeholders to create awareness of the new opportunities that exist in the yam value chain. And then we are looking at how policies can be, how we can have a policy that will actually promote the distribution. Like the suggestion of, I mentioned yesterday, how do we tie seed production and release so that they don't work in silos? The breeders are doing their work, the seed producers, they are there, and there is nothing that is really linking them. Now, if you look at those pictures, you will see the leaves attached to those mini tubers. We also need physiologists to help us to find out what actually happens, because when we make those single node cuttings and we plant, there are some that will not produce any shoots, but they will give us those mini tubers. And if I'm a seed producer, that is what I want to have. You just have your single leaf and it is manufacturing everything to store without having to produce the vines. The big tuber there was actually obtained from a single node cutting after 8 months in the field. So we see that there is really a wide possibility in the use of these vine cuttings. I would like to acknowledge the foundation, Bill and Melinda Gates Foundation, and other YIFSWA partners that made these achievements possible. And thank you for your attention.
Okay, thank you also, Beatrice, for your presentation. We are now going to take 1 or 2 questions, and then we'll go for a break. Yeah.
Thank you, Beatrice. I asked this question before, and you may give me a different answer, but why is that you guys do not clean and have good seed of local preferred varieties like Laboke in Ghana or other varieties? You just focus on the improved varieties, but it seems that farmers may benefit a lot by having a clean Laboke with good quality of seed, for example.
Yeah, we have those varieties also cleaned. Yeah, in Ghana, they have, I think the last release, or not release, registered varieties, 5 or so, they were all land races. So they have been cleaned, and the seed companies are waiting for the materials to be multiplied so that they can start with them. In Nigeria, we also have that. Nigeria was the first to do that because we realized that those are the varieties that farmers know, and we're going to use that as a vehicle for the improved varieties. Because when we went to them with the improved varieties, it's something that they had no idea about. But when you go with Puna and somebody comes to buy Puna and we say, oh, we also have these other varieties, then they can take them to try them. So we have those varieties at NRCRI and CRI. Yeah.
Okay, another question. Yeah, the second one.
Thank you. I wonder if there is some difference in terms of performance between Dialata and Rotundata, for instance, when you apply this technology. Second question is, can we make a correlation between leaf size, maturity, and the performance, the size of the tuber you get at the end?
Yeah. So the first question was on, okay, Dialata and Rotundata. We have focused more on Rotundata because that is where we really have problems with multiplication. But Dialata also performs excellently. Yeah. Then for the leaf size and all of that, we still have to do a lot of work. Like I said, it is within the last 2 years that we came up with these methods of rapid multiplication. But what we have observed is that apparently there is a stage that when you get the cuttings, then you will get more tuberization. More of the cuttings that you plant will start tuberizing. If you get the plants in the juvenile stage, the mother plant is in the juvenile stage, then you have the shoots will come up, will produce more shoots, and you have large plants. But if you get it at the active phase of tuberization, the leaf that you plant may stay just one leaf and start tuberizing. That's what we have observed, but we need to do more studies on that. Yeah.
Beatrice, thank you very much. Nice presentation. I also have 2 questions. One is, you pointed out the weakness of the mini tuber production in the field being exposed to viruses and nematodes. Is there a way to use appropriate tunnel technology, large-scale boxes, 2-3 meters wide by 50 or 100 meters long to have high-density plantings that are covered by screens, so you have screen tunnels? In some countries, screen tunnels are used widely in horticulture to protect them from exposure to nematodes and viruses. Secondly, it seems there are very strong analogies in tuberization between yams and potato. And in potato, when you use the leaf bud system to look at physiological maturity, there's a gradient where the older leaves will go straight to tuber. Intermediate leaves will have a stolon and then tuber. I know you don't have stolons. And the upper nodes, the juvenile nodes, will go to a green shoot that maybe later will make a small ancillary tuber. So have you looked at leaf age on the vine in terms of performance? And could you affect then which nodes you choose to use to get the ideal mini tubers?
Yeah, thank you for those interesting questions. The first question on use of tunnels, we have not tried that. But what we do is that we plant in boxes in the screen house, because we are trying to limit the time of exposure in the field. So if we can get as much of foundation seed as possible within the screen house, maybe we go to get foundation 2 in the field and the certified seed, while the others are done within the screen house and laboratory. We have not tried the tunnels. But in the yam belts of Nigeria, the temperatures get so high during that period of production that having tunnels, I don't know how it will look like. It's something that we could try. But that is just to protect for viruses. The nematodes that come from the soil, when we are cultivating in the screen house, we use cocoa peat. Cocoa peat is very expensive, so we've come up with the local use, with the use of rice husk. That one is more abundant in this part of the country. So we are using rice husk and it is performing very well. But if you have also good nutrient solution, ordinary river sand, you just wash the river sand well and we can plant in it and it does well. So that way we don't have the issue of nematodes. And when we are cutting the vines, we don't have any nematodes in the vines. So for the vines, the problem is that of virus, which we can always detect in the screen house before getting the cuttings. Now, in terms of the position of the vines with regards to tuberization, we started an experiment that which is not conclusive yet. But when we are cutting from the mother plant, we usually cut we leave 2 or 3 nodes, because the 2 or 3 nodes we produce more shoots and we will cut again and possibly a third cutting. before the plant senesces. So it's something that we have to look out. But if the plant is still juvenile, it doesn't really matter. They all will produce shoots and grow. It is towards the later phase of the plant that we have some leaves that will produce just tuber and no shoots.
Okay. The last question from Prof. Otu.
It's a contribution because I've done a lot on that. And the first thing, I agree with them, the scenario that they painted. If you take the very young leaves, they go to production of some vegetation, and for that matter, later get your, if you take the older ones, which are closer to maturity, or it's about bulking, once they get into the soil, start bulking. But our experience, we realize that if you offer them the opportunity to get some vegetation before they start bulking, you have a reasonable bulk. And also, where the material that you are going to use to plant, to cut from the mother plant, we realize that if you fertilize them, we give them liquid fertilization for some time before you cut, we realize that you were getting better success. And it emanated from the fact that sweet potato, you cut and you have no problem. They are sprouting all over the place. So we hypothesize that, okay, why is it that yam, it doesn't? Can we boost the fertility status of the mother plant before we cut? And it did exceptionally well. So congratulations, that something.
Thank you. Just to add to that, we are looking now at getting two different feeding regimes. The first one to boost foliage production, and a second one that will be for tuberization. So those are things that we are looking at. And also because we don't use any hormones to boost the rooting. We just cut and we plant, and within eight days, rooting would have started. So if you have a cotton that really has sufficient nutrient within it, then the chances of its rooting is faster. So there's a lot that we'll still have to do to improve on what we have on ground.
Okay, thank Dr. Beatrice for your presentation. I think this is time for coffee break. Okay, we are going for coffee for 30 minutes, right? It's okay? There was much. Okay, 15 minutes. 15 minutes coffee, and we will come back.
Obozi gini ka nachono. Imana chukwa gozi gomo. Onyelu mi you, nyelu you me. What am I looking for? Say my God, it don't mess me up. He giving me you, giving you me. Opposite keep and not just so. Opposite keep and not just me. My baby, you know, say you got love for me. I don't go.
Atta. Mr. Atta. Is it okay?
So good afternoon and welcome back from the coffee break. I hope we had a very nice break. So I'm starting the presentation. I am Stephen Yakubu Atta, Managing Director of a seed company in Kaduna in Nigeria called the All Green Seeds. So I would be talking to you on an area of interest. That's observations we've made with respect to a variety that was released by AfricaYam, which we try to compare with the varieties that we use in the ISWA project. So that will be my presentation. this afternoon, just to compare likes and where there are things of interest and what is it as business people, what we want to deduce from that. Having said that, I want to also inform you, yes, as seed companies, our major preoccupation is that we identify technologies on the shelf with breeders and even farming systems sometimes from partners, which we commercialize and make it available to farmers in order to improve either their outputs, incomes, or whatever. So mainly we are people who are looking out for technologies that will improve mankind. I want to also state here that during the course of USWA, we are limited to some few varieties. But by this same interaction of trying to find new solutions to problems and ways of moving ahead, we came into interaction with Dr. Asrat. And because of that, he made us to know that African yam had some other yam varieties, of which one of the ones we chose to pick, I haven't had the briefs and discussed with him, was the VAYAM. So what I'll be presenting today is we're trying to compare VAYAM with the yam varieties that we had in the USWA project. Sorry. Next page. It's not moving. Is it okay? Can I use this? Okay. Okay. So I start first and foremost talking about just the background of this discussion. Mainly what we'll be presenting here, because this variety, the VAYAM, we have only gotten it from AfricaYam for about 2 seasons. And we have been on the field with it. And what I'll be saying or talking to you about will be restricted to our experience with this variety vis-a-vis the other varieties within the space of 2 years. The 3 varieties that were popularly used during the USWA project is the Pamio, Acedo, and Swaswa. Those were the 3 varieties. And also, I want to inform you that within these 2 years, we are only able to produce foundation tubers and foundation vines from this variety. And that is what we'll be talking about, our experience within these 2 areas. We have not reached certified seed level with the VAYAM. But when we get to that level, whatever observations we are making will also be presented at that time. So before I start talking talking about this variety, I want to introduce my company to you. That we are a seed company that has been there for almost 25 years, starting from 1997. Dow Green has been there. Initially, it was called Alheri Seeds. We have been in the business of seed production for all this while, and we came into the yam seed system vis-a-vis the coming in of USWA project, where we worked in the USWA project, and we have about 5 years working with yam as a seed. So all these other varieties will be limited to our experience within this 5-year scope that we worked on that. Having said that, also we have been into other seed systems or selling other seed products, the cereals. Okay, in fact, we even have the tubers, which is the last of the entrance, vegetable seeds, and then we are also into tree crops. So having said that, I want to start first and foremost by just refreshing to those who know, or telling some of us who may not know about this variety, the VAYAM. It's a very interesting variety because as far as we are concerned from the briefs, it was a variety that has a lot of future in it, or prospects, like we said. As seed people who are looking for this kind of technology that we can promote to farmers and make money and make business, and they also to improve their production. So this variety, based on what the breeders have said to us, it is an Alata variety. is adaptable for production into the savannas, Guinea savanna, Northern Guinea and Southern Guinea, also derived savanna, and then the rainforest, which is typical of most of our yam varieties. It is also said to have a potential yield of 34 tons per hectare, and also has some, it matures within 270 to 300 days. And it also has some outstanding characteristics based on what it is known for. That is its slow oxidation, which means it doesn't turn brownish after a short time when you cut it, which is a very good attribute for any good yam seed, yam crop. It has tolerance for anthracnose, and it has good boiling quality and good taste. It's said to be good for pounded yam, which we know that most of our people who eat yam in this country, pounded yam is the key issue. And it has a high tuber dry matter, and you could say to some extent starch content. So these are the things that we looked at, and from all the other possible available ones, this was attractive to us because from the dry matter and starch, it's a possible candidate for industrial use. And once you have any product that is used industrially, means production will have to be high to meet up the industrial demand. And that's the kind of product that we want to be involved in, or variety that we want to be involved in. Besides that, if it has a good taste also, it's also for consumption, high consumption. So that's why yam was a variety of good interest. And I must say here, I must thank Dr. Asrat for the fact that what we are seeing already with this variety is quite promising and encouraging. So we hope that it will continue like that. Having said this all about the variety, what is it that we can say from all our field trials in the production from the screen house up to the field where we are rooting it as vines on the field? So one thing we noticed about, the variety, I'm sorry that I didn't put this on, is that it's quite a fleshy variety. If you look at it, the body is quite smooth, and you can imagine what the flesh looks like. And it tells us that it has a tendency that it could be a good candidate for, because from what some of the farmers who have produced it are telling us, it has a high chance of being very good when you fry it, like French fries or things like that. So that is why that shape of it and the rounded shape or oval shape of it tells us that it has that possibility. It has also slow oxidation rate, which means it will fall within the category of the best yam varieties, because the best yam varieties are not the ones that oxidize. The good ones don't oxidize. So which is a very good. Then within the operations from the screen house to the field, we noticed that anywhere we had to in the processing, we had problems with mealybugs. You find that this variety was less susceptible. So it gave us the feeling that it might be resistant to some diseases, even though we have been told that it's anthracnose, but we can see also evidently that there are maybe other ones. So this is noted because what we're trying to do here is also to give the breeders our own feedback for either improvement, or they could even confirm some things that maybe hitherto were not known. Then besides that, we found that when we produced them in the screen house, like Dr. Beatrice showed us there, we have the vines growing up there, and we have to cut the single node vines and root them on the field. We find that the losses on yam in terms of that exercise is always lower than all the other varieties. So we saw also that, like somebody mentioned also, that depending on the fertilizer feeding that you give to the mother plants, the vines that you get out of it actually will be subject to the kind of fertilizer treatment that you did. And we have a fertilizer regime that makes the plants come out with very rigid, will I say, midribs, so that when we are rooting, they are very strong, stronger than when we started. And we could see that VIAM had a kind of advantage over the other varieties. So which shows to us that it's also something that is suitable to us, because if it has less mortality or losses on the field, it means it's a better candidate than the others. Not only that, we also found out from our technical team that in terms of the routing, when they root on the field, that within 9 days, you find that there's signs of rooting in the VIAM variety. While in the others, it has to go up to about 14 days before we see that. So these are some of the attributes we found about the VIAM that is very interesting. Within the confines of the tubers that we harvest, particularly when we do the foundation seed production, we also found that per square meter, VIAM seems to have advantage compared to the other varieties that we are using, with about something like about 10% thereabout superiority in terms of yield. So which means also it's likely going to be a heavy yielder, as also has been stated by the breeders. So these are some of the attributes that makes us to feel that this variety is actually a variety of the future, which we hope to further commercialize as we go along. So those are the things that we found about this variety. So having said that, what can we possibly deduce or look at? Like I said, when it is fleshy, it has the chance or the tendency of being a good candidate for, will I say, fries. Just like we have the potato fries and things like that. It has, and we have gotten farmers who have attested to it that, in fact, when they fry it, it is more satisfying than even the Irish potato itself, which makes it very interesting. So that is why that fluffy look, later you see it from the fruit, it is very different from the other normal yam in terms of the shape and its outlook. As I've said also, it's a very rugged crop of variety. It means mortality and things like that will be less. The only thing we found about it is that the leaf color is lemon color. Most times it's not as deep green as the others, but that doesn't seem to have impaired its yield from either the vines or even on the field. Like I said, it has a yield advantage compared to other ones. So having said that, also, we also realize that it is a variety that from the taste of our own people, just the in-house taste, people feel it tastes very well. And because there is a feeling, well, that swaswa is an alata, it is not, it's a good pounded yam for diabetics. Okay, they feel that this also will have those kind of attributes being an alata also. So these are some of the things that we can deduce from this variety that we feel it's a good candidate for commercialization as compared to the ones that we are currently running on with. So having said this, basically these are the things we can say about VIAM. Then I just want to just go to the photo gallery and discuss the process of the seed production itself from the mother plants. So what you can see in the small packs there is the mother plants that we get from IITA, Ibadan. Up till now, like us in Kaduna, we have to get these mother plants in packs, in cartons, and ship them from IITA Ibadan to Kaduna, from where we now feed them into the boxes in the screen house. On the right here, the other side, which does not have the packs, what you see there is the mother plants planted in the boxes, hydroponic boxes in the screen house. Dr. Beatrice said, either hydroponics or aeroponics, we found that the hydroponics works for us because of the ambient temperatures in our environment. So that's why you find our production are mainly towards the hydroponics system. So you can see there, these are the mother plants planted in the hydroponic boxes. And that is how we do our production of the vines that we normally So next after that, I'm going to be showing you when the system finish booting properly. We'll show you the vines grown in the screen house. Still not there. So in the screen houses, we have boxes there that are 1.5 meters by 6 meters, where we grow. In each box, we put something like about 308 plants, mother plants, from where the vines grow as high as up to about 3 meters up. Okay, so that is how we produce the vines in the screen house. And then from there, we cut these vines into single node vines in a process where we put also to ensure that it does not lose, it does not get dried up. We put them in water and take them to the field where we root them. Now on the field, like a question was asked, how do we do this production? We raise beds about 1.2 meters wide, and some of the beds are as long as 65 meters. Okay, those are the vines. They grow in boxes like that, and it's as thick as this in the boxes. So we have multiple and multiples of vines. And here you can see is palmio on one side and AfricaYam on the other side, and you can see the pale green color of the AfricaYam. But one thing that, okay, I skipped in mentioning also, one of the disadvantages we saw in AfricaYam is that it has very big laminas compared to the other varieties, which means that per vine, we have less single node vines from each. That is the only disadvantage we've seen about it so far. But that is what is its characteristics. So now when we cut the single node vines, we go out to the field and we root them 10 centimeters by 10 centimeters on these beds. The idea there is that, but these beds also, we have to also, will I say, mulch the beds with rice husk, like Dr. Beatrice mentioned. This rice husk is supposed to be a kind of material that will kind of reduce the impact of scorching sun from the soil, and also with a shade on top. So this is how we produce it. And we have found this system to be more efficient in the sense that we can get many plant population very high density, so that we can produce, because like I said, from our experience, like Dr. Beatrice has mentioned, the smaller the tuber, the more beneficial it is. Because you find that when you plant them, the survival is quite higher. And also in terms of sprouting, you don't run into problems of having maybe when you have the ones that are cut, mini set ones, the middle usually does not come out at the same time with either the head or the tail or the bottom. So you find that once we have these small tubers that are like eggs or a little bit bigger than that, it is easier to handle, and you are sure of your survival germination percentage as being very high as 98, 95% because of that. So this is the system that we use to produce, and we found this system to be very effective because from this. The question now is, how can we get the volumes that will suit sufficiently our farmers for them to be able to have seed all over the production areas? So by the time we root them, after about 3 to 4 months, we have something like it, because we have to go also and try to string them up so that the leaves can get enough sunshine and grow, and the tubers can grow big. From the standard that IITA has shown us, you can get up to about 3 kilograms per square meter. Sometimes with some varieties, we get 2.5 and thereabout. But then this is the system by which we produce the seed yam. And then when you compare AfricaYam to all the other varieties, virtually the system of production that we are using for now is the same and seems to suit all of them. So having said that, I want to also show you the foundation seed that is gotten out of this system of production. One of the ones you can see that looks oval in shape, that is the AfricaYam. Very succulent looking fruit and very uniform. So what we do is that in the processing, we grade them and package them separately by size. And that is how we sell. On the other side is the Pamio, which is the same you find out with the Asedu and the Swaswa. They're usually a little bit longer and not oval in shape like you see with the tuber. So this is the kind of product that we think if we make available, farmers call them chin-chin. If we make them available to farmers, these are the kind of products that will make the seed system for yam to be very beneficial to the farmers and to the production of yam itself. So in the middle there and on the side, these are all plants of AfricaYam growing. That's tubers of AfricaYam harvested from the field. Yes. And one thing that you have to know also, AfricaYam proves itself in the sense that it has multiple tubers compared to all the others. The others, on the average, you could say you have something like 1.5 tubers per plant, but that of AfricaYam is higher. And because of that high number, you find that the tubers are sizable. So we find that it has also a property that in its characteristic that will make it a very good candidate for a variety that is good for, will I say, the seed system. Because like I said, the solution we think for the future now is to produce our seed yam in small sizes of whole tubers, which the farmer can plant just like he plants his maize. One seed per hole or something like that, or at best, you just one mini setting. Because the mini set is still a good technology, but the thing is that when you mini set, you injure the tuber. And when you plant, definitely from what we have done in the past, when we started this project, we found that the rate of germination was not as good and faster as that of these mini tubers that we are talking about. So mini set is a good technology, but definitely if you have whole tubers that are smaller, they are much more better and give better result, and the farmers always like them. In fact, they call them chin-chin. So having said that, I want to thank the AfricaYam for this privilege, and also the Bill and Melinda Gates Foundation for supporting both the USWA project and the AfricaYam. If not for this, I will not be talking yam. Maybe I would have been talking maize and other things as a seed company. But I want to thank you for this opportunity because yam is really a great crop. And I can say here that it's one crop that I think its impact compared to other, like Beatrice has shown in one of the clips, is much more profitable and better rewarding than any other crop that I know for now. So thank you very much for this privilege.
Thank you also, dear colleague. This is very nice presentation. We will now take 2 questions. Yeah. One from Côte d'Ivoire, the first one.
Thank you very much. I appreciated your presentation. I'm from Amani from Côte d'Ivoire. We don't have a private sector involved in seed yam production in Côte d'Ivoire. I want to know what is the way we have to follow to get the private sector involved in yam seed production in Côte d'Ivoire. And second question, what is the demand, yam seed demand in Nigeria? Do you have a demand for it, the volume?
Okay, let me start by talking about the presence of private sector. I think it is something that the government has to encourage, because seed business itself must have somebody that has a passion for it. If you don't have a passion for it, it's a problem, because private companies makes the system efficient. Because even in the past, you find that what the World Bank had done in trying to establish seed systems, even for cereals, when this initially came, they started with the government agencies and some other arrangements. It never worked. The private sector participation in seed production is a major issue that can liberate a seed system and for the seed system to be very established in the country. So I think you have to look at that area and look at people who may have passion for it, encourage them and let them start the seed system, because seed systems are very important. Like we say, seed is key to output of any agricultural production. Yes. So having said that, you talked about the issue of demand. I'm sure the president of the Young Farmers Association professors will be talking later. Since the starting of YISWA, because when we started, actually the science has been done for the production. But you know, when you come to the field as a seed man, you need to produce volumes. You don't need one barrel. You need trailers and trailer loads of this seed to go around for people to know that it's a seed system. So that is what we are grappling with now, trying to upscale in mass production. So people are waiting and ready to pay for the seed. We have not gotten there. We're just moving there slowly. But with what the scientists have done of recent, I think we'll move much faster now to catch up in terms of the production. And I can assure you, if we produce, people are waiting there because the benefit in terms of yield and quality is way out compared to the traditional yam seed system. You cannot compare the same with what we are doing now. Yes. So, and for Nigeria, in terms of volume, I think we crop about 3 million hectares every year. So you can imagine if we are talking of the old system of 2.5 tons per hectare, you can imagine the volume of yam seed. Even now that we are saying 25 to 50 grams per stand, if you look at how much you are going to crop for a hectare, it's not small. When you say half a ton per hectare for 3 million hectares for Nigeria. So it's a big, big volume that everybody can play in it. Okay, so that's that. Thank you.
Okay, somebody has the question again. Yeah, Prof.
Sorry if this is a bit of an ignorant question, but I'm interested in the link. For example, you're talking about yam chips or fries or whatever. So how does that work then? Are you in contact with those processes yourself, or it's just through the, how does that value chain actually operate?
Yes, like I said, the issue of yam fries and chips came in by virtue of this new candidate variety that we got from AfricaYam, trying to compare them with what we had in the past. And along the line, I didn't want to tell you this story. It started with a very funny story of a man who works in AgriBank, who was our customer and did the production for the first year of VIAM, which we said is a new yam, is an alata, that. And he produced it and he liked the look of the crop and the yield of the crop. So he packaged some and kept for planting the next year because what we gave him is foundation seed. And the wife, one day, just by chance, saw this variety in the store and picked one of the bigger ones and fried it and found that it was very palatable. So unknown to him, they started picking and picking. So when it was time to go to the farm, he came and found that there was no yam. So we then started asking questions from people. And then we realized that even our technicians have been enjoying it, that it is a very palatable variety. So that is how we got exposed to that. So we saw it as a candidate because those who have eaten it, what they say is that it is more satisfying than even the potato chips. So we, as far as we are concerned, we see that once we sell this idea to the industry, we may have people doing things like French fries and packaging them for the supermarkets. Not only that, it has the highest starch content from what we know, definitely industrial quality starch. So it is a very high candidate for industrial use. So those are the things that we found out about it. Well, it's still in the rudimentary stage. The processors, when they get there, they will do their processing in the lab and get all their facts and figures right. But we think that that's a pointer to us. Thank you.
Okay, a last question from myself now. like my colleague from Cote d'Ivoire said, there is no private company there, and the demand is very high. In Benin also, there is no private company producing seed, and the demand also is very high and high again. Now, how your company that is already having very good results can help those countries to establish, you know, a small production unit, so that people will see and take from that side the establishment of their own private company to produce seed for the farmers. How can you help us? Yeah.
Thank you for that question. It's a very interesting one, because this is the kind of thing we are always eager to be involved in, because even with our colleagues who are the Ghanaians in the USWA project, we try to, compare notes and try to help each other. Definitely Benin Republic here, as far as I'm concerned, is an extension of Nigeria. Sorry.
Yes, no, not sorry. It's like that. Yeah.
So
it's like that.
I think it's a welcome thing.
Yeah.
We in Daol Green will be ready. We've done that for some of our colleagues in Nigeria to train their own staff, expose them to, even give them materials that they can take home and start something small.
Okay.
And then from there, if it's getting too much for us, IITA is always there for us.
Yeah.
That I can tell you anytime.
Okay.
Whether the technology or whatever, they are there to help. So I'm
assist.
sure between ourselves, my other colleagues in Nigeria, IITA definitely will do something that will rub off.
Very, very
even
important.
Cote d'Ivoire, maybe because they are farther, but I think it's something we can work out.
Okay.
Yes.
Okay.
Yes.
Okay. Thank you very much, because we need it. We really need it.
Truly, it is very
for
important.
the project and also for the development of yam production in our respective countries. It's something that is really lacking.
Yes.
Thank you very much. And
Thank
now
you
we'll move to the next presenter. What is who is the next now? Yes. Because it was very interesting, no? And current yam market status, export and processors requirement in Nigeria. Okay, Prof.
Okay, thank you very much. We want to first start by appreciating all of you for the work you are doing. We are the primary beneficiaries of your research efforts, and we are very happy when your research efforts translate to money in our pockets. You just heard about VAYAM. Va is come. Yam, the same spelling with yam, is buy. So in this presentation, I will be calling you to VAYAM to come and buy yam. And that means we need to get to the market. In Nigeria, yam continues to be one of the food commodities that tend to drive food inflation. When the price of yam is going up, we are happy, and you can see the women and myself rejoicing, because that means there will be more money in our pocket. For the tubers, this year around May, we witnessed about 60 to 67% increase from a tuber going for about 1,413 naira to 2,350 naira. That was not the same for yam flour, especially the Ola pounder yam flour. What does this tell us? It tells us that the tuber is much more important in the market than the flour. And the reason is simple. Once you convert the tuber to flour, you have only very limited ways of utilization. But if it remains in a tuber form, I used to tell people that if there is any lockdown that like we had during COVID, I can lock down with yam in my house. I can eat it as pounded yam. I can eat it as boiled yam. I can eat it as fried yam. I can eat it as porridge. I can eat it as roasted yam. So many methods of eating yam. So lock me down with yam and I will survive. But if you lock me down with ginger, I don't know how I can survive. But unfortunately, our government will prefer to put money into ginger. And as we speak today, yam is not on the Anchor Borrower program in Nigeria, but many other commodities are. And we are still asking that if yam is a food security crop, and yam is also export promotion crop, then why do you neglect the yam and then concentrate on other tubers? But that is what we have at the moment. We are still trying to find a way to get government to listen to us. So when we try to find out from the traders why the increase in price for the yam tubers, they gave a lot of reasons. Of course, the issue of labor is one of them. Because of our farming system, which is use of the mound, it's becoming very, very difficult for us to get young people that have the skill to be able to make those mounds. And that is why today they will collect money from this farmer, from that farmer, from the other farmer, and disappear. Because there's so much demand on those few that can make those mounts. And because of that, they tend to take advantage of the farmers. What we are trying to do is to encourage our people to move from the mound or the heap to ridges. We have contacted the National Center for Agricultural Mechanization, and we are telling them that if they can give us high clearance ridges, they give us ridges that can give us very high ridges, and they have accepted that they will work on that initiative. Because the yam mound making machine they have produced can even bring down the number of plants per hectare than what we have under the traditional system. We are doing 4,000 plants per hectare under the traditional system. I'm not sure the yam mound machine by the National Center for Agricultural Mechanization can do 2,000. But ridges, we can do 10,000 plants per hectare. And to us, if you are able to achieve 2 kilogram tuber of yam, which is the desirable size, whether for export or for processing, you are talking about 20 tons per hectare. And that is okay for us under the present system. They also talked about the seasonal fluctuations, and that is why we are thinking of 2 harvests in a year. We want to do wet season, we want to do dry season so that we can stabilize the price of yam across the year. So we have 2 harvests, and that will be good for us. There are other things they talked about which may not be of interest to us here. But then we are finding gradually that in the market, there is price differentiation based on variety. So in the future, we are thinking that the type of variety will define the price because some varieties may attract higher prices than others. Right now, the Ogoja variety is very, very expensive in the market in comparison with other varieties. Now, yam continues to be sold by size and number in the markets across Nigeria. This is not fair. It is not transparent. It is not trustworthy. And we are thinking about standardization in terms of how do we market yams by weight. So with the help of Beatrice, who kept telling us that there is a place in Côte d'Ivoire where they market yam by weight, I was able to approach the Federal Ministry of Industry, Trade and Investment, and I convinced them that we need to start selling yams by weight in Nigeria. And they said, is there any place in Africa where they are doing it? I said, yes. So I had to put up a proposal and it was approved. And I took 2 directors to Boake, and I want to appreciate Dr. Amani and Dr. DB, who made that trip possible. I can say that Dr. DB's English has improved very significantly. When we visited, it was a challenge because we needed an interpreter. Again, it was very funny that we sat down with our fellow African brothers and we couldn't communicate. I don't blame anybody, you know, but it was very painful to me as a person that we couldn't communicate. And we thank God Dr. DB was there and he was able to manage to explain to us what the people were saying. But when I listened to him at this workshop, I said, wow, I wish we had him like this that time. Maybe we would have, you know, come back with much more. So they assisted us. We were also interested as a side attraction in the stadium. We are hoping that in the next Africa Cup of Nations, we will beat Ghana. They come to Nigeria, they beat us. We go to Accra, they beat us. We will beat them in Côte d'Ivoire. So you can see us in the market. The weighing scales are generally used in the, you go to the market, they are everywhere. So when you want to buy your yams, you just put it on the weighing scale and off you go. So very soon in Nigeria, we will be adopting a yam marketing by weight. And these guys that followed me were so happy because when they got there, they found out that even bitter cola, even, you know, cola nut, you know, everything was sold by weight. So they came back and they said, it cannot only be yam. Thank you, Yam, for taking us to Côte d'Ivoire, but we want this to be done for all agricultural produce in Nigeria. So that is the recommendation that the federal government of Nigeria is working on now. What are the popular and best-selling varieties in yam markets across Nigeria? They have remained what we got in some 2 years back or so, 2 years back across several markets and 17 states. Our challenge has always been that the varieties that were earlier released we couldn't find them in the market. Of course, explanations have been given here that perhaps some of these varieties were given different names by the farmers. But we couldn't find them, and we started asking, where are the varieties? But I think this is going to change going forward because of what we are seeing. The Wonder Yam, a lot of farmers are talking about it. They say, oh, there is one yam that is called Wonder Yam. They say it is good. And some of the farmers sent the Wonder Yam to me. I want to believe that this Wonder Yam will translate into Wonder Yam in the market. Same thing with the Super, the Nagode, the Favorite, and of course the Vayam that we are talking about. Now, we started asking about these varieties because some of them we were told could give us 40 tons per hectare, and we are doing 10 tons per hectare at present. And we said, if there is a variety that can give us 40 tons per hectare, give it to us. We want to have it because it's like 4 hectares in one in terms of productivity. Now, the next maybe few slides, we want to talk about why varieties can be popular and best-selling along these lines. In terms of production, varieties that are not just high-yielding, pest and disease tolerant, but also early maturing, and heat and drought resistance due to the effect of climate change. So we need to factor in climate change in our breeding program, varieties that are regular in shape that can be planted mechanically, or whose rooted vine cuttings can be transplanted mechanically, varieties that will eliminate staking like what we saw during the field trip at IITA, because the seed yam for 1 hectare will cost us 800,000 naira. But staking alone is about 200,000 naira when you look at the economics of production that we have prepared and sent to government for funding. So if we can eliminate staking, that's 200 naira savings. And that can translate to lower cost in the market so that yams become not just available and accessible, but also becomes affordable. Then the yam harvesting, we have yams that we could harvest mechanically like the Japanese yam that we can plant on ridges and use tractor harvesters to do. That can make it a variety of choice. For export, the size and shape, very critical, slender and smooth. The weight has to be averagely 2 kilograms. And of course, we know that for export is not the ceremonial yams that we want to have. It is just the what to some people is planting material. Especially under what they call the Akpaku system. The Akpaku system is a system where you make very big heap and then you plant a whole tuber to be able to get very big, harvest. That is what the most farmers are practicing now. But we keep telling them that this big one is for you to win agricultural show competition. When it comes to the dollar component of this program, that's not what you need. Yams that can withstand stress, and in the shipment that we did to the US and the UK around 2017 under the former Minister of Agriculture, we found that Hembankwase variety withstood stress associated with the shipment, and the yams arrived their destination more wholesome than all the other varieties. So there must be something about this Mbankwase variety that other varieties don't have. Currently, water yam in Canada is going for $100 for a box. We keep getting this update every now and then from African Choice Market. They do retail and wholesale, and we keep getting market information across other countries to find out how much this is being sold. And I can assure you that with the arrangement we have with British Airways, we can deliver a box in Canada at $40 per box. So if a box of water yam is being sold at $100 and you can do 40 through British Airways to Canada, then you can just imagine the money you get. If you do shipment, by sea, then the multiplier effect is quite high. But you will have the challenge of spoilage. That's why we are exploring export by air, because it will take you just 3 days to nearly any part of the world, maybe except Australia. We have one of our members that took a 40-foot container of yams to Australia under freezing conditions. Because in Australia, the conditions for export are very strict. But she was able to do it. She will be sharing her testimony with us during our annual yam conference that is coming up very soon. So for processing, the yam peeling requires yam that is smooth, straight, because you see the peeling machine cannot work like human beings, where you have yams that have contours and stuff. So the processors also of yam flour, pharmaceutical grade starch, requires varieties that have high flour yield. And Honeywell flour mills have reported this before, requested from us 66 tons per day of Ogoja yam and Fakitsa for 320 days of the year. And this was a demand we wanted to take to government so as to be supported, so we can be able to produce Ogoja and Fakitsa. I don't know whether Ogoja and Fakitsa are improved varieties, but again, that was a disconnect that we had with the program, where we were promoting Swaswa, we were promoting Pamyo, when actually we ought to have been promoting the varieties that the industry needed. We can imagine if Ogoja and Fakitsa were the candidates for promotion, and then there is a company that is ready to take 66 tons every day. 66 tons is 2 trailers of yam with 6 tons on the ground every day. That's huge. We are also thinking about the other processed products, more specifically yam fries. We don't want this yam market to only reside with our people in diaspora, Africans in diaspora. How do we get the Americans, the Britons, the Japanese to eat yam? Because once we are able to get them to eat yam, then we have a bigger market. So we are working on that. I was asking Dao Green, I said, the Japanese guy that just spoke to us, does he eat yam? This is will be good to us. And if any of you can eat, we can, if you allow us, we can take photographs and make noise about it. That yam is not just an African diaspora thing. It's a global commodity that is to be eaten by all. These are the kinds of requests we get. We got this request. They want to make wine out of dried and sliced yam chips. and they asked us to give them 40,000 tons. And they said the starch has to be 67%, the moisture 14%, the fiber constant must be 5%, the sand and silica 3% maximum. And, you know, okay. So the area of aggregation, handling, storage, warehousing, we need yams that are resistant to elastic deformation. You can see that lorry load, the trailer load of yam. Many of my members are here. For you to load and unload yam, you have to be a member of my association. For you to push yam in a wheelbarrow, you have to be a member of my association. We don't call them wheelbarrow pushers, we call them mini transport service providers, in an euphemistic way of making that value chain very attractive to these young people that push these wheelbarrows. And then, of course, those involved in yam transportation. Well, in terms of consumption, it has already been discussed. Don't need to waste time on this. We have a particular variety that might interest us. They call it in the local language alakpa. Alakpa, when you boil it, you don't peel before you boil. When you boil, you just twist the yam a little and you pull out the peel. It comes out just neatly. The aroma is beautiful, but it's getting extinct, and I had to personally move around to find some which I have planted very close to my house. And we want to see how we can generate a lot of it. Okay, so conclusion, I want to invite all of you to use this opportunity, if you can allow me to invite us to the to the YAM conference that is coming up. The flyers are on the table there. If you are interested, please pick the flyers. And we want to plead with you to work with us. In your field trials, work with our members. In that case, we can monitor what you are doing with us. When you are releasing varieties, call us. We will be able to oblige. I want to thank you for your kind attention.
Thank you very much, Prof, for your beautiful presentation. We will now take one question, so time is
Okay, only one. Okay, so I want to ask, we hear a lot about, for example, exporting to the EU regulations, phytosanitary, sanitary regulation. How much of an issue is that with exporting yam?
Okay, it's easier to export Ghana yams to the EU than export Nigeria yam to the EU. The phytosanitary requirements are there. In most cases, now that we have Brexit, with the UK, they are asking us to have fresh trade agreements, which were under the EU government. And I'm heading a technical working group that is developing a UK-Nigeria trade agreement. So these are issues that are part of that agreement that we want to also tackle. But our yams in the UK, for the fact that the Ghanaians don't pay duty because they signed EU-Ghana trade agreement, which Nigeria refused to sign. It's now that we are working on a trade agreement. We are hoping that the issues of duty on yams coming from Nigeria, that makes it less competitive. That is why our market, we are thinking more of US, where we have a GOA than the EU and Britain, where we have to pay duty because we have no trade agreement with the EU, neither do we have any trade agreement with Britain.
Question. Okay, I think I'll come to you. Let's start.
Good presentation. You don't talk about Brazil, but in Europe, Brazil is one. Ghana is second. And how you will compete with them?
Well, we found that the market for yam is just so much. In one of our trips to the United States, we found that so many chain stores, big stores, don't even have yam. Most of the people depend on yams that is sold in small communities. So they come to do their groceries in the big store, then they want yam, they start looking for one corner. So the market is so huge that we think that there shouldn't be issues. We should rather talk about collaboration, not much of competition.
Okay, so that was a very insightful presentation. I also want to add, okay, at a point in your presentation, you attributed some factors that were responsible for high cost of yams in the market, as we all saw it, particularly in the last 1 year or so. You know, I expected to see the issue of farmer header clash as being really responsible for high cost of yams in the market, and I didn't hear that, you know. So what do you have to tell us about that, sir?
We came up with an innovative system, which is the planting of yams in bags. And we are telling farmers that if they don't allow you have access to your land to farm, get cement bags, put good soil in it, and plant at the back of your house or within the vicinity of your environment. Because there is security. If you just have a farm not far away from your home. And if anybody challenges you and say, why are you using my property? You can simply carry your farm from one location to another. It's an innovation that has caught fire in Nigeria. And even big people here in Abuja, at the back of their houses, they have yam in sacks planted. And we are having good yield to show for it.
Thank you very much, Prof. But I don't think that is what Jenny is talking about. If you want to plant yam in commercial quantities, you cannot say you'll be planting. How many sacks are you going to plant? I think in terms of the farmer headers clash, the problem is gradually being solved. It's not as serious as it used to be now. And I think there are a lot of interventions from government to ensure that they put an end to these frequent clashes between farmers and headers. Planting yam in sacks will not solve that type of problem. It's nice if you can plant yam in sacks, maybe for the people living in urban areas, but our farmers mainly are on the field and not, I don't know how you will convince, really yam farmers to start planting yams in sacks. Anyway, that's my contribution.
Thank you, Prof, for the presentation. I just have 2 questions. When they begin to sell the tubers in the market by weight, who will set the price per kilo? Who is responsible for setting the price per kilo? And then the second one is, what is the size of the box for British Airways to charge only $40 to export or to carry the yams?
Yeah, the $48 is the total cost. When you sit down and you look at the cost of procurement, the cost of producing the carton, the cost of transportation, we have sat down, we've done the numbers, and the British Airways said we can give you 300 naira per kilogram, and you can do 7 tons per flight. So we're talking about how many cartons in a ton, 25 kilogram per carton. And then you put them on a pallet and that makes 1 ton and it goes. So is the total cost of landing the box, the 25 kilogram box in Canada or any of these other countries, the US that we're talking about. The issue of the setting up of the price, we found the arrangement in Boake quite interesting because we visited the system where perhaps they use the historical price system. But for us in Nigeria, we are looking at what is the economics of production for 1 hectare. If it's costing us 1.4 million naira per hectare in terms of EOP, and we have 20 tons harvest on that 1 hectare, we can do the maths and that can be the beginning point for our price. But in Boake, they have a management information system, which I even suggested to government now, as we are speaking, that let's go back there and study it much more and see how they are able to come up with the price, you know, for yams, because they are not selling by size and number the way we do here. And of course, if we want to integrate trade practices in Africa under the Africa Continental Free Trade Area Agreement, we cannot continue to sell yams in size and number. It cannot work. In fact, the people in Cote d'Ivoire were wondering how we are doing it. They couldn't understand that you are selling yam in size and number. Thank you very much.
Yeah, thank you very much, Prof. It's not only in Cote d'Ivoire, even Benin Republic. I think we visited the very big yam market there in Benin, and they're also selling using the weighing scale. In terms of the economics, I just want clarity a little bit. When we export using the British Airways, you said it will be, the landing cost will be about $48. $40 per box. How many yams are, how many pieces of yam will be in the box? Yeah. Okay. I think that is a little bit also still very expensive. Because in the UK, when we buy Ghana yams, it's 20 pounds. No, not a tuber. The box is 20 pounds. So if you convert, you see that you know, flying with the British Airways is still a little bit expensive. Anyway, thank you very much, Prof, for your very beautiful presentation. I just have a few announcements to make. There will be a little bit of change to the order that we have in the program. Our lunch will be at 1:30. Therefore, we still have about an hour, which I will want us to utilize to make 2 more presentations. So when we come back, then we'll be able, after lunch, we'll be doing the work plan. So there will be 2 more presentations. The first one will be from Dr. Pelemo, and the second one will be from Eglantin. So we can still have these 2 presentations before we go for lunch. And when we come back from lunch, we'll go back to the discussion of the work plan to be anchored by Dr. Aswat. Thank you very much. So Dr. Pelemo can come to the podium. Yeah, that's the 2 presentations. And then, yes.
All right. Okay, good afternoon. Good afternoon. Yeah, thank you so much. I think we should be a bit excited. The lunchtime is quite close. I want to appreciate the PL and my supervisor for the privilege to share some of the outcomes and the successes we've recorded so far using the semi-autotrophic hydroponics to enhance activities in the breeding schemes for yam as a crop. A lot has been said about this, but of course sufficient to say is the fact that some of our partners who are here may definitely still need to have some more information to enhance their activities at their various domains. So we'll be looking at, of course, SAR has been introduced to us, we don't have to overflow that. We'll look at the significance to the breeding scheme, and of course, how we've conducted series of evaluations to enhance the protocols for tuber formation. And of course, by extension, we also look at a few things we have done with farmer groups as a way of diffusing the varieties we've released so far. And we do also hope and believe that some of our partners will be able to adapt this to also spread the varieties released in their various domains. There are arrays of ways through which we get materials into the SAH. You can get materials from the tissue culture in vitro materials already indexed straight away into the SAH boxes. You have them grown out in the box, and you can have mini tubers that you can then send to the feed to produce your various materials. So just to say that the easiest and the cheapest means of exchanging germplasm is through the TC is the most acceptable standard of sending. But of course, once you have this, you are not afraid anyway as to how you can get the materials acclimatized and multiplied, so you can do this easily. So when you are receiving materials from IITA, yes, we could send a form of mini tubers, but if the condition in your country warrants that you receive materials as an in vitro plantlet, you can easily send them into the SH boxes once you receive it, and you can have it multiplied. As for the breeding scheme, yes, from having your fruits, you have the botanical seeds grown out in crates, and then you can have them grow, the seedlings transplanted into pots. The vines are selected of the selected materials, then also inequally sent into the boxes, and you can also produce mini tubers from this within a space of 150 days. And of course, one of the challenges we've had till now, which we are currently addressing, is getting to reduce the variance, the wide range of the tubers produced in the SAR, which might also be as a result of some of the challenges we've observed, the physiological maturity of the nodal cuttings being sent into the boxes per plant. And what we have been able to do with this is that in fast tracking our breeding processes, since 2019, we have started the introduction of the seedlings from the botanical seeds into the SAH. And of course, in the year 2019, we have about 1,300 progenies introduced. In 2020, 1,460. And this year, 2021, 2022, from the 2021 crosses, we have the plan to introduce as much as about 2,000 progenies. And of course, why we are introducing the materials, we begin our selection right from the screen houses, and this in a way also fast track the breeding process. The criteria for selections are spelled out. We see that we are looking at the vigor and maybe the shoot yield, or if we are selecting for the bushy type for the non-stick breeding system, we can easily observe these traits in the plants before we go on to introduce them. And those plants that do not meet the trait of interest for which the materials are being introduced are usually not selected. What we did initially was, though the tubers turns out somewhat not that big, we felt that the best we can do is possibly to have them raised in the nursery before transplanting them to the field. But we were also quick to observe that a direct straight planting of even mini tubers as small as maize kernels will sprout on the field, and they can give you a good yield. But of course, like I mentioned, one of the things we've also found out, which you may also discover as you go on, is that we do not have a very close weight sizes for the tubers. So they are in a range. Yes, we can sort, we can select, but for our breeding schemes, as much as possible, we want to have tubers that are uniform in weight for planting for this our subsequent evaluations. And this table here is showing the ranges of we have the minimum tubers and the maximum tubers and the minimum from 4.1 to 0.4 is a wide range of almost 1/10. And so we are beginning to look at how best we can focus on the maximum tubers. We are going to get one, and also ensure that even within the bus that the harvested materials are of very close range. And for the tubers that were planted out, the SH plant seedlings, we planted out to the field straight from the box. We also have arrays of tubers from them, having tubers ranging from 51 gram to as much as 124 gram. And then of course, having all of this is some of the works we are also doing, but sufficient to mention is the fact that those seedlings from the SCH buses, when transplanted to the field, you can get seed sizes of tubers from them from 300, as much as 300 gram, and which is actually our target. It is not as though we have not recorded as much as 1 kg, 1.5 kg from seedlings of SH transplanted, but we keep our focus on the fact that the technology is intended for seed, and either we are going to increase the population or adapt some other agronomic practices to ensure that these materials are within the scope of seed that are required for trials and for planting. And of course, the mini tubers we grew out on the field also give us a wide range of mini tuber sizes. And of course, for the germinations also in a way vary in the materials as planted. But of course, all of these are some of the works we need to do, either looking for a way to ensure that, particularly for the materials intended for the breeding scheme, yes, we could go on to have them treated with some hormones so that they all break dormancy at the same time. But for this commercial seed, it's just a matter of selecting the seeds that are ready for planting, and we roll such out in batches. So that may not be a problem. And some of the things we are currently looking at, which we've run for most of this year, is looking at how we could use the plant regulators to either enhance the tuber formation in the boxes or the ease of sprouting of these materials after they are harvested. We've had materials treated with sucrose. We've had the density played around with to see how well this influences the yield of the materials from the tubers. And of course, we've also looked at manipulating the photo period to see how well this also enhances tuber formation and weight in the SAH boxes. We have arrays of substrate we have tested with, because while we were going about setting up the SAH with the partners, one of the challenge was in the absence of the plasma substrate we imported, which we have shared with the partners. When this is exhausted, how do they continue in the process? But of course, we have also figured out the use of rice husk, either carbonized or composted, the cocoa peat, and the sawdust. All of this growth media can be applied in SAH, though with slight variations in plant establishment and the yield. Of course, initially we started by using different fertilizer compositions to formulate what we call the SAH substrate, where we have potassium nitrate separately mixed with magnesium sulfate, all sort of combination. We're also quick to figure out that this appears a cumbersome process, and we went on to test the straight water soluble NPKs, and these have proven to be very much effective. And this also we have shared with the partners, so you can get one straight or ready-made fertilizer for use in the SAH laboratory. And this photo just showed some of the responses we saw in the application of the hormones. For the jasmonic, the experiment is still ongoing, but what we observed was that it has some bit of an effect on the plants, but of course the plants were able to recover. What we normally do is, after 2 months of growing this out, we apply the jasmonic acid at different, we apply the jasmonic acid at different concentration to see if that is able to, in a way, influence tuber initiation and maturity in the plants, in the SAH buses. Of course, we also have some of the material sprayed with gibberellic acid to see what influence or what role this will play or the effect it will have on the materials. And of course, we discovered that the plants that were sprayed with gibberellic acid appear much more greener and lustrous. The ones you saw in the middle that is pale is actually the control without the application of GA. And of course, those, since this enhanced the shoot, we also expect that in a way that when the dry matters are being partitioned, that this is also expected to reflect on the tuber. But for the material which we applied sucrose to, we discovered that that has the application of sucrose had a negative effect on the shoot mass, because the shoot ceased growing. But of course, we're also looking at how well this can enhance tuber formation. Just to mention that many of these have been attempted to see as a way of ensuring that the kind of tuber sizes that we want and in the laboratories or in the SAH meet our demand as a way of optimizing. So we also have results showing the responses of the varieties we tested in all of these different substrate we use. The one in blue represents those ones grown with carbonized rice husk. And you can see, so beyond the TS3 or the other substrate package that you could have, since most of us have the rice husks in our domains, it's just a matter of having them carbonized and we can have them to use in growth. We saw that it influenced the shoot weight and also eventually the tuber. So we are not limited in the type or kind of substrate we could use in the SAR to grow yams. And of course, here, this one is also presenting to us. We're looking at, for us to have a good tuber, normally when we started, we were multiplying, having a maximum of 25 plants per box, so that when I'm sending about 40 boxes to you, you already have 1,000 plants to yourself. That was how Dagreen was getting it from us. But when we switched to generating mini tubers from the SAH boxes, so that those ones can be handy and well managed, able to manage by some of our clients, or particularly the seed outgrowers that are not knowledgeable enough or do not have facility to manage seedlings. So what type of material do we get to them? So the best form of material we could exchange with this are possibly the mini tubers. But of course, at that density, we discovered that some plants were either not having tubers or were producing so small tuber because of the very minute feeding areas they have. So we also looked at what density or plant population can we have the space well maximized and we still have the best of yield in terms of tubers that we can exchange with the end users. And of course, we here discovered that the more the plants in the boxes, the less the number of tubers or even the weight of tubers you can have. So currently for our breeding scheme, we pin down the number of plants that we could use in the box between 5 and 10. That is for those intended for tuber. So currently for this year, 2022, the seedlings we have introduced from our population development are just in the range of, just we pin them down to a maximum of 10 per box. But as for multiplication, for distribution, for commercial seed production, we still go on with the 25 with the intent that the seedlings will be transplanted or used in other medium for further culture. And of course, the technology itself being so amenable, one may want to ask, oh, having produced the plant en masse, what next? What do I do with it? So we have the materials. Once you have them in the boxes as they are, you can have them transplanted into the pot in the screen house as we have in the first plate 1. And then we have in this year given out some of those materials to our sister researchers in the Institute for use in the aeroponics. So they grow out optimally in the aeroponic system and they have tubers harvested in the plate too. And of course, just like Engineer Atta presented at IITA, but also materials from the SH has been planted out into the trough type hydroponics and also performed optimally. We went a step further to also put a shed, have them straight from the boxes transplanted directly to the feed, as you see in 4 and 5. We did that in the past, and currently this year we also did the same thing. The tuber we harvested from the first one we did, the one we did in 2020, is the one in plate 6 you have. And of course, in this year, looking at how lazarus the materials are, it's expected that we'll get even a better yield or a more uniform tubers from this scheme. So as for the prospect and functionality of the materials, it's expected that the AfricaYam partners could work out a similar approach in their respective and various domains, particularly when it comes to capacity building and the dissemination of the clean seeds so produced among interested private sector seed producers, which they could do. And of course, one other thing, other things we have been able to successfully do with this technique is that it has become a very easy avenue for us to have materials already indexed for virus tested, because when they grow out in the box, you can easily approach to view whether they are symptomatic or asymptomatic in a way. And of course, like I earlier mentioned, having in vitro materials with ease of acclimatization and having an absolute establishment in the SAR is a good way to go. And of course, we have also successfully exchanged germ plasm to Sierra Leone, to Guinea, using these materials, and even to as far as Mali. So the sources of explant for these materials are not limited for use, like I've presented. And of course, we went a step further. Having developed a variety, and as good as it will appear, the activity doesn't end there. And of course, AfricaYam project has gone a step further in ensuring that the materials are not just released. So we have also developed an approach to ensure that we do we conduct a demand creation with farmers, particularly those in the yam growing area. So we don't relate with individuals. So we will also encourage our partners that the best way to make a great impact is to identify the farmer groups, the farmer associations. So in Nigeria, currently, we are working with the Yam Growers Association in about 5 states, and we are working generally diffusing the materials in about 5 states. So working among where we have the Yam Growers Association, we approach them. When we have the All Farmers Association, to which yam growers are also a component, we approach even a AfricaYam group that are engaged in yam production. We approach this. 2 things we do the training for them on miniset. So we are not sidelining the miniset completely, because that is what is handy that the farmers can adopt. And of course, instead of giving them the seed, we rather help them to multiply. We got them engaged. They provide the land themselves, and all we do is to take the varieties to them, have them manage, and then we do the planting. But of course, the farmers who provided the land are saddled with the responsibility of maintaining the materials, such that at the end of the season, they can have the materials shared. Yes, we know that what they are going to get might not be sufficient for them to establish their farms within a short time. So we classify that as a demand creation. So where they need more materials, I can call on the grain to let them know that, oh, we have created a demand for this variety in this locality, and you make yourself available to, in a way, provide them materials. And we do hope that more seed entrepreneurs will come on board to also embrace this approach. We have the figures of the number of materials we've planted per location and the number of farmers we have shared this with in this year. Of course, we worked in 5 states this year. We do hope to extend this to cover as many states within the yam growing areas in Nigeria in the subsequent years to come. This year, we scaled out largely the AfricaYam and Aquabata, that is 316 and 201, our 67 and other varieties that we have released so far. We're also going to engage the farmer groups in ensuring that these ones are scaled out to them as much as it lies within us. We do this such that by the time we are going out to check where the varieties have reached in a few years to come, the impact can be much evident for everyone to see. And of course, this showing the locations, the farmers we met, trained, and the feeds stability, the current state of the field. And of course, virtually in every location where AfricaYam was sent to and Aquabata where they were planted, we had absolute establishment. And we are quite happy that the farmers will have access to seed in the years to come. In the south western part, in Oyo and Osun State, where we had the work done, we have those various locations. And of course, we have also in Niger State, in FCT, and in Nasarawa State, where we have extended this. This is just as a way of assisting the seed companies to get them ready, because like I said, this is more of a demand creation, and it will be of good use to them. Of course, we intend to go on to look at further things that we can do to enhance the protocol. The result we got because from time to time we have materials indexed, the result we got this year showed to us from the virology unit. We had a lot of the materials we sent to them for re-indexing outrightly free of all forms, because at the point of introduction, even materials that has bad nut virus are tolerant, and we tolerate such for use. But the one we sent this year and for re-indexing showed that they were perfectly clean. So we are also going to look into this to see whether the recurrent cutting of the vines or subculturing in the lab has a way of making some of the materials to escape virus indexing, because virus infection, because comparing the 2 materials we got this year, we discovered that this could be something that can also play out. So we are also going to focus on this to see how well we can use SAH in obtaining clean material, particularly using the positive selection approach that is fast becoming popular, thanks to Dr. Egewi. And of course, we are also going to look much more into how well we can have more materials, substrate that are locally available for use in the cell lab, and also for enhancing the tuber yield. Currently, we are focusing on the rice husk, and someone asks, oh, in the event that you don't have rice husk, what do you use? But of course, we know that some of these substrates are readily available in arrays of places that we can make use of. And of course, like I mentioned, we are going to go on with further modification and diffusion of the IITA release yam varieties among farmer groups. And of course, partners could also go on, can be supported to also achieve this in their respective country based on requests. And of course, I want to acknowledge the BMGF, the IITA, the PL, and my supervisors, and everyone that has greatly been part of this progress. And thank you very much.
Okay, Dr. Pelemo, for your nice presentation. We will take the next presenter before the question together because of time. Our colleague from CIRAD, please. What do you want? Eh? You find the presentation? Don't you agree? Okay, she can start first.
Okay. Good afternoon, everybody. So I promise you no more figures, minimum text, and mainly images on my presentation. So I'm Eglantine Fauvel. I'm the project manager for monitoring, evaluation, and learning of the RTB Foods project. And I'm making, is it? And I'm making this presentation on behalf of the RTB Foods and AfricaYam project management units. So I will present you the RTB Foods and AfricaYam training on yam quality, which took place in November last week. Some of you actually mentioned this training in their presentation, so I'd like to take this as an opportunity to have a brief overview for those of you who unfortunately could not attend this training, to present what really happened during these 4 days. First of all, let me reintroduce you to the RTB Foods project. So the RTB Foods project is a Gates-funded project, which was designed since the real beginning as a complementary project to sister breeding programs targeting 5 RTB crops. So not only yam, but yam is part of it. So we actually provide complementary funding to investigate on food quality and especially the drivers of food quality, with the ultimate objective of creating tools to help breeders better integrate food quality as a breeding priority along their breeding pipeline. So we are a 15 partner project focusing on 5 sub-Saharan countries. And we are organized around 5 work packages which feed into the other, focusing, so as I mentioned, on 5 crops: cassava, yam, sweet potato, and cooking bananas. And we have been studying and focusing our research activities on 12 food product profiles, or let's say 12 food products. And as for yam, we have studied boiled and pounded yam together. So the idea of the Cotonou workshop came in due to the collaboration between AfricaYam and RTB Foods, and especially AfricaYam Phase 2, during which it was decided to allocate funding for RTB Foods to actually strengthen the capacities of AfricaYam partners on the food quality dimension. But this Cotonou workshop could not have been possible without the really precious collaboration of the Faculté des Sciences Agronomiques of the University of Abomey-Quin-la-Vie. Indeed, so this training was hosted by the FSA team. So we'd like to thank again Professor Noel Akisoe for his really valuable contribution and support to this training. So the training was tailored to increase the capacities of AfricaYam partners in integrating food quality traits in their breeding programs. We had 34 trainees from the different partners from AfricaYam and 33 RTB food speakers and trainers. So you can see it was quite balanced, which actually enabled a lot of interactions between disciplines and between the 2 projects. In terms of discipline, so we had an important representation of food scientists, especially amongst trainers, but also breeding and genetics, agronomy, plant physiology, and pathology, mainly amongst trainees. We also had social scientists among trained RTB food trainers. So this was a 3-day workshop. The 2 first days were actually dedicated to plenary sessions and panel discussions. We started with the review of AfricaYam experience on quality assessment and participatory varietal selection. So we asked each partner to actually explain us how they integrate quality as a breeding priority and at which level of their breeding pipeline. So we had each AfricaYam partner presenting their activities and how they integrate quality. But most of these 2 days was actually dedicated to the presentation by RTB food partners of RTB food results and protocols. So we started with the presentation of what we call food product profiles for yam. So in RTB foods, a food product profile, as we hear it, is for a particular food product, the list of key quality criteria or key quality traits corresponding to consumers and processors' expectations, or let's say preferences. So in this case in particular, we had the presentation of the boiled yam and the pounded yam product profiles by the Benin and Nigerian team. The trainees were also introduced to the main laboratory phenotyping protocols to measure these key priority quality traits at lab level. So we had presentation of sensory, texture, and other main biochemical protocols. And finally, we had presentation on the opportunities offered by mid-throughput and high-throughput tools to predict these key priority quality traits. So we had presentation on NIRS, on image analysis, and hyperspectral imaging. So after these 2 days plenary, we had 1-day workshop, or let's say workshops, because we had 5 workshops taking place in parallel. So all trainees could actually participate in all these 5 workshops. The first one was dedicated to yam sample preparation at laboratory level. from tuber sampling from different varieties, determination of 3 tuber section for analysis, peeling and dicing before steaming. The second workshop was dedicated to sensory quantitative descriptive analysis of half of the samples prepared. So we prepared samples and half of them went to the sensory quantitative descriptive analysis with panelists. The trainees took the role of panelists. So they were installed in boxes or let's say in compartments, individual compartments, and each trainee could actually describe using the food product descriptors for boiled yam previously generated. So they described the different samples coming from the different yam varieties. So the descriptors actually correspond to appearance, texture, smell, aroma, taste, so different main categories of sensory descriptors. And the trainees also had the opportunity to be introduced to the basic principles of sensory data analysis and how to actually show the results of such an activity. The third workshop was focused on textual characterization of half of the remaining samples. So half went to the sensory panel and half of the samples went to the textual characterization using an instrumental texturometer. And for more information, I kindly ask you to refer to Toyin Ayetigbo's presentation, which will take place this afternoon. The two last workshops were focused on the presentation of principles of image analysis for color characterization of yam, and the second one to the main steps for for spectral acquisition using a near-infrared spectrometer. And you had this morning a presentation from Denis Cornet on the color characterization of yam samples using a camera. I should not forget to mention that we had also a one-day boat trip to Ganvier Island Village. Dr. Oseledbe introduced that yesterday in a presentation. So we could enjoy local music, local dances all together. So as I mentioned to trainees, all videos, all presentations from these 3 days have been recorded and are now available on the RTB Foods website. So if you want to access the presentations, you just have to go on our website. You'll have a specific section dedicated to the training on yam quality evaluation. If you go on this section, you'll have the possibility to download the training report on this, so the training report, which was consolidated by an external consultant. And you'll also have the possibility to access all presentations classified per day. So you can view directly the videos on our website. All videos are also available through our YouTube channel because we have a specific playlist dedicated to the AfricaYam and RTB Foods training. So here again, you can find all presentations. But it's not only the content of these videos, which is available online through our website. So all RTB Food documentation is actually available on open access. So whatever the survey reports, the laboratory protocols that we have developed within RTB Foods or adapted within RTB Foods, or the methodological manuals, they are all available. So this is just a summary list of some main documentation we have produced on yam. But if you want to access them, just go back on our website. You have a deliverable drop-down list and classified per type of material. So for example, if you want to have a look at the different standard operating procedures that we generated, just have to click on this section, and then you'll have them classified per type of analysis. So protocols for sensory characterization, protocol for texture, biochemistry, and others. So for example, if you click on standard operating procedures for texture, you'll have the possibility to download the 2 protocols mentioned yesterday. So the first one for boiled yam textural characterization, mentioned by Dr. Adeshokan. And the second one on pounded yam textural characterization, mentioned by Dr. Otekbayo. And as mentioned yesterday as well, we have 2 new SOPs which are pending for 2022. If you want to have a look at our methodological manual and training material, here again, for example, for texture, you have the possibility to download all support mission reports consolidated by Dr. Ayetikbo, who is our focal point for texture in RTB foods. Last but not least, I should mention that we in RTB Food produced a special issue for the International Journal of Food Science and Technology last year. So thanks to the Bill and Melinda Gates Foundation, this special issue is available on open access, and you have the possibility to find 9 papers related to yam. And this year, or let's say in 2023, there are 12 new papers coming in on phenotyping methods and genetics of yam quality, because currently the RTB food partners are actually working very, very hard to consolidate a new special issue for a new review, for a new journal, which is the Journal of the Science of Food and Agriculture. And on this, I'd like to thank everybody for your attention, and especially the AfricaYam project for this opportunity for collaboration between our 2 projects.
Okay. Okay, thank you very much. I remember it was a well-organized training, very good, very interesting. Now we will receive the question for the 2 presenters. Yeah, 3 to 4 questions.
Thank you very much for the 2 presenters. The first question to Pelemo. I saw in your plan that you will, future plan, that you will continue the efficient of scaling of the yam varieties. I'm just wondering whether or not you also have a plan for adoption, to conduct adoption and even impact in the future of these varieties, how farmers are actually using it continuously. And the next question is to Fao Valley. Okay, the training looks, from your presentation, looks very interesting. I'm wondering whether at the beginning of the training, you had some kind of pre-evaluation to test the knowledge of the participants, and after the training, you also try to evaluate and see whether their knowledge has increased in terms of immediate outcome. Thank you.
Can I? It's okay. All right. Starting from the rear, what we first did was to visit these farmers. Some of them had previously worked with the ISWA project. And of course, what we did was to first conduct a survey among them. So it was in the course of this surveys that we identified these farmer groups that are open to adopting our varieties, open to seed production. Some of them already doing it at a minimal level. So all we just need to do is to conduct a training. And of course, these farmer groups, some of them also will have their leaders among those that we are conducting our own farm trials with. So they are quite familiar with the entire process. But of course, going forward is the fact that we also have to look, if we test the adoption level and how well they run with this seed diffusion system, we also intend to scale that out to other farmers. That is for that. For the second question, please.
No, this is too much for, yeah.
Sorry. Thanks for this question. Actually, no, we did not assess the level of knowledge of the trainees before the workshop. But as I mentioned, we gave them the opportunity to present their views on quality traits and how they integrate quality in their breeding activities at the real beginning of the workshop. And then we had a final evaluation. So after the workshop, we asked them, let's say, more open-ended questions, of course, to assess whether the training answered their questions, their needs, and whether they feel like they could apply what they've learned directly in their institute. But of course, you know, you never know if it has been done or not. So what we are doing now is we are carrying out our a study on outcome evaluation. So just to assess what are the changes that may have occurred due to RTB food activities and results, even partly due to RTB foods outside the RTB foods framework. So we are having different activities, from individual interviews to workshops, to assess how RTB foods has impacted the breeding community, which is actually the breeders are our next users. So we are currently carrying out this study, and Claudio Proietti is now here with us, so he's the one responsible for this study.
Sorry, Weber, just to add that I think Dr. Asrat and Dr. Patrick are here. They will be in the best position. They are my supervisors. They will be in the position to ask, to answer the question as to whether we'll be able to conduct adoption. Possibly this is can be maybe within the lifetime or the lifeline of the project itself. Okay, you have responded. Okay, thank you.
Yes, a question about the SAH. I understand that technology is a property of a company, is patented. What kind of arrangements do we have to use it? I tried to engage the company for rapid multiplication of sweet potatoes at NC State, and honestly, it was not economical for us. So I don't know. I wonder, everybody's using it. Are we paying a flat fee? How is the arrangement? Maybe you don't know how to answer that. Maybe somebody else can answer.
No, I can. Professor Bosen would like to respond to that. Thank you. Yeah.
Not really is the answer, but I know we pay a license fee. But actually, do you know how much we pay? Yeah. So we've paid a site license fee, Carlos. That's what we're understanding. So we have paid that, but I think it's still because of the other crops, yeah, that we're using. Also, the SH technology becomes economical. Is that right? Yeah. Yeah. Okay.
So another question on the SAH. Usually we see them with the covers on, the clear plastic cover on. When you're going to be transplanting out, do you remove the covers for the last cycle so they become more used to a less humid environment?
The lids are only needful for the cuttings that are between a day and maximum of 14 days old. The moment you notice that the cuttings have rooted, on the verge of producing a new shoot, then you can take the lid off. So they grow out without the lid, and you can keep them for additional 60 days before you send them to the feed, depending on the growth of the plant. So the lids are only necessary just to humidify the environment for the cuttings without root. That is at a very early stage.
Okay, no doubt. Yeah. Emmanuel.
Thank you. My question goes to Pelimo. I saw that you used the carbonized rice husk in the medium, the different media that you are trying. I don't know whether you are using that only or you are mixing with topsoil. because I would imagine that if it is carbonized husk only, the nutrient retention, water retention might be of a problem. So could you clarify that for me?
Yeah, it's all right. So the same way you have the fatty plus or the TS3 packed in the box, and then you have the dark cortex sent into it, is also the same way you have your carbonized rice husk also. Just you are now using it to replace the fatty plus you are currently using, and you still have your cuttings producing root and imagine just the same way you have it in vine cuttings, and you still have a good result with that. You know, the nutrients you are applying are not draining away from the boxes. So except you open the lid, that is when the substrate can dry out a little bit because it cannot retain moisture like you have in the TS3 or the fatty plus. But of course, what that suggests to us is that you might have to do a more frequent watering or fertigation in the boss till the plant fully grows out. So you can get to test that once you arrive Tamale, you have a similar result, I want to believe.
Okay, my suggestion is that we have government approved and recognized farmer associations. I would think that if we work with them, it will be more sustainable than just random farmer groups that after the trial, that knowledge does not diffuse into the larger group.
Yeah, thank you. So I think the All Farmers Association are well recognized. All right.
I think to give more information to Professor, remember 2 years ago, we had a meeting calling all the farmers through your network. And then we use those farmers association as entry point in order to expose those material in a bigger plot. So we are not looking for new ones, but they are our contact from the first meeting. So it's part of your group.
Okay. One question, please. Yeah, I want to find out what is the length of time, say from when you cut into a container until harvest in the field for those that you are going to transplant. Yeah, I'm asking the question because we started with the leaf bud cuttings by rooting in the screen house before taking to the field. But when we transplanted, it was observed that for a month or two, the plants just stay, they are not growing, they are not dead for those 1 to 2 months before growth will take off. So I don't know what's your observation.
Yeah, for the seedlings from SAH, maximum 10 to 14 days, the plants have rooted. At 21 days, you have new shoots emerging from them. So when these shoots grows out and you are able to have at least 2 to 3 leaves from the shoot, you can send them straight to, and they are able to attain this in the first 30 to 34 days of the cutting. So you send them straight away into the feed, preferably under shade to avoid the shock. Don't forget they are coming from a controlled environment that not harsh. So we place them under shade so they can remain under that shade for another 1 month before you take the shade off, by which time they had fully established in all within a range of 6 months from the date of introduction into the new boss to transplanting till harvesting. The tubers are ready. Like the one we transplanted to the feed in June this year, by November, they will be ready for harvest. Have I answered the question? Okay. Like we said, the yield also varies. With on the average, we had between 200 to 300 gram seed sizes, and of course, We had a few outliers, which are up to 1 kg from the very first attempt we did. But like I said, that is not focus. As much as possible, the transplanting we did this year, we limited the planting space within the ridge to 20 cm. So as a way of ensuring that the feeding area is reduced, so that we can have the actual seed size. Where the feeding area becomes large, especially for a latter, you are going to have above 1 kg.
One question from
just wondering, looking at the wide variation in the seeds size, I'm wondering if we know that if you take a piece from the head region, it does better than that of the tail region and then the middle. If you isolate them, group them by where they came from, and then clean them, multiply them, the vines, and follow up, I'm sure we will be reducing the variation. So I'm just wondering that if we do that, we can have more uniform set than.
Yeah,
so it's just a suggestion to look at it.
All right. So, Prof.
Thank you so much. I'm just wondering, why are you worried about producing uniform tubers, mini tubers? Yet you can focus on number, on quantity, and then you grade, so that when you plant your trials, whatever, you use the same grade of seed according to size grades. I think that's what is done in Irish potato, for example. Even maize production, they grade the kernels, small, medium, and large. And then the growers would say, for my mechanization, I want the large flat seed or large round seed. You buy now according to the culture of production. Are you considering that, or is already doing it?
Sorry, I didn't hear you clearly.
I'm saying, why are you worried about the uniformity of the many tubers in seed production?
Okay, yes, for seed production, we are not, for commercial seed production, we are not worried about the sizes of the mini tubers. We can actually grade them and then we roll them out in that order. But for the evaluation and breeding scheme, the sizes of the tubers that we are planting matters. As much as possible, we want the variation in the tuber weights or tuber sizes very negligible, so that do not become a factor as we evaluate the yields of the progenies.
My question is the same. Why don't you grade at that level as well, so that you use seed of the same grade for
it's okay.
each trial?
It's possible. Currently, like we have like 10 cuttings per boss. So if we are able to find, say, 5 of those that falls in the closest range or bearing the similar weight, then we can set those ones aside for use. And then in a way, we could also say that the sizes of the material planted also become a factor to be considered while we are feeding the data into the database for analysis.
Okay, I have been informed that some colleagues online are asking questions. Yeah. Sorry.
The size uniformity we need for we call it SH mini tubers coming. The maximum we can get up to, if we retain in the box, is about 5 grams. The minimum below 1 gram. So when those smaller ones, we store them, and I think most of the time they lose their viability. So that's why we want to reduce at least the size of those smaller ones, a little bit bigger. So whenever we move to the next planting, the bigger the size you plant, the bigger tubers you also get in your next harvest. So that's why we are trying to see if there is a possibility in order to reduce the variability so that at least when we have small variability, then even when grade, then within that grade, you don't want like 4, 5, 6 grades, at least 2 or 3 grades. So that's why we are looking for optimizing how we, the way we can get at least a uniform tuber size.
Okay.
So, oh yeah.
Yeah, we have a question here on
Online.
how we can use SAH to introduce advanced breeder lines in the absence of tissue culture. Is the same approach we have. Currently, we send botanical seeds pre-sprouted into pots in the screen house. What is advised is that if you want them outrightly clean and isolated from infections, you generate mini sets from these advanced lines you intend to multiply in the SAH. Such are planted out in the pot. And once they grow out between the, I say 30 to 60 days after planting them, or even up to 75 days, you can approach such plants in the screen houses, and then you excise nodal cuttings from them, and you send such into SAH in like manner as we are doing currently for seedlings from the botanical seeds that we are introducing into the SAH. And I think that is a perfect way to go. We have also attempted to introduce materials from directly from the feed into the SAH, but of course, that is not in a way advisable because you cannot tell if the plant had not been infected on the feed as of the time you are introducing them. But of course, having the mother plant grown in an isolated area using miniset potted in the screen house, you can take materials from such and send them straight into the SAH, and you can have a good establishment. Thank you. I hope that answers your question.
Okay, and then do you still have some question online? No.
No, that's all.
Okay, dear colleagues, I think there is a time for lunch. I would like to thank all of you. We'll break, we'll go for lunch, and then we come back in 30 minutes? No, 1 hour. 1 hour time, okay. Half past two, we'll be here. Continue.
Like one more presentation left by IITA, and then after that one, we will have a discussion of the work plan, which will be led by Dr. Asra, Dr. Patrick, and myself. And then after that one, we will have a discussion from the review from the TAG member, and then we have their own appreciation, and then we have the end remark, a closing by Jim, then the closing, the final closing by Dr. Patrick. So not to waste much time, I will call on IITA to give us his presentation on, sorry, let me try to catch up. Evaluation of the texture of boiled yam and pounded yam in relation to sensory attributes. So, IITA, back to you. Please, can you yes, this is
Oh, okay. You have to click, look at, just click enable external content.
Okay.
Yeah, enable external. Yeah. Yes. So good afternoon, everyone. Nice to be back after the break. Just to introduce myself, my name's Oluwatoyin Ayetigbo. I was just newly recruited by CIRAD in November last year to take over from Layal Dadu, who was the focal texture, focal point for texture in CIRAD. So I will be presenting to us on the discriminant textural methods for pounded yam and boiled yam in relation to sensory and consumer preferences. I work with some of these contributors, Santiago, Dr. Christian Mes, Dominic, and Domingo Romain from our lab in Montpellier. So this is just a brief outline of the presentation. And to start with, we all know that the RTB food project is focused on the linkages between heritable RTB crop traits and consumer preferences for all the RTB food profiles. And there are two major food product profiles for yam in the RTB foods, which are pounded yam and boiled yam. And we, from experience, know that from the two species of Dioscorea that we have, the rotunda and alata species, the rotundata species is particularly more preferred for pounded yam, while both species could be preferred depending on the properties they have for boiled yam. So based on this, it's also important to note that we need to develop reproducible and discriminant textural methods instrumentally to be able to measure this key sensory texture for boiled yam and pounded yam in the project. So there are a number of instrumental texture protocols that were developed, such as the texture profile analysis, the penetration, extrusion, uniaxial extensibility, and the lubricated squeeze flow. So based on all these different textural protocols have their various use in the context of the RTB food product profile. And developing these SOPs will help to improve the screening of large populations of yams for specific selection for genotypes for advanced breeding towards meeting consumer needs. In brief, some of our partners that we work with, especially in Nigeria, Bowen University, NRCRI in Umudike, and IITA in Ibadan. We also have Benin, our partner FSA, University of Abomi Kalave in Benin. And these are their product profiles that each of them is handling. For instance, in Bowen is working on the pounded yam as a food product profile in Nigeria. And this, from the work package one, we have found out the key sensory attributes that are preferred by consumers of pounded yam in Nigeria, such as stretchability, moldability, and so on. These key sensory attributes need corresponding instrumental attributes, instrumental protocols that need to be engaged to be able to measure these sensory attributes. I'll make just about two mentions of examples of these. For instance, some of the case studies that we have seen, for instance, in Bowen, using the texture profile analysis, we have found that some of the key discriminant instrumental attributes for pounded yam include gumminess, hardness, and adhesiveness. And we have found significant correlations between instrumental and sensory protocols. For instance, we see chewiness, gumminess, and cohesiveness are significantly related to the moldability and stretchability of pounded yam as preferred by consumers. While in Benin, we also, in Benin, we have for boiled yam, key discriminant instrumental attributes such as hardness, adhesiveness, chewiness, and cohesiveness are important to be measured. And we have found very good correlations between firmness and hardness and area under the curve for different textural protocols. So as I mentioned earlier, some of the case studies I'll be rolling out so that we see specific cases. For instance, in Benin for boiled yam, 15 genotypes of yam was boiled, and they used the TPA protocol, which we found to be repeatable and discriminant. And we also found that it can provide more textural information than penetration protocol, which just measures only two parameters. But the TPA can measure actually up to 7 parameters, such as chewiness, hardness, cohesiveness, and so on. So the most discriminating TPA attributes that we found for boiled yam were hardness, adhesiveness, chewiness, and cohesiveness. And what we also found out is that the TPA protocol is often less discriminant than the next method, which I will show you, which is the penetration method. The same 15 genotypes were also measured for boiled yam in Benin using the penetration protocol, which was also discriminating and repeatable. However, just like I mentioned, we could only measure limited numbers of parameters such as hardness. It will also be interesting to mention that penetration method usually is considered to be less repeatable than the texture profile analysis protocol, but it's often more discriminant than the TPA for boiled product profiles, such as boiled yam. And we can see here an illustration of that. A discriminant analysis of both protocols shows that the penetration protocols seem to be much more discriminant, a little bit much more than the TPA, which seem to have a lot of overlaps between the genotypes. There is also a third method, which is utilized by IITA yesterday. Mr. Michael from IITA showed us an example of this. What is important to measure is that we found out from the series of experiments we conducted in NRCRI, Umudike, and IITA that the TPA sometimes does not result in discriminant analysis depending on the genotypes that you are measuring. If you have very contrasting genotypes, TPA is going to work well. But overall, we found out that extrusion is a better protocol, which is more repeatable and discriminant even than the TPA. And the extrusion, for instance, extrusion hardness was a very discriminant attribute using this protocol. We can see very clearly here in an illustration of discriminant analysis between extrusion protocol and TPA. You can see very clearly that the 4 similar genotypes for both protocols, you see that the extrusion is much more discriminated between the genotypes than the TPA. So there's an SOP that IITA is writing currently on that and will be validated in not too far from now. Now in Bowen, for the second food product profile, pounded yam, they use a different texture analyzer than the ones others use, but we found very interesting results from there. We developed the standard operating protocol for pounded yam texture instrumentally by using 4 genotypes of yams that are discriminating collected from IITA. Interesting results were that we found very good repeatability and discriminants. So TPA could be used to discriminate between the 2 species of the Scoria that we have, Alata and Rotundata. As you can see on the principal components analysis, you will see very clear discrimination between the Alata genotypes and the Rotundata genotypes. And we can also associate, for instance, the land races such as TDR-IGN, which is a land race that is commonly grown by local farmers, is much more related to genotypes like TDR100048. And you can associate that with specific textural attributes. For instance, like chewiness, cohesiveness, which are key discriminant instrumental attributes for pounded yam. But we also found that we found significant correlations between the instrumental protocols and the sensory protocols that are measured by trained panelists. For instance, we found very good correlations between chewiness, gumminess, cohesiveness of pounded yam, and moldability and stretchability, which are key sensory attributes. In Benin, we also carried out a extrusion protocol to determine the instrumental texture properties for 6 genotypes of yams, one of which is an Alata genotype. The Aga is actually an Alata, while the others are Rotundata genotypes. The interesting thing here is also that we have very good discrimination between all the genotypes, and on the PCA we can see that it's possible to classify the genotypes into 3 specific classes: the good varieties for pounded yam, the intermediate varieties such as Yonadu, and then we also have Aga, and Dodo, which are the poor varieties with poor textural properties. Overall, we found that all the methods generally can be repeatable and discriminable. However, it's best to use extrusion and penetration protocols for boiled food product profiles, while for paste food product profiles such as pounded yam, it's best to use the TPA or the extrusion protocol. In all these interactions with these labs on pounded yam, we found that stretchability was variably correlated with some instrumental protocols, but we needed measurements that textural measurements that can directly measure stretchability, which is often analyzed by sensory panelists. So to find corresponding instrumental methods that can be mimic, or let me say explain the stretchability of pounded yam, we tend to develop new standard operating protocols in CIRAD so that we can have veritable tools to be able to measure stretchability in pounded yam. Since stretchability is one of the key textural attributes that is preferred by consumers in pounded yam in Nigeria and many other parts of West Africa. So like I said, this SOP will help to provide mid-throughput protocols to screen large populations of yam to select yam genotypes for advanced breeding towards consumer adoption and preferences by consumers. To do this, we consider 2 new standard operating procedures, the uniaxial extensibility and the biaxial extension by lubricated squeeze flow. For the first method, the uniaxial extensibility, we use the texture analyzer. This is the standard texture analyzer that is used by most of our partners. And we use this texture analyzer with a standard probe, the P1SP probe, and the tortilla paste bus rig. So with this system, you can measure extensibility, which is a form of measuring the stretchability of pounded yam. So to do this, we have the yam is being diagonally split into halves, and they are mixed to give representativeness, diced and then cooked, and then they are incubated for a period of time in incubators for relaxation and temperature equilibration before they are rolled into a sheet, a dough sheet, which can then be analyzed texturally, as you can see there. To find relevant sensory attributes that correlate significantly with these instrumental measurements, we, in Benin, University of Abame Kalave, 13 trained panelists scored for stretchability and moldability for 6 varieties in 3 replicates using a standard semi-structured scale. And there was also a consumer tests that were conducted, consumer surveys that were conducted in 2 regions of Benin, where about 100 participants scored between 1 to 9, on a scale of 1 to 9, the overall likability of the pounded yam from these 6 genotypes. Before I go to the results, it's critical to mention for this first methodology, it's difficult or almost impossible to use this protocol for materials that fracture or is too adhesive. So if you have yam genotypes that are too adhesive, then it's really difficult to use that protocol. For instance, some Malata species of yams are really difficult to use. And for the results, we found very good accuracy and repeatability and discriminants between the 6 genotypes. But initially, we developed the protocol by using 4 contrasted genotypes, and we can see the discriminants between the genotypes here, both on the PC and the discriminant analysis, and a significant correlation between instrumental area under the curve, which is the work done to structurally destroy the material, to stretch it until it structurally fails. And between that and the stretchability, we see very significant correlation. We used an increased number of genotypes, and we also found good correlations between consumer likability and some of these instrumental parameters. In Benin, like I said, the consumer test was conducted and thresholds were set to determine the range of these parameters that will be acceptable for consumers. The second method is a biaxial extension viscosity, the LSF method. It's based on the principle of compressing the material between 2 plates, one stationary and one mobile. And basically we measure the force, time, and the distance are recorded here. And we have a standard Excel spreadsheet to do some calculations in order to determine the bi-extensional viscosity. This is the setup of the instrument that is used. The particular probe we use is a P20P probe and the HDP platform. This is the process by which we conduct this. We produce the yam dough following the SOP by Bowen, and then we cut out the samples, and then they are lubricated and kept incubated and demolded, and then they are measured, the textural properties are measured. This is an example of the profile of for the bi-extensional viscosity. And just like I mentioned earlier, conversely, in this case, the biaxial extension viscosity methodology is much more advantageous because you can use this for materials that are brittle. So like the Alata genotypes of yams, you can measure the textural property with this protocol, unlike the other method. So we found very good significant differences between the genotypes, which shows that they are discriminable, as well as the cooking replicates. The replication was not significantly different from one another, meaning that the protocol is reproducible, and very good discriminants between the genotypes. For instance, we can see the genotype Laboko at the extreme, which is a very acceptable, consumer accepted genotype in Benin, compared to Aga, which is the Alata genotype, which is Andodo, which is associated with Aga at the extreme end. And this can also be classified hierarchically. We found very good correlations using this methodology. We found very good correlation between instrumental and sensory attributes, as well as between consumer overall likeness and the instrumental protocol. So based on this, in the future, we plan to do a training in Benin, no, in Bowen, IITA, and NRCRI, and perhaps the Benin team will be joining us in IITA to be trained on these 2 new protocols. And we hope to also extend the application of this protocol to some other food product profiles, but not for yam, for cassava like eba and fufu. So we hope to complete the SOP on the extrusion in IITA soon for boiled yam. And we also hope that very soon we'll be having 6 publications in the GSFA special issue on RTB foods. In the phase 2 of the RTB foods project, we hope that we could study on the effect of storage conditions on the stretchability of pounded yam as a key textural trait in order to study how these genotypes behave in storage. So we want to thank the Bill and Melinda Gates Foundation once again for sponsoring the project, as well as CIRAD for coordinating the project, and our partners all over Nigeria, especially the UACFSA from Benin, IITA, Bowen, and NRCRI. So thank you very much, and hope to get questions from you.
Okay, thanks so much. I can see there's already a lot of interest in your presentation, but I will try to give a, let's say 2 questions, and then including myself. That's mean remaining only 1 question. Okay.
Okay, so now I've listened to quite a few presentations on quality and so forth. So I'm sorry if this is a little bit of a stupid question, but is it possible to have a yam that is good for boiling, pounding, and processing into chips or other products? Or is there something intrinsically, not possible about that?
Yes, I think Benin has been able to express to us that one of their varieties, Laboko, is really good for both pounding and cooking. And I think in Nigeria also we have this Igagan variety. And for processing, I cannot say about that, but I think could be, yes.
Okay, on Dr. Busi.
Thank you, Toyi, for the presentation. I just want to maybe ask, these methods, are they standard methods which you have modified, or they are new methods which have been established to measure the texture? Because if they are standard, then what modification did you use for it to be able to measure the extensibility?
Okay, thank you very much, Ma, for the question. These protocols are new protocols that we developed in CIRAD, and the actual standard method that can be used for extensibility for dough materials, especially from wheat, is the Kifa dough extensibility rig. But we don't have that with any of our partners, and neither do we have in CIRAD. So we developed these methods as proxy methods to measure the extensibility of pounded yam. So if we get finance to get this Kifa dough extensibility rig, is the actual classic system that is used to measure stretchability, extensibility of.
I just have a follow-up. And so then if there are new methods, is there a plan to actually send it out for evaluation or review by textural scientists who are internationally renowned? Because this will be a very good contribution of RTB Foods if we can have that methodology reviewed extensively internationally, because it measures African foods, which we don't have.
Yes, ma'am. Thank you for the question. I think based on this, we wrote a manuscript on this, and it will be reviewed by the GSFA in the GSFA. So we hope that we can get feedbacks based on this publication. And I think perhaps we can promote this to textural experts elsewhere and see if this can be. But I think for us, we use it as a method, as a proxy method, because these are adaptable and they are not too difficult to conduct in the lab.
Okay, thank you for your presentation. I have a question about the hardness. As you mentioned, hardness is very important for boiled yam quality. But what is the threshold of a value we can declare if one variety is good for boil or not? Because I'm now working on this data for QTL mapping and so on, but I have really difficulty to discriminate in my population which one is good or not. So we really need to have, I know that Dominic will talk about it, but we really need to have a value where we can declare if our material will be good or not.
I think.
Okay, before Dominic answers first, Benin has worked on this and they have thresholds values for the extensibility of pounded yam. But for hardness, I'm not sure maybe they have that, but maybe we don't have that data from them.
And
But for pounded yam, we've done for extensibility. For boiled yam, there is no, I'm not sure if, but Dominic can shed more light on that.
No, we are in France. There is few yam in France. We have bought from Ghana, we have bought from Brazil, we bring from Guadeloupe, we bring from Benin. But now that it's time to develop panels, sensorial panel with texture measurement to develop, to establish the threshold. This is the QDA method, is a method who is known for consumer testing. We need to put a threshold for the breeder. And this work is to do now in the next 2 years, because we need to have this value for you for your genetic analysis. And this is, you can do only with the breeder. You need the diversity, you need the diversity of yam ready to be analyzed, and consumer testing, and also sensory panel. The test was developed in France with imported yam, and now it's open to be tested in all your breeding program.
Yes, and I think in IITA they also have, they've done some consumer tests on yams, but I don't know if it's for boiled yam or just pounded yam. For boiled yam, so they may have some threshold values there.
Any other question? If not, probably I can just tell a small story of 1 minute before we continue. We are talking about a pounded yam, which is kind of very good for where I come from, especially Benin Republic. When you go to the northern part of Benin, if the yam is not so hard, they will not eat it, because they need that one to be in their stomach when they go to the field to work for a long time. So the softness is not something that in the northern of Benin they will accept. They need a yam after pounding very hard. When they will eat it, you can stay up to 8 hours in your stomach, so you can work very well in the field. So thanks so much. So the next discussion now is the final work plan and discussion for the breeding program in Benin, Cote d'Ivoire, Ghana, and Nigeria. Then after that one, we'll hear from Dr. Astrat and Dr. Patrick, what are the plans in East Africa and then the Central Africa. So Dr. Astrat.
Okay, thank you very much. Yeah, I think for the last 7 years, we made significant progress in yam breeding in terms of developing new genomic resources and also utilizing that resource in the breeding program. We have also optimized or modernized our population improvement pipeline. We try to utilize new source of alleles from gene bank as well as popular farmers materials. And we also developed some new generation of yam clones for further testing and then released on the seed system. And most importantly, I think together all the partners they have released in this process, I think more than 20 varieties, and there is now work also going on to push those materials into the seed system in order to like demand creation, in order to facilitate the delivery of those materials to the farmers. And a lot of progress, I think, have been made through the RTB food project, where we also learned the quality assessment and then implemented in our selection decision. So these are some of the achievements we have made. And for the next, for this year, there are some materials under field. This is the season, middle of the yam season, where we have populations and also we have advanced clones at different stage of breeding program. We try to also develop our product profile the breeding product profile as well as the food product profile. And from our side, I think we continue to evaluate those materials which is planted. And yeah, for the traits we have built, we will identify that. And we also have some of the materials which we also and verification, especially in Nigeria, we want to push those materials to be released on those in-demand creation trials, maybe together with the product manager. We will see how it can be done for like the product launching in those areas involving the Farmers Association and all this. And the national partners also, I think they can also speak. They have received from us regional trials. So our next, I think, I would say way forward is have more strong regional network. So where we can share materials among all of us, contribute the best materials, clean them, and then put together in a testing network, not only in 3-4 locations, but go more to on farms and any available station, and also use of the TPA, the TPE we have already redefined to use those for testing. So I think for us is the next step is more of aggressive testing network where we can take all our good materials to the next stage. And also those 20 something varieties which are released, we need to work very closely with the seed program so that can be taken by the seed system. And maybe at the end of maybe for the 2 years, that may give us a good time for to see whether we have an impact, especially for the gain we are going to achieve at the farmers level. So the testing network on station as well as on farm, if we intensify, I think that will help us to have a good measurement of our progress, genetic gain in the farmers field, as well as genetic gain in our research fields. I think this is the major direction we want to follow. And also in addition to that, in order to refine our, maybe to increase our accuracy, we give more emphasis on the seed multiplication, having a quality planting material for trials in order to move away from using tubers coming from the testing plots and have a separate seeds multiplication technique. And we have the technique there. Then we need to, I think, resource reallocation to have a dedicated seeds multiplication team for the breeding testing as well as for the delivery. I think this is the focus we're going to use. And also for improving accuracy, we have to utilize the markers we have developed, the QTLs we have to identify. We need to integrate those in the routine breeding program to validate and also use the historical information we have generated from all the trials. And again, look for more markers that can assist our breeding program. And we have also new partners we want to extend to East Africa, so Ethiopia, Uganda, and the DRC Congo is part of, I think we are there now in the same, they are boarding the boat. So in this area, I think our focus is to understand the preference, like what is needed there. And the same times you want to also think about sharing the germplasm. So I've been discussing with also the pathology team. So if we can go for like the survey or maybe diagnostic of the pests and the disease, and then how it looks like, are they different from West Africa? Are they the same? I think this will be our priority. And while we're doing this, have a fast track variety testing of advanced clones in order to have like within coming 2 years, either assembling the material they have already with them, and also sending new materials to come up with trials, and at the same time also have a training. I think that's what we are discussing and putting in the phase to improve their capacity for handling the breeding process in yam in terms of germplasm handling and selection, data analysis, type of data they are going to collect, so that they will be also the same level as those we who here in the Western West African side. So these are some of the area I think we are planning our side. But most important, I think, is in all our selection or our evaluation, food quality, depending on our capacity, will be our, I think, focus for making the materials to the next stage. So this is, I think, from our side, we are preparing for that, putting all our advanced clones into virus screening, and then we also have, I mean, for virus cleaning, and also try to multiply some of planting material to be shared with the national program. So this is, I think, what we plan in terms of from my side. Maybe we can also get from each of the countries.
Thank you, Dr. Aksat. In Côte d'Ivoire, CNRA, we are with you in your objective, new objectives. But I think we have to insist on 4 points for Côte d'Ivoire. This is, we are in climate change time. And I think we have not enough work on the tolerance or resistance of the variety on during the climate change, flood or drought. So I think you have to work on it. In Côte d'Ivoire, you want to also focus on CDM multiplication, how to increase our capacity in food CDM production to have our variety being utilized by producers. We also have flooring physiology. We have been talking about the physiology of yam flooring, and I think we don't have enough work on it. We have started in Côte d'Ivoire, and we have to do more on it. Of course, we don't forget the food quality. I think these are the 4 points Côte d'Ivoire is going to focus on for the next years. Thank you.
Okay.
Okay. Jude, okay.
Hello. It's as if Amani was reading from my script. So I wouldn't bother you, but then we've listed almost the same stuff. There's going to be a new phase of collaboration with the food systems resilience program. It's coming on next year, and all these activities have been listed. The sea systems, we are doing very well, and the biotech laboratory is at the forefront. So we don't have problem there. Just we get the material and then we can go ahead with it. The SAH is in place and it's also working, and we have dedicated one scientist to that. We've have 2 new breeders which have been joined, who are joined the program permanently, recruited by the government of Ghana. So we are at a better place to perform than previously. That's what I can see.
Okay, thank you.
Thank you, Asrat. Number one, I'm sure before the end of the year, we'll be able to conclude on the release and registration of our 5 varieties. It's almost at the point of registration. And so we will ensure that we multiply the varieties, and then also through the established yam seed companies in the north, in the southern part of the country, we are able to get the varieties to the farmers. We have also established our SH, as we mentioned yesterday. We'll try to use the facility to multiply breeding material so that we can increase the number of test locations. The challenge we have with the SH is that we want to actually plant materials that are coming from the tissue culture. Unfortunately, even though we have a tissue culture facility where we do not have an autoclave that will help us to be able to do negotiation. So like I mentioned yesterday, we still depend on the Crop Research Institute. So maybe an autoclave will help us to be able to achieve this objective. We still carry on our breeding programs, clonal evaluation, advanced breeding on farm, and we'll continue with that. And hopefully, again, like I said, next year we should be able to get some very good materials released. But one thing that was mentioned at the beginning of this phase is about this track or the training. Yesterday and today, some presenters mentioned that we have not had that opportunity of track or training. So I will want to maybe bring that on board so that if it's possible, we are also trained so that we can use the tool to be able to carry out our the on-farm trials. So in a nutshell, this is what I have. Things are going on well, and we hope to improve. Thank you.
Okay, thank you.
Yes, thank you, Dr. Asrat. In Benin Republic, as you know, we have started to evaluate the regional, to do the regional evaluation with the clone that you sent. The first round was this year, last year, let me say that. So now we are repeating it now so that we will have a better result to assemble data and put it in the YAM database. We also have our best clone that we are evaluating. We will continue to evaluate that. Yes, the tricot approach, we have started with 14 villages. It allows us to identify those farmers that will continue to evaluate the YAM with us. But this year, we will go for a second round now to have at least data for 2 years before we start concluding on the varieties that are best for this and these localities. What is very, very important in this tricot is that when you put in place your experiment with them, it allows the farmer to quickly get the materials that are good from it. You see, that is very, very important. And we will try to formally install the group of farmers for the evaluation, including the certain number of women that you said. Yeah, like Dr. Amani said, in terms of breeding, and when we see the result from the survey, climate change aspect, drought tolerant, was raised everywhere. I think we have to think on that, on how we can find some material that are tolerant to drive. The seed meat multiplication aspect, we should seriously consider it. With the SAH issue, we will try this year to improve the work at that level. But you also said that we should introduce 10 best market varieties into SH. So some of those materials are not registered in the catalog. But if, like we are talking about a private company, if they are not released or registered in the catalog, the private company will not be able to produce seed and sell. So we will try to work with the, how do you call it, the registration committee to have all those names inside. I know Laboko is already registered, the varieties such as Morocco already, but there are some. Yeah. And one thing that is really important for us, apart from what we raised today, and it is also what you are saying. You know, it's something that I raised and 2 years after the beginning of the project. And it seems that we have to do it. And it's exactly what we are saying now. We have some best genotype in Ghana, in Nigeria, in Cote d'Ivoire, even in Benin also. And apart from the yam, AfricaYam, farmers used to exchange material from the countries. And when we find it in the system, we just say, there is a very good variety here, we go and collect. Why not put together what Nigeria has, what Ghana has as a best, even if it's 10 or 5 best of best, and put together, and that set of material is evaluated in the different country, exactly like you are doing for the regional evaluation, and it allows us to quickly find some good genotype for the farmers along our breeding scheme. You raise it right now, and I find that I think that is a very important thing. So for the other things, we continue the crossing, of course, and other small activities that we used to do. That is what I can say for Benin Republic. Thank you.
Okay, thank you, Professor Dancy.
Thank you very much, Astrat. For National Root Crop Research Institute, Umudike, for the year in question, we continue the normal breeding scheme, the establishment of the crossing block. And in this area, we intend to have more training of personnel, especially to improve our crossing efficiency and our results because of exchange of staff and some staff that have left the system. So we need to increase the manpower. Some training has actually been done before and we picked up and we lost quite some of the personnel, and this we need to reinvigorate. Of course, our normal seedling evaluation will go on, all the breeding stage gates from clonal evaluation to preliminary, advanced, and national performance trial. And within the year, what is very significant for us is that we will be going back to the National Varietal Release Committee because of the candidates clones that we showed during the presentation. We'll have to pass through defense on this panel. And we are very optimistic that they have shown some potentials already, and we have to give them that status of variety by the end of the year. For our SAH, we intend to collect more land races within our area and environment, and to complement, we clean it up with TC, tissue culture, and have them as into the SAH platform. And we increase our production of already released varieties as well using the platform. And now based on the presentations that we had today, and we will agree with me that RTB has actually produced a lot of protocols that we can uptake in the breeding stage gate. And I think the results so far has shown and that the synergy when we work with these outputs from RTB. So for our evaluations at the advanced stages of selection, or most of these tools will be progressed in a breeding pipeline. And something was said about the farming of yam in bags, which is becoming very popular within the urban setup where there's limited land opportunity. And I think as a responsible institute, we have to optimize this production system, what could be the merits and demerits at the moment, and how can we improve the system and add value to the production, since it's becoming a very popular, even in my area, almost every family is having, in the town is having yam in bag, growing like horticultural crops or flowers. So I think we can improve the system for farmers by driving some innovation in the system that can boost production. I think in a nutshell, that is a major focus for us for the year. Thank you.
Okay, thank you. Ifsu.
Okay, thank you. For us, we will continue with repeating the regional variety trials that we had last year. And then we will also go on with our crossing block, and the clones that have been generated from previous crosses. We'll follow up with the process of evaluating them preliminary in order to know which of the clones will follow up the breeding process to PYT and other ones. And then concerning a survey on farmer preferred traits that we are doing in collaboration with NRCRI, we will continue on that. So basically, that's what we will go on to do. You know that we are always a younger brother to NRCRI, so our activities are not as elaborate as other stations. Thank you.
Okay, thank you. But I have one question to Jude. Before that, for our new partners, maybe want to hear what you are thinking about if we go to your place.
Yeah, thank you, Asrat. And first of all, I really want to thank AfricaYam, especially you and Patrick. After you visited us, you gave an indication of giving us support. We are very grateful for that. I should say this on behalf of the other countries that will come on board. Yeah, allow me just take 2 minutes to say that we started our work on yam actively some 5 years ago after picking on previous efforts. And this was driven by commitment from our bosses in the country that we should position yam as one of those crops that can supplement cassava, sweet potato, where it is common. So all we aimed at was uplifting, starting really a functional breeding program, and the germplasm that we received. Basically, the last 3 years we have been phenotyping to see diversity, but also we now started some kind of active crossing program. This year we are running a crossing nursery, and hopefully to take the pipeline forward. And now when you mentioned that there could be more advanced germplasm we can test and see their usefulness, that would be a good entry point for us. This year we did farmer preference assessment using the 1,000 Minds tool. That result will be shared with everyone. What indeed is lacking is what you mentioned rightly, how different or similar are the pathogens in East Africa compared to where yam is popular. That is knowledge gap that we feel can help us move forward. So the 4 points you outlined for us as entry points are really nice. And we hope that we can have support around the SAH technology. I know for cassava and the next gen, we initiated that, and I'm sure AfricaYam can help us supplement that. But we are there. We want to be an active yam breeding program. Thank you.
Okay, thank you, William. Jude, I think we always think about the Okokoro product profile. And my feeling is this one is more of within Nigeria. So the national program maybe has more of mandate to address that smaller volume of the market, which is very important, I think, in Nigeria. So do you have any plan to address this? The Okokoro? That's the dry yam.
So for which products?
Yes, the variety, this one. It produces multi-tuber. Yes.
It's not. Yeah, at the moment, the two major product profile is boiled and pounded yam. But I know the amala, it's more of a Southwestern food system. And I know we have a national mandate on yam research, but most times we are marked to the food system in the Southeast agroecology. But I think we can expand on that and look into that. It's a national assignment and we have to deliver on that policy. So we can always discuss more on that and see how we can introduce materials towards that threat. And I know my good friend, Professor Tebayo, she's passionate about amala, and we've discussed about it some time ago, and I think we can take it up further. Thank you.
Yeah, the Ikokoro is a land race popular to the southwest, and it is used for amala because it is multiple tubering, 5, 6, 7 tubers per stand. And the tubers are not so big, but they are of reasonable size, say average size tubers, 5, 6, 7. And it is processed whole tubers and dried and then made into flour for amala. So I think it's peculiar to the Southwest. Yeah. And even been,
yeah, I think that's one of the product we see, but if the national program is not ready, maybe we can supply some of the materials which can address those, but think about how we can come up with one of us pipe product profile for that.
Mine is just to give a small precision. Ikokoro is not a variety, it's a complex of variety. Within Ikokoro, you have so many varieties, so many genotypes inside. Some are highly productive in terms of number of tuber and the total weight, and some produce low. So first thing to do is to find the genotype that are that produce high, and start from there. So it's a complex of varieties, not.
So I think it's good to know that it's one of the market class for yam. So, and amala is the product, food product, and then how it can be addressed with that.
Thank you very much. I know kokoro, as Dr. has said, is a complex. The kokoro that you're talking about, that at least I know about 3 varieties: Ehobia, Korondo, and I think 1/3 one. Those ones are highly priced for amala. So it's not just, that kokoro is not just a variety. They have about, and they call them kokoro because of their peculiar shape and because they are hard and they have some characteristics that makes them to be very good for amala. So that's why they call them kokoro, but it's a complex of varieties. Ikokoro is round, short yam, like milked yam, like this. I mean, like seed yam. Yeah, very slim and all that. Thank you.
The main quality of ikokoro why it's acceptable is you can store it for a longer period.
So I think we have to look at that market class for yam in Nigeria and maybe some part of Benin. So that product is available in the market. I'll come here. And also we are thinking in the next transition phase as shelf life as one of our focus area. So maybe I can also, Dr. Busi.
So now, let me just indicate that I am completely confused. I'm completely confused. So now I am between, I hear it's a group of varieties which got that name Ikokoro, but the food product coming out of those group of varieties is amala. So if I work with Astrat, I now screen the varieties for amala. And then I will have satisfied the issue of Ikokore so that we are just clear what we are supposed to do.
I think that's right. The class of the varieties is Ikokoro, but the product is amala. So we have to breed for the amala quality. I think that's maybe we need to discuss with in our Nigerian team how we're going to address this one the next 2 years. We can generate germplasm. We have some small backup crosses where to generate those materials, but I think we should make it next level of product profile on the breeding program. And maybe, okay, for the others, maybe from BTI, can you have a plan for?
Sorry, what is the question? Sorry. Yeah, so we'll keep working mainly on the tools that we've started developing, and I think we have lots of ideas how we can improve them, and you know we'll continue to provide support and workshops and you know add new features to the database as is needed by the breeders.
Okay, I think maybe I have already for food science. Alamu or Lucy? Yeah, but I think we'll discuss later on. But yeah, what's the plan for the work plan for the next 24 months? Maybe?
I think the first important activity is for the food quality work and the food scientists to be integrated into the different countries we are working with. That will be one activity. The second one, it will be basically to get more out of the equipment which we acquired from RTB Foods, how we can extensively use them to support the screening, the high throughput of the quality work. And further work on the methodologies coming out for texture analysis and the profiles. In the new countries, we would like to just do the survey of what the product quality, the major form of consumption, and the product quality, just like we did with the other countries, but using the methodology we developed in RTB, NATRIB foods. I think that is enough for 2 years.
Yes, I think it is good. So for the next 2 years, we are our work plan. I think we're going to refine it because we are collecting all the results and all the information. But can we be sure that next year, when we are presenting our candidates for release, if they are good for pounding and boiled or whatever other product, can we hear that information from the food science, the national program? Are you ready to integrate the food quality or food science in your team? And instead of the breeder telling this is good for boiling, is it any possibility to do that? Or maybe if next year, if not, then maybe in the next 2 years? For whom it is not possible. If not possible, we can, otherwise we should not commit when we are preparing our plan. Am I clear? I mean, we as a breeding program develop a variety. And at the end, we identify a good one for release. So when we are nominating a good one for release, we say this is good for boiling, this is good for pounding, this is good for yield. So can we really integrate the food science in our evaluation process? And the next time when we meet, so the variety we identified for release, we want to hear if that variety is good for the product, the intended product for the food science. So do we have like such kind of, can we make such kind of arrangement in our team to come up with this result? So we all commit for that. Okay, good. Okay, so I think from my side is okay. Maybe we can pick some questions or maybe other opinion. Patrick?
Yeah, thank you very much, Asras. I think the food scientists have made a statement, they have made a commitment that they really want to work with the various partners in terms of assessment of the food quality of the materials that they are screening. So I'm also sure that the partners are ready to work with the food scientists, not only from their program or from their home country. I think we also have IITA food scientists ready to work with them to assess the food quality of the materials they are working with. For the work plan for next year, we will still be doing the same thing. We are going to the same thing by saying we are going to come out with the work plan of each of the partners. This is what we do every year before we even have before we sign an agreement. So I'm going to sit down with the ASRAT. The two of us will work out the work plan based on the funding that is available for each of the partners. So you will still have it in black and white, and that will form the basis of our assessment of your progress after the year. So it's not only the discussion that will end here. We are going to continue with ASRAT, and we'll put it in black and white for each of the partners. I think with this, I don't know if there are other contributions or other questions. Okay.
Yeah, just a couple of quick comments. First of all, I was glad that Dr. Mani, we finally heard the mention of climate change, which I think is very important. I'm sure that yam is not the only crop in the world which is not affected by climate change. So I think we do need some consideration of that. Pest and disease will be affected by climate change. Of course, you have stresses. Probably, I don't know, but I would guess also that quality is affected. by climate change, and within that then probably needing more knowledge of genotype by environment interaction for many of these traits. And the second thing is we have breeding, we have seed systems work going ahead and going on in Prosiva. So that's two legs, but the third leg of the stool is agronomy. So we asked the question, what is happening in yam agronomy? It's very important if we're really going to realize all the advantages of these varieties, etc., in the field. So I would urge people to think about that. We are very fortunate, let me repeat, as was said right at the start, that we have amongst our midst, and I'm looking around, I hope he's still there, he has his back to me. We have amongst our midst John Derrera, who is a newly appointed Senior Director for Breeding and Pre-Breeding. So he is going to be interacting at that level with the other Senior Directors, including that for agronomy. Excellence in Agronomy is an initiative led by IITA. It's active in West Africa. It's led partially in West Africa by Fred Kazito. So I would encourage you to work with John, to work at whatever level you can, to really think about promoting the need for yam agronomy to be carried out within Excellence in Agronomy or outside Excellence in Agronomy, because there's a lot of work that still needs to be done on the agronomy of yam to relate that to the breeding work, to the quality, to the other aspects that we've been hearing about, and the seed systems. Thanks.
Another comment from
Thank you. I imagine that for developing solid work plans for next year, there will be a remote meeting, Patrick, or you just collect the plans and put them together, because I think it's important that people meet. And I mean, this is a serious planning exercise, okay? We used to have it in person. So that's one thing. The second thing is the advancement event, the product advancement event. I think Ashra mentioned that you guys had it last year. Is it an idea or a wish to have national programs involved and have them also participate in that event next year? That's a question that don't need to be answered, but I suggest that you guys do so. Going back to what Michael have said about climate change, I think there's no discussion that something is going to change. But the question is, when we develop a product profile, including climate change, we need to make sure that we know what's going to happen, which is we don't have a crystal ball. We think it's going to happen this, we think it's going to happen this. But if we're not sure, breeding yams for certain attributes takes many, many, many years. So we need to start thinking, okay, or do we really need to develop new varieties, or we project that the varieties that are developed in southern or in northern Nigeria are going to migrate south, for example, or things like that? Because sometimes we don't need to develop new things. It's just a matter of projecting and, okay, we're going to move this here, this here. I hear a lot about that, Michael, and I'm not against the concept of climate change. What I'm in favor is, let's make sure we know what we're going to do, because climate change is something that is so broad, and breeders are very good at addressing specific things, not broad things. It's just a warning. We're going to talk more about when we do the recommendation. Thank you.
Thank you very much, Carlos. For the product advancement meeting, we plan for the product advancement every year. And I think next year is not going to be different. And we usually also invite all the national programs to participate. So it will be in our plan to hold that meeting also next year. There's one other question you I've forgotten the first comment you make. Maybe something about I've forgotten the first issue that you raised, but maybe when it comes to my mind, I'll come back. With this, if there's, yeah, that reminds me. I will do that mostly, most of the time we do that virtually. We organize a meeting, you know, among ourselves, inviting all the national partners, and we draw up the work plan. But if you want us to share the work plan with you, we can do that. It's surely an annual event where we don't usually meet physically, but we do it virtually and then prepare the work plan. Maybe after the work plan this year, we can then share with our TAC members to let them see it and then maybe make advice or comments on it. In the absence of any question, I think I will hand over to Agri.
Thanks so much for the valuable contribution from all of you, including our national partners. Then the suggestion from the different scientists in the room. So our next activity is we are going now to get a recommendation from our TAC member, and also what is the fate of let's say AfricaYam, and then also the transition into the 1CG. And then I'm going to call on, I think is Carlos, that will lead the presentation. And thanks so much. Yes. On the flash? Where? The flash? Okay. Yeah. Don't worry, you can get it. What is the name? AfricaYam. AfricaYam. So, okay, good. Sorry. Okay. Okay.
So, hi. I hope you guys understand that we have been putting this together sort of in between sessions and in a very short time. So understand that when we present is sometimes it may not be that organized, but between David, Noel, myself, and a bit of contribution from Eric, who has not been able to attend or participate much because of some personal problems. David Marshall is sort of the lead in this technical advisory committee, and he's online, and he is going to present, open up the presentation. David, I can move the slides for you, okay?
Okay, thank you.
Hold on a second, we cannot hear you here.
Hello?
Can you speak, David?
Hello.
Yes. Now we can hear him well in the back. Yes? Okay. Go ahead, David.
Okay. First of all, at a personal level, I'd like to apologize for the fact that I'm not with you. I very much regret that, but because of some health issues, I'm not able to travel. The first thing I'd like to say is that very much as a group, we're impressed by the progress that's been made. In particular, when we look back to the very start of this project, we see very real progress. And we're delighted to see the yam breeding groups across West Africa and potentially beyond that coming together as a very real community of practice in the very real sense. And I think it's also important to acknowledge that over the last 2 or 3 years, this progress has been made in the face of very real difficulties due to the impact of COVID. And one item that I can particularly comment on, it's very good to see the development and progress of a younger cohort of scientists, which we feel is very important for the future of yam in West Africa. Next. Okay, so let's get down to some details. And I should acknowledge, as Carlos said, is that we've been putting these things together as we went along. So some of the things may also be slightly redundant because we've learned something else as we've gone along. But to say that it's very apparent there's been substantial investment in yam genotyping and considerable progress. And this has utilized quite a variety of technologies. So from what I can see, there's dark genotyping, there's GBS, there's whole genome sequencing based genotyping and target genotyping for candidate genes or QTL. But one issue I think it's really important when you've got this variety of genotyping technologies, I think it's going to be quite important to try and look towards bringing this data together. And really, I think increasingly that's going to be possible on a genome or a pan-genome framework. So you can actually cross-relate everything and every piece of information. I know it's quite difficult to do in terms of bringing together genotyping technologies, but I think this is something you really need to start thinking about. I think it's also important that this data should be integrated with the sequence data that's going to come from the Illumina investment in YAM sequencing. I'm sort of surprised that given that Illumina are making a major contribution to YAM sequencing, we haven't heard anything about this in this project. I think this is, again, has a potential to be quite a major game changer, and it's important it's taken into account in the planning framework going forward. There's going to be a lot of, really quite good quality sequence data for YAM. The importance of developing an understanding of and the technologies to deal with sporadic flowering and expression of sex are a given priority. I mean, we've heard about this as its importance literally since the start of the project. It's often a major limitation, and we hear about it all the way through. And we can acknowledge the progress of the work in Japan on sex determination, but I think it's really important to emphasize the importance of this area, in particular in terms of the reliable induction of flowering. You don't want to be in a situation where there's the crosses you really want to make and you can't make them. So again, looking to find partners in this area, I think is going to be very important for YAM going forward. We also believe there's a need to do research in terms of tuber dormancy and tuber wound response. And this may require the recruitment of a plant physiology, either in-house or in partnership. I'm particularly glad to see the work on dissecting key traits, determining quality, and developing technologies to screen for quality. It's incredibly important and it's welcomed. But given the limitation in the number of samples that can be processed, it's important to identify where in the breeding cycle that these technologies can be most effectively deployed, and to explore the potential of higher throughput technologies such as NIR. And it's important that these standardized methods can be recognized at every stage in the value chain. So it's not just in terms of the breeding. You want, if you're going to use these technologies and come up with figures for a given technology, what that says about YAM or a particular line of YAM, it's important, for example, that a processing organization knows and understands that as well. So you need to have this identified all the way through and standardized all the way through the value chain. I can see from the talk that was given that breed base in general and its implementation in yam base has had significant improvements. But it's really important that the data enters there from all the programs in a timely manner if breeding decisions are being made using the capability of the system. And again, Carlos was talking about, product advancement events. You really want to be sure that if you're going to be making decisions about what to cross and what to take forward, either as parents or potentially as varieties, that you have the up-to-date information at your fingertips. So this means that data really has to get in there quickly, and it's got to be cleaned and available. I mean, certainly I know from being at similar meetings with commercial breeders, they want all the data at their fingertips because these are big decisions that you make, and you want the data to be there. You don't want to be scrabbling around and saying, "Oh yeah, just how did this particular line do? I'm sure somebody said it had a problem," or something like that. You want the data there, and you want the data there in an accessible format. Next slide.
Thank you, David. I'm going to take now the part of plant breeding. I think I need to commend we need to commend the fact that the breeding cycle has been shortened, particularly at IITA with the use of molecular marker technologies. But we also need to think that this needs to translate also in national programs having shorter cycles, because they also need to generate diversity and to select it in a shorter cycle. Okay. As well as I was last week in the Next Gen meeting, and I was expecting to see the same here, that the development of elite materials is entering sort of an exponential phase after 6-7 years of work. And we should expect a greater number of materials, of elite materials, being promoted to later stages, okay? And somebody mentioned very rightly so that a tracking system for recording adoption and measuring adoption and feedback is extremely important, okay? We hear in the cassava meeting that we cannot promote too many varieties. I think farmers are willing to receive diversity, incorporate it, look at it, and accept it or reject it as it fits to their systems. So I think it's always important to really have elite materials that have the quality attributes, they have the agronomic attributes, and expose them to the final users, okay? We see a great adoption of the SAH system for rapid multiplication. And I asked the question, how much are we paying for this system? And not knowing, but knowing that there are alternative systems, I'm sort of kind of urging to at least look into alternative systems, like the Cubans and the Dutch have developing very interesting systems that are being tested in Brazil now for sugarcane, for example, okay? One of the things that I particularly have been very keen is to include farmers' preferred varieties in the cleaning and production of good seed, because those are varieties they know, and it can open the door for them to accept other technologies like other varieties, okay? I have a story of working at Syngenta, what happens in Syngenta in India with corn. We decided to develop three-way crosses. You guys know, if you're in the US or in Brazil, Argentina, it's all single crosses. 2 inverse cross is the optimal system, okay? Three-way crosses are a thing of the past. What happened is that these farmers, because three-way cross seed is much, much cheaper and is higher performance than their open pollinated varieties, they adopted the three-way cross hybrids, their incomes raised, and after that, they started looking at F1 hybrids, which are much, much more expensive, but they generated the base income to adopt that, okay? So it can be similarly said with things like this, offering them Laboque, for example, with very good seed quality and a management system that Laboque can go from, I don't know, 5-6 tons to 12 or something like that. Michael mentioned agronomy. I'm a breeder. I'm very interested in the G, but I'm more interested in optimizing the G by E, okay? I think we really need to be conscious of this. We can go so far through genetic improvement, okay? And we talk in the video, we saw that the yield gap is pretty broad. Patrick was saying in the video, current yields is around 10 tons per hectare, but it is possible to reach 60. That's a big gap. So if we can get 60 or let's say 40 tons right now, it means that we have the genetics for that. So if we keep working on genetics and we improve 5% of the yield, can you imagine where the yield gap is going to go? Probably the yield gap may expand. So we need to think and stress the need, and John, very rightly so, Michael pointed at you that the interaction with the excellence in agronomy is essential, okay? Our goal is not that just to generate better varieties. Our goal is that yam farmers and the whole society that goes around yams improves, okay? So again, we talk about the relevance of generating knowledge that is lacking for yams in areas like flowering, dormancy, wound response, or shelf life. I think it's extremely important to engage institutions in the developed world. I'm working the last time I was here, I was working for Syngenta. I couldn't do much for yams. Now I'm at a university. So one of the things that I'm planning to do is to introduce a small set of clones. I'm talking with Asrat, talking with Michael, that we can use those as a basis for our physiologists, our scientists, not only at NC State, but other universities as a base to do some studies, okay, and help. We hear about the future expansion to other countries. I think it's going to generate a lot of opportunities in terms of genetic diversity and testing sites. But please be mindful when you move germplasm. Be careful. We hear this morning it's very difficult to clean yams, okay? Eastern Africa may not have problems that we have in West Africa, okay? Our experience with people moving germplasm from Africa to Asia has not been very good. We introduced CMD into Africa, okay? Which is a serious problem, okay? So be mindful of that, okay? Again, great progress in introducing quality traits. I think I'm repeating David here. We need to make sure that we develop tools that can be applied at early stages in very high throughput, okay? About capacity building and NARS, we commend the great number of student researchers and other professionals that have been trained in the infrastructure that has been developed at national program. I think it's from when we started is day and night, okay? Noel was pointing out about that he thinks that SOPs for SAH are not uniform across all national programs, and that's something that can be worked out very easily, okay? And also the need to deepen the food quality assessment when we release new varieties and make sure that, as Michael was saying, how much flexibility we have with that variety for pounded yam, for boiled yam, for different uses, okay? Finally, looking at the video that was produced by AfricaYam, we pointed out before that there needs to be a greater level of recognition and highlighting of what the national programs are doing within this project, okay? because in that, okay, you guys at the national program will feel more part of this family, and also will highlight the potential of a project like this to bring benefits to all the countries. So I like the video, don't get me wrong, but next one, let's make sure we have a lot of people from the national programs showcasing what they are doing, okay? Going back to you, David, for the closing slides.
Okay, really just to say a couple of things.
Oh no, hold on. Sorry, David, this one is mine too. I know you were looking at the slide and say, what the hell is this? Yeah, this was the slide that Eric was supposed to present, but he apologized because he has some personal conflicts, okay? And these are a couple of thoughts from Eric. Let's take a look about the engagement with national programs and how can we make it better, okay? And also given, again, what Michael was saying, given the challenges that yam production will face, climate change being probably a major one, the need to be more efficient and effective in bringing technologies that develop products that are more resilient and nutritious is extremely important, including gene editing, okay? Talking with David, we think that gene editing may be applicable for flowering traits because what we're seeing that Dr. Terauchi presented today, it may not be that difficult to manipulate that trait if we want to. Traits for resiliency are a bit more difficult, okay? So I think the last point I wanted to make is that yams, and we say this, and forgive me if you guys think of what I'm going to say is not appropriate, but yam is sexy, okay? For us in the developed world, it is sexy. There's a lot of institutions that would like to work with yam and help, okay? If you think that the value of yams is as much or higher than the value of cassava in Africa, and if you think that the value of the farm gate production of yam is higher, is like 20 or 30% higher than the whole wheat population in North America, the wheat production in North America. Think about this. You have a crop that has higher value than wheat in North America. Do you know how much resources are invested in research in wheat from the public and the private sector in North America? It's amazing. We should expect, probably not as much, but we should expect some investment both from public and private sector, okay? Finally, one thing that I wanted to mention before I hand it over to David is we are in a transition process, well known. Resources are going to be available to enable this transition for a certain time. I particularly want to stress and warn you, let's not be complacent. As I said, yam is very sexy for institutions to work, institutions to develop joint proposals for specific topics like dormancy, flowering, wound response. Let's work on that. Let's not just leave, oh, we have resources for transition, forget about it. No, this is the moment that we need to seize and develop ideas. And if you need help from us, we'll help you, okay? David, now I'll go to you for the finances.
Okay, before I talk about this directly, I'd just like to emphasize what Carlos has just said. I mean, I think the potential of yam is great, but again, I think the knowledge of the potential of yam is not great. So there are two things that need to be done here. I think you need to advertise it, and you need to find ways of letting the, as it were, people in Europe or North America understand why it's such an attractive thing to work for. And it also point out, for example, in the UK, although there's been changes in terms of funding that can go to the developing parts of the world, there is actually quite a lot of money, in particular if you can involve companies in it. So there is money for joint research from UK organizations, both companies and academic organizations, and at least some companies in Africa. So there are a number of funding schemes that are available, and it's a very timely to be able to looking at these opportunities now. Okay, just really a couple of quick points I really want to make at the end. Really, one is again this whole issue about when you're producing varietal material from this project. I think you want to really find ways of being able to advertise it. I mean, perhaps prefix it with something that identifies it's come out of AfricaYam. And in terms of developing publicity material, I mean, I know, for example, for companies in the UK, when you release a new variety, you release a PDF of 2 or 3 pages that describe that variety, compare it with existing varieties, highlight why it's as good as this variety in some ways, but it's better than this variety because of this, and a few visual things about it. And you also use the trade press and other things to be able to advertise it. And I think this is really, so you need to be able to identify it's yours and develop your brand, as it were, as a trusted brand. But also you need to advertise the material and advertise, why you should choose something from this project as opposed to something that's been around for a while. I also want to really, I suppose, add, a comment that fits in with what Carlos was saying, is that I think it would be interesting to know what the impact of the funding from the Gates Foundation has done on regional investment in yam through the NARS. Ideally, it would have acted as a seed to attract further funding. And it's important for research and varietal development that this needs to be recognized. So you want to be able to ideally leverage the Gates funding to get more funding, not just simply to rely on the Gates funding. And I think this is incredibly important because you really need that to broadening your funding to have several sources gives you resilience. And I think resilience in funding is incredibly important for breeding. You don't want to be always in a 2 or 3 year cycle of thinking, the funding's going to end there. What's going to happen next? Because it's important for retaining material resources and also people that you have some form of resilience in terms of your funding. Okay. And just we wanted to say thank you. And because it's been raised about yam in the UK, this is a picture of yam in the market just beside where my son lives in London. You can just walk along the street and yams are there. I would also say they're yams from Ghana. I've also added a thing to say that if I want yams delivered to my house, I can just dial them up on Amazon and get yams delivered to my house as well. Okay, thank you very much. And again, we'd be happy to answer any questions that any of you might have.
Thank you from my side also, and I echo also Noel and Eric's. Thank you, because we learned a lot. Well, I think that's the most important part. I hope we have contributed something, and I hope we can continue. I mean, don't hesitate to knock at our doors, and we'll certainly be willing to help, continue helping.
Thanks so much for those comments coming from the TAC committee. We do really appreciate your feedback about what we are already doing, and also about what needs to be done for the future of the yam, and then how we can move the yam from where we are now to the next stage. We do really appreciate them. I'm so happy when Carlo said that anytime we need some help, we can just jump on them. So that is good for us. It doesn't mean anytime we have some issue, alongside with the team, we know where to go and then what to do. So feel free, they are more open than what we think, and then they can help us as much as we can. Sorry, Marshall. Hello, Marshall. David. Yeah, it's good. No problem.
Yes.
Okay, good. I just want to call on the Ghanaian team to confirm if the picture that is there is from Ghana or is modified yam variety. Okay, good. So you are free then. Thanks so much. And then any other question from contribution? Yes.
Okay, thank you very much for the TAC members. I think very important recommendations raised. Maybe I want to get your opinion about one of the area really I think as a game changer if we make it in yam. We are trying in the breeding program to develop the change the growth habit of yam to come with bushy type or dwarf yam or maybe other or less tendency to climb yam. We did some collaboration with SLU, it's a Swedish group, to come up with some project in order to understand the expression with steak and non-steak, and then for RNA sake and all those kind of molecular analysis. But we applied for 3 years. I think the current one is we have not had the result. But for the last 2 years, it was not successful. And I want to get your opinion or maybe your help to link us with people who can really walk into the deep on this area. And in tomato, there are even some genes. Gene editing will be also a good area in order to really make yam more suitable for mechanization and for open field cultivation.
David, I'm going to speak first and then you can jump in, or Noel.
Yeah, sure.
I had the pleasure this is the first time I come to a yam meeting and I see yam in the field. Because we always came here in February, March, and we saw yams in the market, okay? So I had the chance to, I insisted Ashra to show us the staking yams, which is, I think it's an amazing concept. I think we always think about our farmers here in Africa as being conventional farmers, and we need to develop technologies for them to lift them. And I think that's no doubt, but we also need to think of the future of Africa, the rate of urbanization, the fact that younger generations don't want to dig 10, 15 minutes yam tubers, okay? A lot of them don't want to do it. So we need to think of how we modernize and make it more efficient for the use of resources. Staking is one aspect. I think planting is the other aspect. I'm happy to hear all the options that we have. Talking with Jim about what is being done in potatoes by companies like Simplot, that they went away from tubers to sprouting stems in big control environment factories. Okay. I'm not saying we're going to reach that level, but we need to dream about how yam is going to be produced in the future. Okay. You know, guys, I look forward by the end of the century, the population that's going to be in Africa is going to be about 3 times what is now. We are sitting here in soils that are not as fertile as South American soils where I come from. So we're going to have a challenge, okay? And we need to think about the ways that we're going to produce crops like yams, not only in terms of non-staking, potential mechanization, but also what kind of cropping system in terms of rotations, in terms of management practices. Maybe biologicals will play a role in that future. We don't know. David or Noel, any comments on that?
I mean, again, a very more specific comment. I mean, if you want to modify the shape and style of the plant, I mean, certainly in a number of other systems, there are identifiable genes that can help to do that. So you may have actual targets you could look towards. And again, if those also may be good targets for gene editing.
Thank you. I have no comment on this question, but I have an additional comment on our reports. I heard that people here want that trichote data should be integrated in the YAM basis. I don't know if the man in the charge of the YAM basis can do this.
Yeah, a quick comment from the TAC is to see how possible we can adopt the trichote approach into the YAM base.
Yes, I think it's possible. I think it's not even that hard. It just hasn't happened yet because of different reasons. But I think we'll work with a very high priority on this in the next few months.
Thanks so much, Lucas. Okay. Any other comments from or suggestion on area to focus more? I think there's no additional comment. The next point is we are going to receive a suggestion, a point of view from Jim. So I will call him to give us his remark on what to do.
Thank you, Agre. And let's thank the TAC. So they've invested a lot of thought. It's been difficult for them also through the COVID period. And, you know, you've got 2 here in person, 1 online, and I'm one with some conflicts, but let's thank the TAC because they've done a fantastic job. Thank you, Carlos, Noel, David, Eric. And they're available not only for the meeting, but as mentors and people who can help lead the way or find resources or people with expertise and make connections. So don't be afraid to call on them. Now, my problem is they've pretty much covered the things that I wanted to say. But that means that's good. It means we're hearing the same things, we're seeing the same things, and I really value and prize their wisdom. So it is a transition. But as we transition towards a new investment in RTB breeding, I just want to remind you that some of those elements of working together as RTBs are already embedded in the program. We have RTB foods, in which many are engaged. We have Breed Base, which has been doing a fantastic job of keeping up with breeders and developing the tools and ontologies and doing what it takes to be able to use new things that you will be needing, whether it's new multidimensional data sets or new ways of new experimental designs, analyses, mixed models. They've been, I mean, it's a privilege for you to work with developers who are so responsive. So thanks to the breed-based development team led by Lucas and with everybody engaged, they really are serving the RTB community well. So some of the commonalities that have come out, I mean, there's one thing that I think is just crying out, and that is YAM and RTBs are in desperate need of good physiologists. You know, we've heard about specific things, dormancy, flowering, but as you look at how to optimize mini tuber production, you know, how do you optimize sequencing and bulking of mini tubers? What about curing mini tubers to be able to store them until you need them? Or in general, curing seed, shelf life. There are many, many needs that are unaddressed besides just flowering and dormancy. And YIFSWA or ProSiva very much needs the ability to further optimize their systems, to make them efficient and productive and cost-effective and enable value chains, seed value chains that really will work and can be taken up because people can make money with them. There's tremendous need for further optimization. One thing that didn't come up just now, you know, I spoke a little bit with Michael about why the gene bank only has clonal material, not seed. And then someone else said, well, what about pollen as a means of storing genes? This isn't the first time it's come up, but for the breeders, what about pollen storage? Because one of the ways to deal with lack of synchrony in your male and your female flowers is to be able to go to your cold storage and pull out the pollen for the cross you need when the female flowers are there. So again, I ask the question, what about pollen storage? Can that help your breeding programs make the crosses that you target? Carlos already mentioned, with new opportunities, new partners, new seed exchange or germplasm exchange comes risks. And along with programs with reputations to protect, the Gates Foundation also doesn't want to be associated with moving pests or pathogens. So please, as you think about collections, maybe a point of reminder, I usually remind national programs that their national germplasm collections are also their national pest collections, national virus collections, national pathogen collections, because that's what happens if you collect without cleaning. But as you look at bringing in new germplasm, sending out valuable germplasm, please be very, very careful that diseases, viruses, pests don't go with them, the germplasm. I wanted to maybe end where the text started. And I think about the history of this project. And I can say through the first phase, through the beginning of the second phase, the seed cycle was a major problem that really hindered acceleration. It's hard to go fast in breeding if it takes you 3 years to get big enough seed and enough seed to be able to have good plot data. And I want to commend ASRA for being innovative, being able to grab onto technologies. The rapid multiplication of seed wasn't a new idea, but actually implementing rapid multiplication of seed to impact breeding was. And now I can say that I can really see the acceleration in breeding with AfricaYam. And that's moved you up in breeding modernization, maybe almost as much as some of the tools coming from EIB or modern breeding program have, because the seed system was your bottleneck, and you're making big strides in dealing with that. You can make it still better, but I'd like to applaud you for that good progress. And this really is a tremendous community. The national programs that are here already, the international programs. This is a truly valuable community that has brought together a lot of knowledge, made a lot of scientific progress, made it possible, or is making it possible, for breeders to deliver varieties that will have the end user traits that are demanded to be adopted and achieve widespread impact. If you look back, it's fantastic. I think if you look forward, there's tremendous potential. And, you know, as things go, I also wanted to end where the TAC ended, saying yam is maybe the most valuable crop in Africa. There should be tremendous investment in this crop. There should be support from national governments. And this is a community that can take that message home. And you have more progress to demonstrate. So not only the value of the crop, but the value that breeding can deliver, and the need for the supporting disciplines to make it even more valuable and make it stronger value chain. So your future is potentially extremely bright, but I think it's good for you to develop that messaging and be your own champions, to look for more funds in country, more recognition in country. I mean, what country in Africa is richest in terms of the size of the economy? This one is number one or number two, depending on how exchange rates go, right? Nigeria, where is your investment in yam research? What can we do to work with commodity groups? This crop generates a lot of money. What can be done to make the government realize the opportunity that lies before them for both internal value chains and export value chains? So I'm not only looking at leaders, I'm also hopefully looking at champions. I hope you can really do well in making the case for yam investment. And for those who've seen new permanent researchers come into their programs as a sign of government commitment, congratulations. So I look forward to your future. I hope to stay engaged in your future. And the more you can work as a network and expand your network and bring in other scientific collaborations, the better it will go, because there are many, many scientific opportunities. So thank you all for your hard work, the good progress, and the bright future that you're making for yourselves.
Okay. Thanks so much, Jim. That was so wonderful to hear such comment from you. And then, as you said, we are very young, and then we do our best to take or to do as much as we can to bring up the yam, and then establish a very good network based on what is already there. We do our best to achieve most of your recommendation, if not add more to what you are already expecting. So I think we are coming to the end of the program. So I will call on Dr. Patrick to give us the final remark, and then another application.
Thank you very much, Agre, for helping me out. I want to first appreciate all the participants that attended our meeting. I will first start with Jim. I really appreciate your coming. and your full participation in the program. I don't know what state YAM will be, if not because of Jim. He's been very passionate about the crop, and I can say all what we have achieved so far is partially because of his interest in YAM. We are very grateful, and we want to really appreciate you for supporting us. My appreciation also goes to the TAC members who have been also very helpful. Carlos, thank you for, after having, a long next gen meeting, you still decided to wait to attend our meeting. We do really appreciate. Thank you very much. And for Professor Noel, thank you very much. Even with the very uncomfortable, I can say, type of traveling, you still made it to Abuja. Thank you very much, because we have to send a vehicle to go and pick him in Benin because of some hitches. So I really appreciate your coming and your full participation. Thank you very much. And for the TAC members that were unable to be here physically, I'm sure that they also participate fully online. I want to thank David Marshall. So within a very short period of time, you put up, that type of comments and recommendations to the program. We really do appreciate. We will continue to engage. We'll continue to disturb you. We'll continue to, ask you questions and continue to ask for advice from you. Your comments are very valuable. We are not taking them lightly, and it will help us, going forward in making sure that we do the right thing for the YAM program. For Dr. Abertin and Professor Aderera, thank you very much for coming. This is the first time I will see Professor Abertin to fully participate in the program. Well, anyway, I want to thank you for being with us for the whole 2 days. Your contribution have been very valuable also. We are not taking all your advice very lightly. We have a professional rapporteur that is helping us to put all what have been discussed here together. So we are not going to forget any of the points that you have mentioned. Thanks also to Professor Derera for joining us and participating fully. We do really appreciate. For our partners who also didn't made it to Abuja, Professor Terauchi, I'm not so sure whether you are still online. We want to really thank you. Thank you very much, Professor Terauchi, still online. We do really appreciate. Also, Dr. Lava, who also did not make it to Abuja, we want to say thank you for participating. I think Dr. Badara also, I'm not sure whether you are still online. We want to appreciate you. And for our external invitees, Professor Simon Itwenge, I'm not sure whether he's still around here. Oh, thank you very much for coming and sharing your experiences with us. It's really very helpful. Who else again that have our seed company? Dr. Is it Issa? Okay, it's not here. Okay. We please extend our appreciation to him. We also have the 1 Acre Fund. The lady from 1 Acre Fund, Mrs. Funke, Dr. Mrs. Funke Adewale. We appreciate her coming. And finally, for all our partners that travels from West Africa, from Uganda, thank you very much for coming. And I wish all of you a safe journey back to our destination. For my colleagues from IITA, thank you for your attention and thank you for participating fully. Colleagues from BTI in the USA, Thank you, Lucas, for coming. And for Dominic and the group, Eglanti and the group, thank you very much for attending our meeting. We believe that going forward, the collaboration between AfricaYam and RTB Food is very important. We are taking it seriously, and we'll be working together to make sure that the food quality will be one of the things we'll be focusing more on going forward. Thank you very much. My colleagues in IITA, Dr. Hope, thank you for coming. He is the head of M&E in IITA, Ibadan. I deliberately invited him to be here so that he can also follow us with what we are doing. Who else have I not acknowledged? If I didn't mention your name, please just forgive me. There are many names to mention. So thank you very, very much, everyone, for coming. It's been a very long 2 full days, but very fruitful. And all the discussions are very interesting. Thank you very much. Now, a few announcements.
Okay, sorry, Patrick, to come up and take the microphone, but I know you wouldn't want to thank yourself. So I have taken on the job of thanking you for all your work in organizing this meeting. Please, come on. And also your team at the back there, Paulette, and the other members of the team who have been involved in the logistics, and the Buddy and these guys here on the video displays and everything. Thank you all very much indeed. I think we should also thank Prof. Sani, the head of Abuja Station, who hosted you all. Now, when was it? The day before yesterday. So thank you to this guy who's now filming me as well. Yeah, but Patrick, I wanted just to really make sure you and your team also had your thanks.
Thank you very much for helping me for reminding me about other people that contributed to the success of this program. The communication guys, you are very great. Thank you very much for coming all the way from Ibadan. You've been helping us. And thank you, Michael, for reminding me. My people there, thank you very much. The project admin, the project assistant, Paulette, and our chauffeurs, the people who have been driving all around. Thank you very, very much. We appreciate your contribution. So on this note, I just want to make a few announcements. I passed out, Paulette passed out the itinerary of everyone. We want to know when you are leaving, and maybe the time of your flights, so that we can make necessary arrangement to make sure that we get you to the airport, as soon as possible. You have something to say?
Is it different than the one sent by Canon for next gen? Because I got an.
Yeah, if Canon is organizing that one, maybe it's fine. But for people who are for AfricaYam, only AfricaYam, I think we need your itinerary, because we organize your coming, and we also want to make sure that you get back to your destination, safely. So I don't know the arrangement with Kenan. I don't know. Okay, she's still around.
Yes.
The short two. Okay. That sounds better. So on this note, I want to officially declare the program closed, and we hope to see you next year. I believe this is not the end of our meeting. Hopefully, maybe by next year we'll also meet. It might not be around September. It depends on the way we planned it. But then we hope that the issue of COVID will have been gradually going down. in Nigeria, I think the level of COVID infection has really gone down. And maybe that is why many people have been able to travel. So hopefully next year it will get better and we'll be able to meet again. Probably maybe in another city, it might not be in Ibadan. Thank you very much, everyone. I wish you safe journey back home. Thank you.